very much, and I really want to thank CU Veterinary College for the invitation. It's a joy to be back in Bangkok. I was here first time last year and first actually met Sani Sirdat in person. We've known about each other for many years because we're both hardcore T-cell immunologists working on infectious disease. But we finally got to meet each other, and now we're trying to get a collaboration funded. It's already started, but now we need to get the funders to cooperate. So Sani asked me to give a series of lectures. In the first one, we're going to talk about history of livestock vaccine development. Then we'll go on to new technologies used to analyze how vaccines perform, taking it to a level I think we need to take it in order to respond to all of the new pathogens that explode onto the scene, like Zika virus and porcine diarrhea virus, etc. And then I'll give the last lecture this afternoon talking about some of the research that happens in my laboratory. So the field of vaccinology has been very much driven by veterinary science. I don't think the human medicine people have a real appreciation for how much veterinary science has played in driving the development of vaccine technologies. And then we've gone through this long period where everything was done in mice, and they found out the hard way that none of the mouse data translates to human or large animals, and moved on to even trying to go directly to human, which a lot of people have chosen as their path. But of course, human studies are extremely expensive. You need to recruit patients, you need to get through the ethical committees in your medical schools to do the studies. Whereas in veterinary species, we've developed excellent vaccine design and testing technologies that we can apply very quickly, effectively, and economically. So the first vaccine ever developed was against smallpox, variola virus. It was a horrible disease in the 17, 18, 1900s. Because of its high contagious rate, it would rapidly spread through naive populations. Obviously, there was no treatment back in those times. And then in the early 1700s, Lady Montagu observed what was called variolation in being practiced in the Ottoman Empire. And basically what that is you take the pox lesions of a sick person and you inoculate a healthy person, and if you get it right, they become protected against the disease. But what happened, of course, is they were often under-inoculated, so they weren't protected. And even worse, they were over-inoculated, so you gave them disease. And that's because they had no idea what was in that pox lesion, because viruses hadn't been discovered yet. So they had no knowledge of these filterable elements that came to be known about a century later. But then a very observant Richard Jenner, who was a country doctor, noticed that all of the milkmaids never got smallpox when smallpox came through the population. And he correctly surmised that that's because they were exposed to cowpox, the virus that the cows they milked every day would get. And so that exposure to the cowpox virus, he surmised, was protecting them against smallpox. So to test his idea, he did something that was completely unethical. He took a young boy in the village and inoculated him with cowpox matter. And 2 months later, he came back and he inoculated him with pox matter from a smallpox patient. And that young boy, James Phipps, did not get any disease. And because the cowpox virus was called vaccinia, or the cowpox disease rather was called vaccinia, that was called vaccination because the Latin word for cow was vacca. So that was the field of vaccination being born. For a long time, physicians would debate whether to do variolation or vaccination, and eventually, because of all of the problems with over-inoculation when doing variolation, vaccination became the chosen practice. And it was practiced for years all around the world. And then, of course, modern vaccine formulations came into the picture. The first live attenuated vaccine was that smallpox vaccine. And vaccination became uniform around the world. And in 1980, the WHO declared the eradication of smallpox from the face of the planet. And now there's only a few very well-regulated laboratories that maintain the virus, and otherwise it's been destroyed. The second story I'd like to tell you is another cattle disease, tuberculosis. About the same time, it was well recognized that there was cattle tuberculosis as well as human tuberculosis, but Robert Koch, who was probably one of the premier bacteriologists in the history of the field, not only created Koch's postulates, where you prove the pathogen is the source of the disease, but he was the person who figured out that there were two different bacteria involved, the Mycobacterium tuberculosis that caused human disease, and the Mycobacterium bovis that caused the cattle disease. So of course, after the success of vaccination and using cowpox virus to vaccinate against smallpox, people were very excited about the possibility of doing it again and using the bovine tuberculosis bacterium to vaccinate against human tuberculosis. Unfortunately, humans are just as susceptible to bovis as they are to tuberculosis. So the disease is almost identical whether you're infected with either of these mycobacteria. So that didn't work. But then along came Albert Calmet and Cecil Guerin, who were at different institutes, but then joined together working at the Pasteur in Paris. And they began to develop a system where they cultured Mycobacterium bovis over 200 times over the course of 13 years and kept testing and finally isolated a strain variant that was highly attenuated. And that became known as Bacillus Calmette-Guerin, BCG, and that's the tuberculosis vaccine that's still used today. Use of BCG in the 20th century was marked by a lot of problems. Again, they really didn't have a handle on how much to use. There were problems, especially when vaccinating children, but it's still the best tuberculosis vaccine we have. But with the Mycobacterium story was born yet another field. So cattle have given us the field of vaccination, and now cattle are going to give us the field of adjuvanting vaccines, because one of the best adjuvants available to us in experimental systems is called complete Freund's adjuvant. It's no longer ethical to use it. It creates such a aggressive inflammatory response that the reactogenicity is no longer ethically approved by any of the animal use committees, and it's certainly not used in human vaccines anymore. But what CFA is basically killed mycobacteria in mineral oil. And you put in anything you're trying to vaccinate against, a protein or another bacteria or a killed virus, and you get a really strong response to that antigenic load because of what's in the CFA. Well, it turns out the reason this is such a good adjuvant is because of the ability to stimulate toll-like receptors. So in 1989, Charlie Janeway gave a keynote address at one of the Cold Spring Harbor symposiums on immunology and immune responses. And without any data, Charlie proposed that there was something called pathogen-associated pattern molecules, sorry, that word doesn't belong there, that stimulate innate immune responses. And this is something that Charlie always talked about when he was giving talks about vaccine work, that the dirtier the vaccine, the better it worked. And that's because of the things that were in the vaccine that you hadn't purified out were actually helping by stimulating these nonspecific innate immune responses. And then a very brilliant postdoc, Ruslan Matistov, joined Charlie's lab a few years later, and Ruslan actually was the person who first cloned and identified the mammalian toll-like receptors These molecules were first described in fruit flies in Drosophila and had been studied for years in Drosophila. And what Ruslan did was show that these TLRs, these mammalian TLRs, were actually the receptors for Charlie's proposed PAMPs. And then went on to do a lot of brilliant work showing that the TLR agonists can act as adjuvants via this receptor activation and stimulation of the nonspecific inflammatory response. And it's interesting, some of these TLRs are expressed as cell surface proteins on cells of the immune system. Others are internal membrane proteins, and they combine various nucleic acid agonists. There's other PAMPs like CD14, which is the LPS receptor, and the NODs and the NLRs. And they all stimulate various innate responses, but a large number of them stimulate the interferon response, which is, of course, a lovely nonspecific inflammatory response that can protect you against viruses and even other pathogens. So here's a short list. There's more than this now, but here's a list of the 10 TLRs in mammalian cells. So TLR1 binds lipoprotein, which is a uniquely bacterial product. Peptidoglycan is an agonist for TLR2, again a bacterial product. TLR3 binds double-stranded RNA, which is unique to viruses replicating in infected cells. TLR4 is an LPS receptor, as well as CD14, which is a prominent protein expressed on macrophages. Flagellin, which is the protein that is involved in motility in bacteria, is a TLR5 agonist, and so on down the list. So very interestingly, of all of these TLRs, mycobacteria expresses a whole bunch, 6 of these agonists. So that's why CFA works so well. The molecules present in the killed mycobacteria that's in that adjuvant actually stimulate a large number of these TLRs. So you get an explosive inflammatory response when you use mycobacteria as an adjuvant. Modern technology is trying to use these various agonists in a much more purified form and a limited number. You don't want so many in your adjuvant. You don't want that essentially detrimental response. So let's go back to vaccines. So the next major vaccine that was developed that I'd like to talk about is the vaccine against rinderpest virus. Rinderpest is a disease called cattle plague, a viral disease of cattle, domestic buffalo, and other ungulate species. It's a paramyxovirus, and it has an extremely high mortality rate in these animals. In 1956, after the Second World War, Walter Plowright was a veterinarian in the British Army, and as everyone was discharged from the Army after the war was over, Walter was assigned to the East African Veterinary Organization, Maguga Laboratory in Kenya, and his assignment was to work on rinderpest. And what he developed very early on was the ability to grow the rinderpest in tissue culture cells and do serial passages of the virus. And much like Calmette and Guerin, he passaged the virus in these cells for over 20 years and finally was able to isolate a highly attenuated strain that became the vaccine strain. The vaccine is, because it's a live virus, stimulates both humoral and cellular responses. Modern technology, we now can look back at that vaccine strain and show that there were over 150 mutations in the virus, which is why it was so attenuated. And actually, scientists at Plum Island Animal Disease Center, my institute for 18 years, developed a system to lyophilize or freeze-dry the virus. And this was critical to the eradication program because you no longer needed cold chain to bring vaccine to its target, to the animals they were trying to vaccinate. In the early 1960s, this disease was basically just about to be eradicated, but then it started spreading again, and that confounded a lot of people. in 1983, FAO did an audit on the vaccine stocks, and it showed that only 20% of the vaccine stocks had any efficacy. So basically, 80% of the vaccinations were useless because the vaccine stock had lost its efficacy. In response to that, the African Union, which was a newly formed organization, created what they call the Pan-African Veterinary Vaccine Center, PANVAC. And up until the creation of this, this regulatory agency, any vaccines that you're going to deploy in Africa would need to be registered and tested in either the EU or the UK. Once you got that, you had to go back to each individual African country and get approval to deploy the vaccine. But what happened with the creation of this agency is PANVAC is now responsible for testing efficacy and safety, and it doesn't matter whether you're manufacturing the vaccine in Africa or importing it to Africa. And the other important thing for the vaccine manufacturers, especially the small companies that are now developing in sub-Saharan Africa, is once you're approved by PANVAC, you're approved for all African countries. So that creates a much better regulatory environment for deploying the vaccine. So the eradication program for rinderpest was initiated by WHO and FAO in 1994, and it was declared eradicated on the 25th of May in 2011. So that's the most recent disease that's been eradicated. And again, it was vaccination that got us there. So I'm going to talk a little bit about my favorite virus, foot and mouth disease virus. Of course, this is considered one of the most contagious viruses known. The vaccine for foot and mouth disease was developed directly off of the work done in polio. So both polio and FMD are picornaviruses. And you all know that Saban developed the attenuated strain of polio that became the vaccine strain. And Jonas Salk developed the killed virus vaccine by chemically inactivating virus. And then the problems still today are that whenever you use an attenuated vaccine, especially if it's an RNA virus, which these are, you have a tremendous risk of reverting to virulence. And actually, the present-day polio eradication program has eliminated the attenuated vaccine from the program because the only cases of polio they've seen in some areas are actually vaccine that's reverted to virulence. So they've taken the attenuated vaccine out of the system, and they're only using the killed vaccine now. For the last few cases, they're very close to eradication of polio. So in the 1950s, it was very quickly shown that you could do the same thing with FMD as Salk did with polio, chemical inactivation. And in fact, FMD was the first filterable element ever described in 1897, and that launched the field of virology. So now we have veterinary pathogens have launched now 3 fields: vaccinology, adjuvants, and virology. So like I said, the human medicine people need to get a better appreciation of what the veterinary medicine has done for them. So just quickly, FMD is a single-stranded RNA virus. It's very diverse. There's 7 major serotypes. Within serotypes, there's many subtypes. Strain matching is the key to vaccination in FMD because of the high mutation rate in FMD. And as I said, it's the most contagious virus known. Causes a systemic disease in cloven-hooved animals, which is characterized by high viremia, and it clears very quickly, actually. So this is what it looks like in a cow with the vesicles in the mouth, creates all of this salivation, and that vesicles in between the toes of the hoof. One of the biggest problems with the outbreak in Britain in 2001 was the sheep were the first ones to be infected, and it was a highly attenuated strain for sheep, so you could hardly see the vesicles in these animals, and they were being shipped all over the UK through the various markets. And before they even knew they had an outbreak of FMD, it had been spread all over the country, and that was one of the big problems with that outbreak. As I said, the vaccine was developed by chemical inactivation. The standard method today is biethylamine. Originally, it was grown in cattle tongues, which was very expensive and difficult. So the only major technological advance in foot and mouth disease vaccines was developing a system for growing it in cell suspension and then allowing manufacturers to go to bioreactors, which is the standard way to grow most viral vaccines. So this is what essentially you're doing. So you have the capsid of the virus, the mature virus, which houses the RNA genome, which is linked to the inside of the capsid by VP4 and VPG. And by treating with the biethylamine, you essentially destroy that RNA genome, so you have essentially an empty capsid. But one of the problems with doing vaccine generation that way is to make the doses of vaccine that we need, you have to produce billions and billions of live virus. And of course, when you do that, every now and then there's problems. And in Pirbright in the UK, the vaccine plant leaked, and they had a little outbreak in the area right around the manufacturing plant. So this is the Marial facility on the Pirbright campus. This is the Pirbright Institute facility. These buildings are now all gone, and that's the new Animal House, and the new Jenner Institute is here, and the new Pirbright Institute is here. This is a picture from when the outbreak happened. And they knew it was the vaccine facility because the strains matched perfectly. So is there a better way to make it? And this is what we've done at Plum Island for the last decade. We developed a human adenovirus delivering the empty capsid, which is a fairly straightforward technology. Not 20 years ago when Marvin Groven started looking for this, but now it's a very accepted technology. So basically you have a replication defective human adenovirus for delivery in the gene expression space is cloned a promoter from the cytomegalovirus, and in that space we put the P1, which encodes for all of the capsid proteins, and the C3C protease that processes the P1 into capsids. And basically what you get is an empty capsid vaccine, which we licensed in the United States about 5 years ago. So there are many, many veterinary vaccines that are using this technology of a virus vector as the delivery system. So these are the diseases that the vaccines, these are commercially available vaccines. The diseases they're against and the virus vectors that deliver them in the vaccine formulation. And the last thing I want to talk about is a unique vaccination strategy. So East Coast fever is a parasitic disease caused by a pathogen called Theileria parva. It's a tick-transmitted pathogen transmitted by rhizocephalus, and it infects lymphocytes, which are the T cells and B cells of the immune system. So it infects the immune system cells. It is known that the cellular immunity, the T cell response, is the protective response. And for years, we've been trying to make a recombinant vaccine to this and have yet to succeed. But some years ago, the International Livestock Research Institute in Nairobi, Kenya, developed what's called the ITM method, which is infect and treat. So basically, to control this disease, what they do is they infect the animal and they treat with antibiotics immediately. And that system, even though it's wrought with potential problems, including antibiotic resistance, it actually is very effective. So this is, I'm stealing this from my friend Guy Palmer, who runs the Tropical Medicine Institute at Washington State University in Pullman, Washington. And he gave this talk, and actually the press, sorry? press showed up for his talk, and basically this was the headline on the website from National Public Radio in the United States is, what does vaccinating cows have to do with a girl's education? So East Coast fever in the Rift Valley area of Africa is devastating to livestock farmers. And if you have an unhealthy animal, you basically don't have an animal. So it's not productive. The milk production is down, the meat production is down, and it's actually, this disease is fatal to most cattle. So when they started doing the ITM some years ago, it was immediately determined, so there's been a lot of epidemiological studies on this, that food expenditures per household went up. The health of the smallholder farmers per household improved. There was improved farm income, and eventually there was more money for school fees. So the first thing with the improvement of income was paying school fees for the boys and taking care of medical fees. So it's always, you have to have a lot of improvement to get to the point where these smallholder farmers will pay the education expenses for girls. And the introduction of this very unique vaccination method actually ended up with a significant increase in the number of girls that were going to school in Kenya. So that's the answer to the question the reporters were asking. So vaccine technologies have exploded lately. There's a whole range of different vaccine technologies, and we're going to talk a little about that next, that are available to us in addition to the classic attenuated vaccines and killed pathogen vaccines like bacterians and killed virus. So with that, I'll stop and take any questions.
Thank you so much Dr. Cody เดี๋ยวผมจะขอสรุปเป็นภาษาไทยสั้นๆนะครับอย่างแรกเลยคือดร. Cody อันนี้เป็นความจริงที่เรารู้อยู่แล้วนะครับว่าสัตวแพทย์มักจะถูกละเลยในบทบาทต่อการพัฒนาวัคซีนและองค์ความรู้ต่างๆนะครับดร. Cody ก็แสดงให้เห็นแล้วว่าบทบาทของสัตวแพทย์เนี่ยนอกจากจะทำให้เกิดการลอนช์สาขาใหม่ๆเช่นไวรัลโลจีแล้วเนี่ยยังมีส่วนร่วมอย่างมากในการผลักดันการพัฒนาเทคโนโลยีในการผลิตวัคซีนนะครับนอกจากนี้ดร. Cody ยังได้พูดถึงว่าเรามักจะชอบทำการทดลองในสัตว์เล็กๆเช่นในหนูอะไรอย่างเงี้ยแต่บางครั้งเนี่ยการ translate ความรู้จากการทดลองในหนูมาสู่สัตว์หรือมาสู่คนเนี่ยมันเป็นไปไม่ได้นะครับต้องทำในการในคนและในสัตว์ target จริงๆอยู่นะครับนอกจากนี้ยังเน้นถึง adjuvant นะครับซึ่งเป็นสิ่งที่จำเป็นมากในการจะกระตุ้นให้เกิดการตอบสนองทางภูมิคุ้มกันต่อวัคซีนที่เราให้เนี่ยจะได้มีประสิทธิภาพนะครับและในส่วนสุดท้ายดร. Cody ก็ได้พูดถึงเทคโนโลยีใหม่ๆในการผลิตวัคซีนซึ่งหัวข้อนี้ดร. Cody จะได้พูดต่อไปในทอล์กที่2ของดร. Cody นะครับในตอนนี้ก็เป็นเวลาของ Q&A แล้วครับขอมีคำถามจาก floor ไหมครับ Is there any question from the floor or you can write it down in a paper and pass it to me and for the next talk I can ask Dr. Cody for you later. Are there any question? If there aren't any, the floor is yours again, Dr. Cody, for the second talk. New technology in veterinary in livestock vaccine development.
Okay, so what I'd like to talk about now is something that's going to be a lot more scientific and a lot more technically sophisticated. And that's what I've been trying to do for a long time now, and a lot of my colleagues, like Gregers and Sani, have been trying to do as well. And that's basically get technologies applied to vaccine development that are more oriented on analyzing the immune response, because we can't wait 20 years to develop an attenuated vaccine strain. We can't wait 13 years to develop a bacterial attenuated strain the way has been done historically. We really need to be able to respond to things like the porcine diarrhea virus and the PCV2 and all of these much more rapidly and come up with efficacious vaccines that are economically viable in a much shorter amount of time. And in my mind, the way to do that is to understand the immune response to vaccination. So quickly, I like to do this to orient everyone. Basically, the immune response is a really bad thing. When you have a massive immune response like a cytokine storm that you get when you're infected with the influenza virus, or toxic shock syndrome is an even worse cytokine storm. It's really detrimental to the body. You get tissue destruction. And then, of course, there are autoimmune diseases like arthritis and diabetes and MS that are obviously bad for you. But the problem is, not making an immune response is a lot worse because you can't fight off pathogens and you're going to perish. So in my mind, vaccination is the act of purposely inoculating and inducing an immune response. So it's an artificial induction of the immune response. So to understand how vaccines work, we need to understand the immune response to vaccines. So this is how most of my colleagues in the virology world think of an immune response. When they're really talking about serum antibodies, which is a very late effector mechanism of the immune response and a very small fraction of what is an immune response. They call it an immune response. Well, it's serum antibody response, and it's very limited because for my virology colleagues, this is a terribly complicated cartoon, and for my immunology colleagues, this is an oversimplified version of what happens when an immune response happens. So it's a very complicated thing, and it's very intricate, and there are very important steps that we need to keep our eye on as we develop vaccines. So one of the things I'd like to point out is one of the reasons protein-based vaccines, killed virus vaccines, killed bacteria vaccines don't give you long-term immunity is because the immune system has developed a very efficient feedback regulatory mechanism. And basically what happens is if the antigen is no longer around, then the regulatory part of the immune system kicks in. And things like T regulatory cells, once upon a time called suppressor cells, they shut off the immune system because the pathogen's not there anymore. So there's no reason to keep going. And as I said, in general, the immune response is bad. So you don't want it going any further than it absolutely has to. So when you have these vaccines that are based on protein, which a killed virus or a recombinant protein vaccine are, once the antigen's gone, this part of the immune response kicks in and shuts everything off. And very importantly, you don't get immunological memory, either B cells or T cells. And that's what you're really trying to generate when you vaccinate. So this memory is blocked because of the lack of driving from the presence of the pathogen or the antigen. So what happens is you end up with just the antibodies, and then once the antibodies wane, which is usually about 6 months, you don't have immunity anymore. So we've developed some technologies to try and better understand the virus-specific B cell responses. I'm a viral immunologist, so you're going to hear a lot about viruses. So we've developed a system of using what is a well-known technique called ELISpot, where we have flipped things around, and we've been able to show that we can tag the virus and then use the virus as a probe in these assays. So basically what we do is, you can do this in a tissue culture plate, in an ELISA plate rather. You coat the plate with antibody, then you put your detecting antibody down on the plate. Then you put your cells down on the plate, and what happens are these cells are B cells that are secreting antibody. When the antibody comes out of the cell, it gets captured by the anti-IG antibody. And then the step in this technology that's significant is we're using virus with a biotin tag on it as the probe for where these cells were. And then you can use a very well-known and readily available detection system called strepavidin to find that. And basically what you get are these lovely ELISpots where you can count the number of cells that are making antibody to virus. So within your blood sample, you can plate this out and show that you're either not getting an antibody response, the cells making antibody, or you are getting it, depending on what you're looking at. So this gives you a very nice system of detecting antibodies at a couple of different stages. When the B cells first expand, they start secreting antibodies, so you can find these guys. And then when they terminally differentiate into plasma cells, which are essentially antibody factories, little cellular factories of antibody, you can pick them out. So that gives you a way to look at these. And if you do this on, for example, an infected animal, you can show that the early B cell response, which is characterized by Ig class M, you get a nice burst of cells making antivirus antibodies, and then they disappear. But interestingly, the long-lasting cells, which are the IgG1 and IgG2 producing cells, they come on later as expected, but they also disappear by about a month, 28 days. We never saw any of them after day 35, out to day 35. So how do we look at the other members of the B cell population, the cells that make antibody? We can do that by flow cytometry, and basically, again, using this tagged virus as our probe, you can bind the virus to the cells because they have an antibody on their cell surface that's specific for the virus, and then you can probe those cells with other markers to see if they're memory cells or see if they're proliferating, do all kinds of analysis of what these cells are doing. So the nice thing about this is these two technologies joined together give you the ability to analyze B cell biology across the entire range of the response, when they first see the antigen until the antigen is gone and either memory has developed or not, the plasma cells have developed or not. So it gives you a way to look at the entire breadth of the B cell response. And in my talk this afternoon, I'll discuss that further. So basically the results, little spoiler, the results are that when you use killed virus vaccine, in the case of foot and mouth disease vaccines, you don't generate memory. You have a small expansion of the B cell population, they differentiate to plasma cells, they make a lot of antibody for a short amount of time, and then they're gone. So you're living on the half-life of the antibody for your protection against exposure to the virus. So you have to come back and boost these animals every 6 months. And that's an expensive thing if you're a farmer, especially if you're a farmer in Montana with 10,000 head. So how do we improve this? And basically, my approach has been to get the T cells involved and design systems to create vaccines that stimulate the T cell response and methodologies to analyze that T cell response. So again, I'm a virologist, so I'm concentrating on virus infections. The primary immunity to viral infections is T cell immunity, the induction of cytotoxic T lymphocytes. These are cells that can recognize and kill virus-infected cells. So basically, the virus doesn't stand a chance, because when it gets into the cell, it leaves a signature on the surface of the cell. The T cells come along, see that signature, and they kill the cell before virus can be made. So you don't get any mature virus produced by that infected cell. Antibody can't do that. Antibody has to wait until the cell has made a ton of virus, the cell lyses or the viruses bud, depending on what kind of virus we're talking about, and then the antibody can bind it and take it out of the system. But the cytotoxic T lymphocyte can kill the cell before the virus even gets made. So that's a much more efficient antiviral immunity, and it's the one that I keep trying to target. So unfortunately, in the literature, you'll see a lot of people talk about measuring cellular immunity, and actually they're not. They're measuring proxies for cellular immunity that are very, very inaccurate. So they're measuring PBMC proliferation. They don't even describe what cells are proliferating. They're measuring interferon gamma production by total PBMC populations. So many cell types can make interferon gamma, you don't know what you're measuring. Then a lot of people have purified CD8 T cells, and they say, okay, that's the cellular response. Well, there's different functions of cells that express CD8, including non-T cells, because NK cells express CD8 and make lots of gamma. So that's a proxy. And then interferon gamma in serum is a really rough measure. You don't even know what it is. There are excellent killing assays One of them is using a radioactive isotope called chromium-51, and I eliminated that from my laboratory 15 years ago because dealing with radioactive isotopes is a nightmare. But we have developed now flow cytometry methods to measure T-cell responses. So the antigen receptor on a B cell is that antibody molecule that binds the whole virus. But the antigen receptor on T cells are very different. So the T cell receptor doesn't see intact three-dimensional proteins the way antibodies do. What the T cell sees are antigen peptides derived from the pathogen, anywhere from a 7-mer to in the case of class 2, a 12-mer, 12 amino acids, 7 to 12 amino acids bound in the major histocompatibility complex class 1 or class 2 protein. And these are expressed on the surface in the case of class 1 of all cells, and in the case of class 2, just the classical antigen presenting cells, which are dendritic cells and macrophages. And what happens is this peptide is bound by this MHC molecule, and that's what the T cell receptor sees. So about 10 years ago now, postdoc Jared Patch and I were hypothesizing how we can make this vaccine that had been developed in my colleague Martin Grubman's lab that stimulates a lovely antibody response but no T cell response. How can we turn this thing into a T cell vaccine? And we surmised that if, hello? If we knocked out the protease, either removed it or mutated it, that processes the P1, then we'd have a protein that the cell doesn't know how to deal with. And those kinds of proteins end up being degraded into peptides, and that's what we wanted because peptides are actually the T cell antigens. So, there we go. We made such a construct, and sure enough, here's the standard Ad5 vaccine construct. It looks just like an intact live virus. You get the components of the precursor protein processed by the 3C protease. But in our Ad5 T cell vaccine, where we had removed the protease, we have the intact P1 and no processing. So that made perfect sense. It was predictable. Oops. So when we did this, basically what we got was the intact P1, no processing as you have in the other construct, and hopefully that would go to the proteasomes and become peptides. Those peptides loaded into the it's decided not to work. All right, do it the other way. I'll just do it with the computer. No, I'm good. So, and this is what we were hoping we would see, which is these large molecules that the cell can't handle get ubiquinated. That ubiquination targets it to a cell protein cell complex called the proteasome. In the proteasome, it gets chewed into peptides, and then they become available to load into the MHC molecules. So we predicted that we had created a T cell vaccine to complement the B cell vaccine that Marvin's lab had developed. So how do we show that this is actually what happened? In mouse and human T cell biology for over a decade, maybe even 2 decades now, they've been able to track the antigen-specific T cells using something called an MHC tetramer. And what that is basically taking this MHC molecule that binds the peptide from the virus and redesigning it to be expressed without the transmembrane region, but rather with a biotinylation on the truncated tail. And then biotin and strepavidin have a very high affinity for each other, so you get a strepavidin molecule that has a fluorophore tag on it. You can also get it with enzyme tags and all kinds of things, so it's very handy technology. And then when you mix these two together, you get what are called tetramers, because there's 4 binding sites for biotin in each strepavidin. You get 4 of these molecules. attached to the strepavidin. And why do we want to do this? Well, if you isolate T cells by a number of means and take a look at them in the flow cytometer or under a microscope, you have no idea what T cell is what. They all look alike. And so in the case of something like this, where they're all CD8 expressing T cells, they all look alike. There's no way to distinguish them. But if you bind them with one of these tetramers, you can show that you've got a virus-specific cell because the tetramer has a peptide specific for the virus. So you know that T cell receptor is seeing the virus. And for instance, show that that cell's making interferon gamma. Or you do the same thing with a different tetramer or the same tetramer, but in this case, the cell is killing. And now you can distinguish what's going on in this pot of T cells. You have cells that are doing one function or another function, and then there's obviously further functions you can assay for. So you get a very accurate assessment of what's going on. You know that cell is specific for the FMD virus, and now you've identified what function that cell is having. So the way we do this, make these MHC tetramers, is we have to look at the MHC molecule of the animal and then figure out what kind of peptides it can bind. And this is a technology that was developed by a good friend of Gregor's and mine, Søren Buss, in Copenhagen. It was first developed at Emory University in Georgia, in the United States. And basically what it does is you can analyze large proteomes like the capsid proteins of FMD, and by knowing what MHC molecule you're working with, you can take something like this, where you have 795 possible 9 amino acid stretches. You have the same number of 8 amino acid, actually more, of 8 amino acid stretches, et cetera, et cetera. So you have over 2,000 possibilities for peptide antigens. And by looking at the ability of whatever your MHC molecule is to bind those peptides, you can run what Søren calls the scanning peptide library. So this is a swine class 1 MHC molecule, SLA10401. And what this shows you is the positions in that MHC molecule that bind certain amino acids. And then our colleague Morten Nielsen puts it through his algorithm, and he tells you what are the amino acids that MHC molecule can bind. So this is all done on a computer. All you need is the sequence of the MHC molecule from the animal, a lot of which are all in the database already, and the sequence of the protein from the virus you're trying to look at, and the computer will do this for you. And it's called the NetPan MHC. I think we're up to 4.0 or 5.0 now. We're way past 3.0. This is an old slide. And basically what it does is it predicts what the peptides are out of that big protein, which has got over 809 amino acid stretches and 1,008 amino acid stretches. And it says which of these peptides should bind these two molecules. We have the SLA1-0401 and the SLA2-0401. And then you actually go and do the experiment. So it's a simple ELISA to test whether or not the peptide actually binds. And this is what that kind of data would look like. And it turns out that in a lot of cases, the prediction is really good. So it was predicted to have a 0.01 nanomolar binding capacity, and it had a 0.8, which isn't too bad. But then there were predictions that were completely off. So this was predicted to be a binder, a weak binder, and it was a strong binder. This was predicted to be a strong binder, and it didn't bind at all. So you use the computer program to predict which peptides to look at. Then you do the experiment to confirm which peptides actually are bound. And then you can make tetramers and do the experiment. So basically you take this entire proteome. The computer predicted this number of peptides that should bind, and the binding assays say that 5 of those 10 actually are binding peptides. So now you can use these to make your tetramers and do your experiment, which is, as I said, you take the bacterially expressed MHC molecule, you add the peptide and the strepavidin, and then you can stain T cells and show that they are antigen specific. So further refining the technology, let's come back to that cytotoxic T cell I was talking about, which is, in my mind, the most important antiviral immune response. And basically, now we can actually say which of these cells are specific for the virus and have killed, because the killing mechanism in a CTL is delivery of this perforin molecule, which puts a hole in the target cell membrane, and then these granzyme molecules, which goes into the target cell and kills the cell. Well, this vacuole in the CTLs that house these molecules are lined with a protein called LAMP1, which now has the designation CD107A. And so what happens is, when the cell hasn't killed, it's all internal. But when the cell has killed, you have the LAMP1 on the surface of the CTL. So now we've automated the killing assay, because we can now use the tetramer, the anti-CD107A antibody, and use flow cytometry to show that you have an FMD virus-specific cell, because it binds the tetramer, that has killed because it's binding the anti-CD107A. No, I'm fine. We'll finish this one. So now you can assay CTL killing by flow cytometry, and that takes it up to a very automated stage. And again, very accurate assessment of the vaccine, because now the tetramer identifies a virus-specific cell, and the anti-interferon gamma tells us that that cell is making interferon gamma, or the tetramer identifies a virus-specific cell, and now the CD107A binds, and that means that cell has killed. So it's a true CTL, cytotoxic T lymphocyte, that has killed a virus-infected cell. So what all of this does for us is gives us much more accurate information immediately. We can assess post-vaccination within 2 or 3 weeks. We don't have to wait, a month, 2 months to do a challenge. Eventually, we hope we can make these correlates of protection, correlates of immune function, make these immune function correlates predict how well the vaccines will do. And since we don't have 13, 20 years to wait to make each vaccine, we believe this is going to really accelerate vaccine development and performance. So with that, I'll take any questions. Thank you very much.
Thank you so much, Dr. Cody. That's a kind of mind-blowing technology we have nowadays. Alright, so ผมขอสรุปสั้นๆนะครับ5 นาที คือตอนนี้ดร. โกดี้ได้เล่าให้ฟังว่าเรามีเทคโนโลยีอะไรบ้างอยู่ใน toolbox ของเราในการจะทดสอบวัคซีนที่เราผลิตขึ้นมานะครับ หนึ่งในนั้นก็คือเราสามารถ predict ได้แล้วว่าเพปไทด์ของแอนติเจนอะไรที่ MHC ของโฮสต์เป็นตัวนำเสนออยู่ทำให้เราสามารถโฟกัสได้ว่า T cell response ต่อไวรัสชนิดหนึ่งเนี่ยมัน restrict ผ่าน MHC peptide complex อะไรและด้วยฟังก์ชันแบบไหนเป็นการ killing หรือเป็นแค่การสร้างไซโตไคน์เฉยๆนะครับซึ่งอันเนี้ยจะช่วยทำให้การทดสอบวัคซีนของเราเนี่ยมันเร็วขึ้นกว่าเดิมมากนะครับก็มีคำถามจาก floor ไหมครับ Is there any question? Okay, Dr. Sanipa, please.
Thank you very much. Very nice presentation. Just wondering on the tetramer design. As pigs also have various type of SLA molecules, how would you comment on differences in SLA molecules and the difficulties of developing this type of CTL assays in other part of the laboratories in the world, not in your laboratories?
Well, what we found certainly in cattle, and Gregors can probably speak to the swine better than me. In Holstein Friesian cattle, it turns out that there's maybe a dozen dominant class 1s and 8 or 10 dominant class 2s because they're so highly inbred. At one point, Brian Charleston told me they stopped doing this, but at one point, the UK, every Holstein Friesian calf was from the same bull because it was such a highly productive strain, a line rather. So in the domestic species for food production, I don't think we're going to have a problem because they've been selected for so heavily for production that it limits the number of MHC molecules you got to worry about. The human guys are in trouble. They've got 10,000 molecules to deal with. So, you know, we can look at almost every animal by having our handle on 25, 30 molecules for each class 1 and class 2, which puts us, again, in a much better position than the human guys. That said, human tetramers are available for probably over 100 different specific HLAs.
So you say that puts the veterinarians in a good position, but does it put the pigs or the cattle in a good position? Because T-cell heterogeneity, isn't that also something that's really important for disease defense?
Absolutely. Absolutely. So the limited diversity of the MHC in these domestic breeds that have been selected for production, not for MHC, but for production, as I said, in the UK they made this colossal mistake of everybody using that bull because the cows that came from that bull gave so much more milk than anybody else's. Of course, people stepped in and said, no, this is dangerous, don't do that. So they diversified again. But it's a problem with the fact that we're always selecting for production. You know, the chickens are in the same boat, especially the broilers, because they've developed these broilers that have such huge breasts, you know, because it's so much meat, and they have to do they've had to rethink as well to diversify their genetics because of susceptibility.
Another question. So you, I like T cells too, as you know, obviously. But what if you can find a way to give a better B cell memory response with recombinant or killed viruses?
Of course, that's T cell driven by the CD4 subsets and very poorly understood. So I think that's a very, very important part of the system. But again, if you can make so for viruses. And I won't even speak to intracellular parasites or bacteria. I'll stick with viruses. For viruses, it's by far the best immunity because antibody can only see the virus after it's come out of the cell, or the cell has lysed. What the T cell can do is arrest maturation of virus particles. So the virus infection is stopped at the cellular level. But of course, the best thing, and what I'm really driving towards, is having both solid T cell memory, and solid B cell memory. And with these flow cytometric techniques, we can analyze very quickly whether the vaccine formulation in the adjuvant that we've chosen, or however the company we're working with has decided they want to formulate it, are we doing good things or bad things? Because a lot of times what will happen is you'll reach a point of commercialization, and this happened with us. You reach a point of commercialization, and we didn't have an adjuvant, which we preferred because we were thinking we needed to boost, and we didn't want a really strong response to the adeno delivery vector, because when you do the boost, the antibody response to the adeno will wipe out the boost, so it's a waste of time. Well, the company insisted on putting in an adjuvant, and it's a pretty amazing adjuvant because we tracked out to 12 months post-vaccination. And even though the anti-FMD titer was gone, the anti-adeno titer was still so high we couldn't boost those animals. So now you have to use a different vector for the boosts. So that's my example of how you go to commercialization, your industrial partner has a proprietary adjuvant, they insist it goes into the formulation, and it screws up what you're trying to do. The other thing that could happen is you could add an adjuvant that actually blocks the development of memory, and nobody's doing any assays to determine whether or not that's happening. Did you get memory in this formulation, and then this other formulation you're not getting memory? Those are really important analyses that aren't being done until these kinds of technologies are applied to the vaccine development systems.
We just published together with LifeX Center from Berlin last month in Nature Immunology that adding TLR8 agonist with bacterial RNA drives T follicular helper cell and B cell memory response. So that could be one technical solution.
Yeah,
then for adding
so
just
obviously
recombinant.
you and everybody else are working their way through all of the various innate stimulators to see what are the best adjuvants. I mean, CPG was everybody's favorite for the longest darn time, which is a TLR9 agonist, and I don't think it's in any vaccine formulation yet, and they've been working on that for a decade and plus.
คริสเตียนเดี๋ยวผมจะแปลผมจะสรุปคำถามให้ฟังอีกทีนะ
Very high level presentation, Dr. Golde. Thank you for sharing. I would like to bounce on one point that you touched on the side on your presentation. You mentioned usually when we measure immune response, we can look into interferon gammas and we are looking into signals, but we are not actually identifying the T killer or the actual cell doing the work. So I would like to take that point because usually the customer is always asking when he has a vaccine, how do I measure protection? How do I know that my animals are mounting an adequate response against vaccine X? So what do we do today as veterinarians in practice? Most of the time we can understand the epidemiology of a given disease by looking into IgG, but that's only understanding how the pathogen is moving on the population.
That's looking at exposure, right.
It's not telling us anything about protection. Then we can go into this interferon gamma to try to understand better the protection, but I take from your presentation that it might not be the complete answer. So now comes the question. Do you see potential for this kind of technology, this kind of flow cytometry techniques to be used not only on vaccine development as a predictor of efficacy, but also as a more commercial and massive use to understand the response of vaccines in the field?
At this point, because of the expense of the equipment and the expertise of the people doing the work, I see it as mainly accelerating vaccine design and development and testing. That said, when I started, only a few institutes could afford a flow cytometer, and you know, now you can buy a benchtop flow cytometer for $30,000 US and do basic flow. So my bias is that the ability to measure induction of memory is going to be the best correlate of how well your vaccine is working. If you induced antigen-specific memory in both the T cells and the B cells, then you probably have an excellent vaccine that's going to afford you good protection for a reasonable length of time. Given the development of the flow and just the whole field of the physics of light and the biochemistry of fluorophores, how fast that has been developing over the last 10 years, I could imagine a $20,000, $10,000 machine with a small blood sample, totally automated, you know, done in a large veterinary clinic in a region isn't more than a decade away. But we need to tell them exactly what the components of that test are and how to do that measurement. And I think that's coming because we do know the markers of memory B cells and memory T cells. So the question is, how do you get it down to a simplified 2 or 3 step test, pop it in the machine and get your answer?
Any other question? Okay. So I'm gonna ผมจะสรุปคำถามแรกก่อนนะครับจากอาจารย์สันนิพา อาจารย์สันนิพาสงสัยว่าไม่ใช่สงสัยถามว่าด้วยความหลากหลายของ SLA molecule เนี่ยมันจะเป็นไปได้ยังไงที่แบบเราจะดีไซน์วัคซีนเดียวที่จะกระตุ้นภูมิคุ้มกันผ่าน T เซลล์เนี่ยได้ทุกๆสัตว์ทุกตัวเลยดรวิลเลียมโกดี้ก็เลยตอบให้ฟังว่าคือจริงๆแล้ว SLA molecule นะครับมันจะถูกโค้ดด้วย6อะลีลนะครับ3มาจากพ่อ3มาจากแม่ถ้าเกิดเราในระบบอุตสาหกรรมที่บลีดเป็นอุตสาหกรรมเนี่ยพ่อเดียวใช่ไหมครับว่าเรามั่นใจได้เลยว่าแฮปโปไทป์1 3อะลีลเนี่ยต้องเหมือนกันทุกตัวในแม่ก็มีอยู่จำนวนหนึ่งดังนั้น SLA variation ในสัตว์ในอุตสาหกรรมเนี่ยไม่เหมือนในคนที่ variation สูงมากนะครับจึงเป็นไปได้ว่าที่เราจะสกรีนหา major SLA allele ใน population ของเราและดีไซน์วัคซีนที่จำเพาะต่อ SLA นั้นๆนะครับอันนี้เป็นไปได้อยู่นะครับส่วน I kind of forgot your answer your question alright so มาถึงคำถามของดรเกรเกอร์นะครับก็คือดรเกรเกอร์พูดถึงประมาณว่าในการจะได้มาซึ่งวัคซีนที่ดีเนี่ยเราควรจะได้ memory ทั้ง B เซลล์และ T เซลล์แต่เราจะทำยังไงให้ได้มาถึงสิ่งนั้นทั้งคู่อะไรเงี้ยครับคือสรุปก็คือเป็นไปไม่ได้ที่จะได้ memory B เซลล์โดยปราศจากการช่วยเหลือของ T เซลล์ดังนั้นโกตอนเนี้ยไม่ใช่วัคซีนที่จะกระตุ้น strong antibody อย่างเดียวแล้วต้องเป็นวัคซีนที่ได้ balance ทั้ง B เซลล์และ T เซลล์ memory ซึ่งเราจะอาศัยเทคโนโลยีที่ดรโกดี้เนี่ยอธิบายให้ฟังในวันเนี้ยในการที่จะ measure สิ่งนั้นพร้อมๆกันได้ซึ่งจะนำไปสู่คำถามของคริสเตียนว่าทุกวันนี้เวลาเราคุยกับลูกค้าในเวลาที่บริษัทคุยกับลูกค้าเนี่ยเราก็ดูไตเตอร์แอนติบอดีใช่ไหมครับหรืออย่างน้อยตอนนี้พัฒนามาหน่อยเราดูอินเตอร์ฟรอนแกมมาเรสปอนของ T เซลล์จากวัคซีนนั้นซึ่งดรโกดี้ก็แจ้งให้ทราบแล้วว่าเป็นแค่ proxy marker ของการมี response อาจจะไม่ได้บอกถึง killing หรือการ protection อะไรเป็นไปได้ไหมว่าเทคโนโลยีเหล่านี้จะถูก transfer มาสู่farmer มาสู่ผู้ใช้งานจริงๆนะครับอย่างถ้าเกิดอยู่ในฟิลนะครับจะทราบว่าเทคโนโลยี flow cytometer เนี่ยมันเคลื่อนที่เร็วมากจาก10 ปีที่แล้วจนทุกวันนี้เครื่องมือเนี่ยเปลี่ยนไปอย่างหน้ามือเป็นหลังมือเลยแล้วก็ถูกลงมากก็เป็นไปได้ครับว่าภายใน 5-10 ปีข้างหน้าเนี่ยเราอาจจะมี fully automate flow cytometer ที่ใช้ใน lab regional lab หรือว่า company lab ได้อย่างง่ายดายนะครับแต่ว่าก็เป็นหน้าที่ของนักวิชาการของ researcher ที่ต้อง introduce ความรู้เหล่านี้เข้าไปในฟิลเข้าไปให้ company ต่างๆรับทราบนะครับเพื่อนำไปสู่การพัฒนาแอปพลิเคชันนั้นๆนะครับก็โอเคมีครับโอเค
Just kind of very interesting on the findings of the FMD vaccine, the Q vaccine is not activating memory B cells good enough. And I think that has been seen in this region as well, where farmers need to vaccinate very frequently. And here in this region, we don't really have access to those recombinant adeno, and particularly in pigs, because this is a swine vaccinology. I don't think they've been tested well for swine, for pigs, for the adenovirus vector, right?
All of the original data was in pigs.
Oh, okay.
So it was our partner, Department of Homeland Security, that told us they wanted to license it in cattle.
Oh, okay.
We have a lot more data in pigs than we have in cattle, but they just didn't
Okay.
want to fund getting it licensed for pigs.
So that's okay. But at least here, most of the people would know like Q vaccine more than the recombinant. We don't have access to those recombinant vaccines. What's your comment on how can we improve the vaccine formulation for improving the memory T cells? And Gregor was suggesting that maybe adding the adjuvants or TLR, some of the TLR agonists for improving that memory B cells response. Have you seen or do you have any comments on that? That maybe we can start, because we also have vaccine manufacturer in this region, so maybe we can think about how to improve the FMB vaccines in a better way.
Well, this is your expertise, actually. We have to figure out how to block the Treg response to that, because that's what's shutting everything down. So you have two choices. You can prolong the antigen exposure, and there's ways to do that. What a lot of people have tried with very marginal success is slow release kind of vaccine formulation. So the adjuvant isn't just potentiating the immune response, it's retarding how long it takes for the antigen to be released into the system, so you get a longer antigen exposure. Theoretically and in experimental systems, that works pretty well. They haven't been able, I don't know of anyone that's come up with a formulation that works in a commercial product. So translating that from the laboratory to the commercial product has become a hurdle that apparently nobody's been able to get over. I think if you're able to redesign the vaccine to get at least the CD4 T cell memory out of that initial vaccination, that's going to set you up for much better things as you do the first and second boost. Because now you've got that memory at least in the helper cell compartment that's going to expand the response very quickly when you come back and do the boost.
So the suggestion is that not only measuring of the antibody, but we need to also set some techniques or have some method to detect and assess the level of the memory T helper cells
Yes.
in following the vaccination.
I think that's more important in analysis of the vaccine performance. Measurement of the CD4 helper cell response is more important than measuring antibody or CTLs because it's driving both. And understanding as you design your formulation that you're doing good or bad to that component of the immune response is absolutely critical. And I think the flow cytometry methods with the tetramers, where you're only looking at the antigen-specific T cells, is going to be the way we get there.
ก็มาสู่คำถามที่2ของอาจารย์สันนิพานะครับอาจารย์ถามว่าคือวัคซีน FMD ที่เราใช้อยู่ในบ้านเราเนี่ยเป็นวัคซีน Q ไวรัสใช่ไหมครับซึ่งก็อย่างที่ดรโกดี้ได้เล่าให้ฟังแล้วคือ Q ไวรัสหรือเปปไทด์ไวรัสหรือ recombinant protein vaccine เนี่ยไม่สามารถกระตุ้น long term memory ได้เพราะว่าเมื่อ immune response kick up ขึ้นมาแล้วเนี่ย T-reg จะเข้ามากดไม่เกิดการสร้าง memory ขึ้นหนึ่งในวิธีแก้ไขที่ดรโกดี้นำก็คือว่าทำไงก็ได้ให้ prolong antigen exposure อยู่ซึ่งก็มี adjuvant บางชนิดที่เหมือนเป็น slow release ค่อยๆปล่อยออกมาอะไรอย่างเงี้ยนะครับแต่ก็ยังไม่เคยมีการทดลองจริงๆในฟิลด์เลยว่าใช้แล้ว work ไม่ work อะไรอย่างเงี้ยนะครับก็คือสุดท้ายแล้วคือเราต้องเอมไปว่าคือเหมือนกับว่าเราต้องทำให้วัคซีนเราเนี่ยใกล้เคียงกับการติดเชื้อธรรมชาติที่สุดเพื่อที่จะได้ทุกอย่างได้ทุกๆ component ของ immune response ที่เพียงพอในการป้องกันแล้วก็ long life ด้วยผ่าน memory นะครับนี่ก็เป็นหนึ่งใน challenge สำคัญมากของการพัฒนาวัคซีนอีกปัญหาหนึ่งของviral vector vaccine ที่เมื่อกี้ผมลืมสรุปให้ฟังก็คือเวลาเราฉีดอย่างเช่นในกรณีของ FMDV ที่ Plum Island develop ขึ้นมาคือ Human Adenovirus 5 นะครับเป็น viral vector ในการนำFMDV จีโนมเข้าไป present โปรตีนข้างในโฮสต์ปรากฏว่าเมื่อฉีดเข้าไปแล้วเราได้แอนติบอดีต่อตัวเวกเตอร์สตรองกว่าต่อตัว FMDV ที่เราอยากได้ทำให้เราบูสเตอร์ไม่ได้เพราะเราบูสต์เข้าไปครั้งที่2ปุ๊บแอนติบอดีที่สร้างต่อ Human Adenovirus ก็ neutralize Human Adenovirus vector หมดเลยนะครับอันนี้ก็เลยเป็น challenge ว่าทำยังไงที่เข็มแรกจะไม่กระตุ้นต่อเวกเตอร์แต่กระตุ้นต่อของที่เราต้องการเข็ม2 boost เพื่อให้ได้ต่อของที่เราต้องการมันเป็นเหมือนกับปรุงอาหารนะครับต้องค่อยๆปรับต้องค่อยๆแต่งไปเรื่อยๆนะครับแต่ว่าจะรู้ได้ก็คือต้องมี tool ที่สำคัญเช่นที่ดรโกดี้ได้นำเสนอไปแล้วนะครับเรายังมีเวลาอีกประมาณ3นาทีนะครับสำหรับคำถามมีคำถามจากฟลอร์ไหมครับโอเคครับ
I have one question. Since I am the practitioner, so not so will understand for the process of the laboratory, but one thing that I always found during when we do anything in the laboratory is mean that there are some, it should be something that error during the work. So for the new technology to detect the CTL, I would like to know that what can be the possible error or possible factor that can cause some error or some false positive or false negative to the test.
So if I understand correctly, being able to tell how well the vaccine's doing, I think we can develop those technologies because it's going to happen in the research laboratory and the pharmaceutical company that's going to make the vaccine. For the field tests, that's going to be much more challenging because there's going to be a lot more samples to look at. I think it's going to be, so one of the things that that I heard a very good talk in the African swine fever meeting that happened a few weeks ago is in human vaccinology and deployment of new human vaccines, there's a tremendous amount of follow-up after the vaccine is approved and starts to be used. There's a tremendous amount of work done looking at people that were vaccinated, looking at the protection levels in the next outbreak, say, next season of flu or whatever they're looking at. And in veterinary vaccines, that doesn't seem to happen very much at all. And I think what you're asking, if I understand correctly, is actually it needs to be done a lot more in veterinary vaccines in general, even using the simple antibody assays that we have available to us immediately. But certainly as these new, more sophisticated assays become available, I think it really would, and what this speaker was saying with very much passion, is the vaccine companies and the regulatory agencies that approve veterinary vaccines need to do a lot more on the follow-up side after the vaccine is actually licensed and now being sold. So we get data to know how well the vaccine is doing in the field, because that kind of data is really sparse for veterinary vaccines, whereas the database for human is amazingly large. And it's so large that they can actually do, people are now doing experiments in the cloud, so they have so much data about so many patients. You can actually do experiments without vaccinating any more people or doing any more sampling. There's so much data to work with.
So it means that to have the good result or more accurate, we need to do a lot of pre-market
Yes.
study.
Yes, absolutely. And that's what this person was saying, that, that's one place the veterinary field has fallen down.
ก็คำถามนะครับคำถามคือว่ามันมีเออร์เลอร์อะไรจะเกิดขึ้นได้มาจากการวัดฟังก์ชันนอลต่างๆเหล่านี้คำตอบก็คือมันก็เหมือนในแลบทุกอย่างครับมันมีได้ครับแต่เราจะไม่รู้หรอกว่ามันส่งผลต่อวัคซีนที่เรา develop ยังไงจนกว่าเราไปลองใช้ในฟิลจริงๆซึ่งทางสัตวแพทย์เนี่ยเราไม่ค่อยมีการ follow up หลังจาก launch product แล้วนะครับนั้นก็ต้อง encourage ให้แบบรวบรวมข้อมูลให้เยอะๆให้มากพอนะครับเพื่อจะดูว่าสุดท้ายแล้วใน long term เวลาไปใช้จริงๆแล้วเนี่ยมันมีความผิดพลาดอะไรที่เราอาจจะ extrapolate ผลจากการทำในห้องแลบหรือเปล่าอะไรเงี้ยเมื่อนำไปใช้ในฟิลจริงๆนะครับก็จำเป็นต้องมีฐานข้อมูลที่ดีมากในการ follow up วัคซีนต่างๆนะครับ
So thank you very much, and thank you very much for the invitation to come here to Bangkok. It's really a great pleasure. I was here when I was a young student in November 1989 and visited Dr. Kengsak Saitanu at Chulalongkorn University for a 3-week period on mastitis, and then traveled around the country for a couple of months. So really delightful to be back, and thank you very much to Virbac and Jotin for inviting me. So I have also 3 talks today, and this is the first one. It's in 2 parts. It's general about how we are handling vaccine prevention in Europe, particularly in Denmark. But also I'm giving you the how the vaccine was developed against PCV2, because it's really nice experience and one of the most recent ones that have had a great success, and it's an annoying story for an immunologist like me, because the first vaccine shouldn't work, but it does really well anyway. So when we are talking about vaccines, there are different approaches, and all of you have of course been vaccinated with prophylactic vaccination. That is the standard we do, and that is what is being done in for both humans and also for our companion animals. This is what we do just to prevent the emergence of any diseases. But in veterinary science, we are not handling all vaccines only from a prophylactic department. We're using that for the endemic diseases, but we are also having the possibility to do emergency vaccination in, it's really slow, this one, isn't it? Yeah. emergency vaccinations in connection to outbreaks, for instance. So for all our exotic diseases, we have a possibility to vaccinate as well. But that's a very different approach, and sometimes we want to vaccinate to kill, as we say. This is, I understand you, this just doesn't work. So yeah. This is the nothing. Maybe just needs to find it. Now I think it works. Yeah, it's working. Okay, or maybe you want to vaccinate to kill, which sounds drastic, of course, but it's really important for a national control procedure. In Denmark, if we were to vaccinate for some of the diseases that we do not have, we would have to kill the animals afterwards because we just do not want those antibody responses in our national herds. And then, of course, there are possibilities to do post-exposure treatment or post-exposure vaccination, for instance, like you do with rabies, or today development in tuberculosis, where they get the BCG vaccine and then maybe try to boost it later on, even though they have the infection. So when we want to do this and how to measure vaccines, we need to understand the disease we are working with. And the basic reproductive index, or R0, is a measure we're using to tell how infectious is this disease that we are working with at the moment. So in this case, we have we have the diseased animal here, and the number of other animals that it will infect will determine the basic reproductive index. So in this case, it's 1, so 1 diseased animal. I just need to. 1 diseased animal will then infect 1 more animal, and as this one is then becoming healthy, this will infect 1 animal, and therefore the disease will just be maintained. It will only be flat. If you have more than 1 being infected, of course, then the disease will spread and it will increase exponentially. So in order for our vaccine to determine how well that works, we can relate that also to R0. So if we have a vaccine that's protecting, if we get R0 below 1, then the vaccine is actually working. So that's, of course, very nice, and you can put this into this perspective, but you can also use it into this perspective of herd immunity, which is really, really important. So when you are vaccinating, the vaccines never work perfectly. You cannot expect a vaccine to be 100% effective. So therefore, either you can say that every animal is maybe 90% infected, but you could actually also say that maybe only 90% of the vaccinated animals are protected. And therefore, you need the vaccine to sort of help each other out. And here you have, without herd immunity, you haven't vaccinated enough people or animals, or maybe the vaccine isn't efficacious enough because it can still spread throughout the population. Here we just have a few more vaccinated, but that will stop transmission. The vaccine never gets into these susceptible animals that it was in the other hand. So herd immunity is really important. And as this slide from National Institute of Allergy and Infectious Diseases also shows how in a completely non-vaccinated population, everybody will become infected more or less. When you vaccinate just a few, that really won't help anybody but those few. But if you vaccinate almost everybody and there are just a few people that are non-vaccinated left, only a minority of those will actually become infected because they are protected because of this spreading stops within those circles. So herd immunity is also really, really important in the veterinary field, that you need to understand, since the vaccines are not 100% effective, you need to make sure that the herd immunity is sufficient to protect both those that were non-infected and those where the vaccine maybe didn't work 100%. So without herd immunity, actually those that were vaccinated already could become infected because the vaccine isn't perfect and the disease level is just increasing in all the other ones. So make sure that you have a program for the whole herd and vaccinate enough so you get what you want to see. There's a little bit of mathematics in terms of how many you then need to vaccinate. Of course, it's easiest just to vaccinate everybody, but we know with anti-vaxxers and so on that there's always those that do not vaccinate, and there may be one animal where it slips, something happens. So how many do you need to vaccinate? Well, this is the critical level of vaccination, how high a percentage. This is 100%, and that depends on the basic reproductive index of the virus or the infection. So very infectious diseases like measles, where every infected individual will infect more than 50 new ones, of course you need to have a very, very high level of vaccination. With other, like influenza, where actually not everybody becomes infected, you can go by with a lower level of vaccination and still get protection. So you can have this critical level of vaccination is 1 minus 1 over R0. So once you know R0, you have an idea about how you actually should vaccinate or to have an effective control program. And this is just a list of all the human ones, where you can see that some, like measles, is really high, and Ebola or influenza, actually, even though they're very, Ebola is very lethal, the reproductive index is not that high. And in terms of veterinary vaccines, classical swine fever has a relatively high one, but again, now we are talking into an animal population, and since there are so many variables that come into play when you're talking about either wild animals or animals where you have 1,000 sows going into the same building, the reproductive index may change in that setting because of the distance to the next susceptible animal. And also African swine fever, very, very different reproductive indexes, because the vaccine has so many different ways to transmit, whether it's from ticks or from individual to individual. And rabies, of course, with the dogs. And then foot and mouth disease is a really, really infectious virus. For pigs, it's estimated to have a reproductive index around 30, and for cattle, up to 75. But, as Dr. Goldie just talked about, in the outbreak in the UK, the sheep did not show much of a transmission. Well, they were transmitting, but they were not showing any clinical signs. And therefore, maybe they, and if they had a reproductive index of 70, you would have seen it much, much faster. So there the disease was going slower. So all these things, you need to have a basic idea about what you want to do when you want to vaccinate. And if you then come into a setting where you then look at this all together, we have the epidemiological triangle. It was also mentioned yesterday by Dr. Otaka, I think his name was. So you have the agent, the infectious bug up here. So of course the R0 is important, but also the dose. And the dose will vary, of course, with how close you are to the individual. Then you have the host. So what is the immune status of this host? Are they stressed? Are they very healthy? Have they been vaccinated? What is their age? And very importantly, what is the genetics? So some animals will be genetically more susceptible or more resistant to the infections. There's the environment. How close are the animals together? What is the density? How much animal movement is around? Even what is the weather? All things that are very important in our veterinary setting. And all these things will influence how the disease is actually coming on. And if you put it more or less together, you then have the possibilities to work on the animal husbandry. How can you counteract these things? What can you do? Well, you can use your vaccines. You can maybe have a targeted use of antimicrobials. What is the housing? What can you do in terms of biosecurity so that you have changing the dose of those agents? So all these things are really important for infectious disease control and how you want to do it. And of course, vaccination is really one of the key ways to work with this. So Denmark, this is Denmark. It's a very, very small, flat country sitting on top of Germany. We've got water on every side, And that has actually given us a unique opportunity to keep infectious diseases out. So Denmark has a long history. We've eradicated all wildlife, all nature has been tarmacked, and it's basically just an agricultural and industrialized little country. We don't have any wild pigs. We have some wild deer, but and now we have got a few wolves, and people want to kill them because that is just so dangerous. But otherwise, everything is really in husbandry, and that means that we have been able to do things that not many other countries can do. So actually, we haven't been really fond of vaccines in Denmark. And we have a national policy where we have eradicated foot and mouth disease completely without vaccination. So we had an outbreak last time in 1982. It was endemic in 1920s, but then it was eradicated and we had an outbreak once in a while, but now we haven't seen it since 82. We eradicated classical swine fever, again, without vaccination. We eradicated brucellosis in the 1950s without vaccination. We have also, more recently in the 90s, eradicated Augiesc's disease. So all these infectious diseases are not there, so we will never think about using vaccines today because that would completely destroy our control program and the documentation that we are actually free of these diseases. And also for cattle, we have eradicated both tuberculosis, IBR, leukosis, blue tongue, and even in fish, viral hemorrhagic syndrome has been eradicated from our streams. So that's a non-vaccination approach that has been very, very prevalent in Denmark for many, many years. And classical swine fever as an example of why we do this and how we can do this. This is what happens when you have an acute attack of classical swine fever. There's a short incubation period, and within a very few days, you'll actually have the virus in the blood. So you'll have the viremia, then you'll have the fever, pigs will start dying, and after a couple of weeks, the immune response sets in. You get the antibody response, and that will, of course, continue all the way to the end of the haul with a live virus infection. So as long as they are alive, afterwards they will have antibodies to classical swine fever if they experience the infection. But if you want to use this in a diagnostic setting, there are some opportunities, and antibody ELISA is not an opportunity to diagnose the acute infection because that comes at a very late stage. So early you can use some RT-PCR or virus isolation. Those are the things that are being used in the acute case of infection. But if you want to document that the animals have not seen the infection before, you can only use the antibodies. So antibodies is staying for a long time, and we can use that only to document that the infection is not there. We cannot use any of the other agents because the window is so short, and we'd have to sample a lot of animals. So today we are testing about 40,000 blood samples every year to show that we do not have classical swine fever, and we do the same thing for many of the other infections, so we can use that as an export promotion to say that you can be absolutely sure that you will not get any of these infections when you're using Danish pigs. So that's a big problem. If we started using vaccination against these animals, infections, it would destroy everything, and therefore the vaccinate to kill strategy comes into place. If we have an outbreak, everything would stop. Denmark would be suddenly changed into a completely different scenery with police barracks everywhere, and all pig transport would be banned, and everything would stop. It would be a disaster for a country like us if we had now classical swine fever or foot and mouth disease as an outbreak. So everything would stop, every export, every slaughter. And all the samples, all the herds would have to be visited by vets. They would have to take samples, and probably around every case they would start vaccinating. But you would need to make sure that everyone you vaccinate, you know which animal it is, because you need to kill it afterwards so you can show that when you stop vaccinating, the disease will not spread. So a very different way of working with vaccination compared to the prophylactic approach that we would use every day. So lots of work, lots of losses, certainly in billions of kroners very, very quickly. And in terms of vaccinating future against infections like this, the concept of DIVA vaccines is really, really important. So a lot of effort has been trying to make vaccines against both foot and mouth disease, but particularly classical swine fever, to have this possibility to differentiate infected animals from vaccinated animals. So the traditional antibody test we're using, if it's positive, it has been infected. The problem, if you're using a normal vaccine, it will also become positive. So therefore, you cannot tell whether it was a vaccinated animal or an unvaccinated animal. So the DIVA vaccine will have a marker ELISA, some test that accompanies the vaccine, so that you can now actually show that you can still use, if it's antibody negative, it has not seen the infection. And to give you a practical example about how that could be used, it's instead of actually differentiating, it is the possibility to diagnose infection in vaccinated animals. It's an important concept. So this is a wild-type virus. It's got several different antigens, and the immune response will respond to all of these antigens. If you have then a vaccine where you are only using a few of these antigens, that will give you a possibility to then use a DIVA test where all the green antibodies are reflecting natural infection, but the red ones, all the green ones are coming from the vaccine or from infection. That doesn't really matter, but red ones can only come from infection. So if you're then making a new test where you're only using the red antigens, you can say that now if it's negative for red antibodies, it's also negative for classical swine fever, but it's still protected. And that is the concept of DIVA, and that is the only way that we would start vaccinating against infectious diseases like that in Denmark. Even African swine fever, which is emerging, I can't see any picture where we start vaccinating unless there is a DIVA company diagnostic test that works together with it. So next, I thought I would give you the PMWS story, porcine multi-wasting syndrome, because it's really an interesting approach, and it's a very recent example of what happens when a new infectious disease emerges. So most of these slides I have received from Professor Gordon Allen from Queen's University in Belfast, and he was one of the pioneers in this disease and development of this vaccine. So thank you for this, Gordon. So PMWS started in Canada and very shortly after also in Britannia in France. Suddenly pigs started dying. And this was this multi-wasting syndrome where all sorts of organs were affected, and pigs were, of course, in this case, really stunted, so not a lot of growth, but all sorts of tissues were affected, the intestines, the lungs, the kidneys, and very much involvement of the immune system, so enlargement of lymph nodes. Accompanied this new disease that was a mystery disease all over where it came from. And very quickly it spread to the rest of the world, actually. This is an example from Great Britain. So remember, it started, the first verified case was in '95, I think, in Canada. And already in 2000, you had 5% of all submissions were diagnosed with PMWS. And very quickly, this was really a predominant disease. So this is also where a population of animals are very susceptible. So it tops and then it goes down, and the vaccine emerges in 2006, but already at that time the reproductive index has changed because the susceptibility in the population is actually going down. And we saw the exact same thing in Denmark, that this was suddenly a major diagnostic challenge and also management challenge for pig farmers in Denmark. This is from the US also. It trends lots of infections, and with a very, very high morbidity and even mortality up to 50%. So sometimes even 50% of the young animals were dying from this disease after weaning. That's really a very big challenge, and a big challenge to find out what was happening. So what was seen was that there was this PCV, so porcine circovirus staining of lymph nodes, but porcine circovirus was not believed to cause any kind of infection. So this was made with a monoclonal antibody from Gordon Annan's lab, and all the other porcine circovirus antibodies, they did not react to it. So this was a polyclonal antibody, the only one that reacted, so probably it was a different serotype from what was seen before, which was porcine circovirus 1. And it also caused these multinucleated giant cells in the lymph nodes. So it looks like it was infecting the immune system, giving an intracellular chronic infection, but still with a very acute onset. And very quickly, it spread everywhere, also Southeast Asia. Actually turned out from some of them that was almost in parallel to what was seen in Canada and France. So a big new challenge for the veterinary. So what they found was that very quickly that they saw there was this PCV antigen. So porcine circovirus could probably be there. They were also able to grow this porcine circovirus from pigs, and it was immunostained with monoclonal antibodies. So certainly porcine circovirus was involved. It appeared. But first, the first antibody was only against the porcine circovirus 1, so PCV1. So that involved it. Then they could grow it and show that, well, there's homology to PCV1, but it is not PCV1. So something is different. And then they made reactivity with other PCV1 antibodies, and again, some worked, some didn't. And then eventually they found out that this was PCV2. It's only less than 80% homology to PCV1. So this is a new virus strain, and this must be the one that is involved. So then, of course, you want to prove Koch's postulates and see that this is actually the thing that is causing these diseases with lymphodepletion, inclusion bodies, porcine circovirus around these lymphocyte depletions, but actually also present very much in normal lymph nodes. So PCV2 was not invariantly associated with disease. This is a bit strange. So it's not like foot and mouth disease or classical swine fever. If you got the infection, you will be sick. Apparently, this is also present without giving disease. So they had this isolated and worked with it, and next, of course, wanted to see what happens in terms of infections. So they worked with the nosobiotic pigs. This was done in Canada in John Ellis' lab, and they wanted to prove Koch's postulate. They have this PCV2 virus. It's everywhere. It's really high loads and all these infections. So let's put it into pigs and see what happens. So first the study they did, they reproduced the infection. But unfortunately, it looked like there was porcine parvovirus contamination in this tissue homogenate and then subsequent culture that they did. So they needed to reproduce it again, and then they had a pure PCV2 isolate, and nothing happened. So this was, of course, really strange because PCV2 was the cause, and porcine parvovirus alone did not give any indications like this. So really an understanding challenge, and also remember everybody in the porcine immunology and virology world said that porcine circovirus is not anything to do with disease. So there was a lot of inherent resistance from both immunologists and particularly virologists that this had anything to do with a causative agent. But what happens then is that maybe this was, and then they proved that it needs to have a co-infection. So the contamination with a small amount of parvovirus in the first inoculation, that was what triggered the infection. So you need to have something else along with PCV2 in order to get the infection. So why does that happen? This is an example from pigs that were co-infected with PCV2 and porcine parvovirus. And you can see the porcine parvovirus is coming out in days after inoculation, so relatively shortly, where the PCV2 is then coming on after the first infection. So a strange set of series where the one infection paves the way for the other one. And of course, the natural explanation would be then it's immunosuppression, then that gives the way so that the immune system is less functioning, and then the PCV2 can come in. And that obviously it's the exact reverse for PCV. So porcine circovirus 2, the PCV2, it actually takes advantage of the immune activation. And Steve Krakauka, he had an idea that what happens if we actually do an infection where we inject keyhole limpet hemocyanin, this is just an antigen, together with incomplete Freund's adjuvant, so in an oil and water adjuvant. This is just an immune stimulation, it's got nothing to do with infection. And what happens is that if you do that, if you give only the PCV2, nothing happens or very mild happens. But if you give it together with a strong antigen, you actually get really strong clinical disease. So this is contrary to everything we believe in immunology, where you want to have the activated immune system to combat it. Here it's actually activation of the immune system also gives a portal of entry for the PCV2 virus to give significant disease. So this is really strange, but it proved then that what's actually going on in here is that some infection is paving the way for PCV2. But why does it then come suddenly at this point? I mean, why does that come in 1995 to 2000 all over the world with an infection that apparently had been there actually, and they could show that it was not a new emergence. So really, really strange. This is the example again, PCV2 together with antigen, and this is a log scale, so you get huge amounts of virus together with an immune activation and much less without the immune activation. So some kind of activation of the immune system is what drives it. So now they know that PCV2 is the causal agent, but something else needs to happen. And then we actually come back to this epidemiological triangle that there are things in the management that may prevent disease, but there are also things in management that may promote disease. And that may actually be it. Could it be that there are some changes, and I don't know enough about swine husbandry to say what has been in focus at this time point, but we know that early weaning has been something that has been pushed and pushed and pushed all along. Maybe also the new generation of vaccines where you can vaccinate them very, very early, so you actually get an immune activation that will pave the way for a PCV2 activation. And some farmers saw that they have severe outbreaks of PCV2 when they started a new vaccination regime against other infections at a very early stage. So there are lots of different things that are all associated with management and things that we are doing that we thought was good for the pigs, but actually in this case was really detrimental. So there are two ways to then approach it. You can either change your husbandry, and of course all these things were implemented because they had other benefits. So even though you have these MADAC possibilities to limit it, what about making a vaccine? And that is of course something that can be done. The autogenous vaccines and similar to the backfeeding that you're doing now with the PED was the first thing, but very quickly there was commercial vaccines thanks to Merial willing to go into that area and develop the first vaccines. So again, you can vaccinate 2 stages. The disease is post-weaning, so it is right after they are taken off from their mothers and put into that stress situation. But maybe you can actually prevent it by vaccinating the sow so that the colostral antibodies in the milk will protect the pigs. And that was the first approach. That way you have transmission of passive immunity, and you get actually a really nice return of investment because you vaccinate, of course, only one animal that have maybe 14 or 18 offspring instead of vaccinating 18 animals in the piglets. And maybe you can do both. So that was what they did. And again, this is again where it becomes really annoying for an immunologist because it was a chronic intracellular infection where something else promotes it. So we would say we need to have a T-cell response. I mean, there's absolutely no way this could work with a killed vaccine put in a simple adjuvant, oil and water, but it did. So the first Merial vaccine was something that I would have sworn wouldn't work. It's just an inactivated PCV2 in oil and water, and it works beautifully. Newer generations have made chimeric vaccines or one-shots and also even baculo recombinant viruses. They all work nicely, but it's annoying sometimes when an immunologist entering the field of vaccinology, where you want to be able to explain everything, and then it actually just turns out that it works in spite of everything you wanted. But anyway, it's really good, and it's now being used both in sows and in piglets, and works both places. This is an example of sow vaccination. Mortality in 7 different farms before vaccination and after vaccination. It's not, so mortality not in the sows, of course, but in the weaners. And it is significantly reduced. Also here in fattening pigs, again, significantly reduced mortality in vaccinated farms. So really nice effects from sow vaccination, and also if you're vaccinating the piglets, you can see that the mortality percent is significantly reduced in all these different farms. So it's really a nice example of a single agent vaccine, even though it works together with another infectious agent, that it gives a nice protection against the disease. Other examples of how it works, that's not so important. So even antibiotic use has been nicely reduced in this case, because of course once you have these multisystemic disease complexes you want to treat with something, you have following bacterial infections afterwards, you want to do whatever you can. So really significant, for instance, UK costs here very quickly 1/10 of the costs per animal in antibiotics. So that's really a success story for PCV2. It's probably one of the most successful stories in vaccination very quickly. And because of this, the syndrome of having an effect with other infections and other things, there was even a benefit beyond what was expected. So it protected, it even gave better production parameters without clinical disease, and as it drives the swine industry. Boehringer Ingelheim made a huge building in around Hanover based on their PCV2 vaccine sales. Other companies have really been able to put some more money into veterinary vaccine development, because this was such a huge success that it was something that can be used a lot. So very fast development of vaccines. We've got other infections where we're still struggling to get something that's been around for decades and centuries. And this one was basically 11 years after the first case controlled the vaccine was on the market. And at least now we feel that it is pretty much under control. So with that, I'd like to thank some of my colleagues at DTU from the work. Lars-Erik Larsen is our veterinary virologist. And then, of course, Gordon Allen, who has been instrumental in the whole vaccine PCV2 story here. And I would have given a completely different talk if it hadn't been for what he provided me. So any questions? This is, of course, the big equation. You don't need to solve it now. You can think about it. But otherwise, I will be happy to answer.
Thank you so much, Dr. Jankersen. Yep. Okay, I will summarize your talk a little bit, and then we go to Q&A session. Okay. So, Dr. Jankersen นะครับก็เล่าให้เราฟังถึงแบบภาพรวมของการใช้วัคซีนในการคุมโรคในยุโรปนะครับโดยอย่างแรกที่เราต้องรู้ก็คือว่าวัคซีนมีหลายแบบนะครับเราเลือกใช้ว่าเหตุผลต่างๆกันอาจจะเป็น prophylactic วัคซีนคือวัคซีนที่เราเข้าใจกันอยู่แล้วว่าฉีดเพื่อกันโรคหรือจะเป็นวัคซีนที่แบบใช้ในกรณี emergent เช่น ring vaccination ครอบคลุมบริเวณที่เกิดการระบาดนั้นอยู่นะครับหรือจะเป็น post exposure vaccination เช่นเหมือนในกรณี rabies vaccine นะครับแต่ทีนี้วัคซีน efficacy แน่นอนเราหวัง protection เราหวังให้การแพร่กระจายของโรคมันลดลงซึ่ง parameter หนึ่งที่เราต้องดูก็คือค่า R naught นะครับ R naught ก็คือว่า1ตัวแพร่ไปได้กี่ตัวนะครับเราต้องพยายามลด R naught ให้มากที่สุดโดยยิ่งน้อยกว่า1ก็คือแปลว่าวัคซีนเนี่ยใช้ได้ผลแล้วในการคุมการระบาดของโรคนะครับแต่ R naught เนี่ยมันแตกต่างกันไปในแต่ละโรคขึ้นอยู่กับว่าโรคนั้นๆ contagious แค่ไหน มีการแพร่กระจายได้มากแค่ไหนเช่นมีโซโรคหัดเนี่ยแพร่เร็วมากเป็นแอโรโซลนะครับเหมือน FMD อย่างเงี้ยแพร่เร็วมากเราก็จะสูงมากนั้น coverage ของวัคซีนจำเป็นต้องสอดคล้องกับ R0 นั้นๆถ้าเรา coverage ไม่ถึงยังไงก็กันไม่ได้นะครับซึ่งดรยังกเซนก็ยังได้พูดถึงโมเดลของ CSFV นะครับว่าที่เดนมาร์กทำยังไงแล้วก็ได้พูดถึงดีว่าวัคซีนนะครับดีว่าวัคซีนเนี่ยคือวัคซีนที่สามารถแยกได้ระหว่างการติดเชื้อตามธรรมชาติกับภูมิคุ้มกันที่ได้จากการทำวัคซีนนะครับอย่างในบ้านเราก็คืออย่างเช่น FMDV เนี่ยเราใช้ 3ABC ELISA kit ใช่ไหมครับเพื่อแยกว่าแอนติบอดีต่อ FMD เป็นจาก vaccination หรือจาก infection เหมือนกันก็คือในเดนมาร์กซึ่งเป็นประเทศที่ไม่มีการใช้วัคซีนเลยเนี่ยถ้าไม่มีดีว่าวัคซีนเขาก็จะไม่ใช้เพราะเขาแยกไม่ได้เขาใช้แอนติบอดีในการ surveillance ในประเทศเขาว่ามีการระบาดของเชื้อหรือเปล่านะครับแล้วเขาก็โชว์ให้ดูว่าในเดนมาร์กเนี่ยเขารับมือการระบาดแต่ละครั้งยังไงเป็น by the book เลยคือปิดฟาร์มไม่มีการเคลื่อนย้ายอะไรเงี้ยนะครับแล้วเจอเมื่อไหร่ทำลายทั้งฝูงซึ่งบ้านเราก็ต้องลองพิจารณาดูว่าจะทำได้หรือไม่นะครับแล้วก็ได้พูดถึง PCV2 นะครับซึ่งเป็นเชื้อที่ค่อนข้าง against กฎต่างๆที่เราเข้าใจกันมาตลอดแต่ก็เป็นหนึ่งใน successful story ที่นำไปสู่การผลิตวัคซีนที่มีประสิทธิภาพมากมีหลายยี่ห้อให้เลือกใช้แต่ทุกยี่ห้อมีประสิทธิภาพในการป้องกันโรคได้หมดนะครับแล้วก็นอกจากนี้ยังช่วยลดการใช้แอนติไบโอติกได้ด้วยเพราะว่าหมูไม่ป่วยนะครับโอเคน่าจะทั้งหมดแล้วมีคำถามไหมครับ Are there any questions any okay
Dr. Jungersen, Denmark is usually presented as a benchmark for good practices, at least in swine. High prolificacy in sows, high conscience in terms of prudent use of antibiotics, good biosecurity practices. But I would like to come back on one point that you mentioned on your presentation. You shared that 20,000 to 45,000 samples of CSF are tested as part of a surveillance program to basically validate year after year the negativity of the disease in the country. Could you share with us if this is an initiative that is being funded by the government, by the pork producers, a mix of both, for us to understand better what's behind this Denmark way of dealing with disease?
Yeah, that is funded solely by the government. So we have a national veterinary contingency funding that has been funded the Veterinary Institute for many years, and now it's actually being moved to sort of what you call One Health initiative. So it's being taken in together with the human health surveillance. But that is so certification of freedom of disease. That is funded by the Danish government. But there's also a lot of biosecurity measures that are funded by the pork producers. So in Denmark, the pork producers are working together. So 70% of all pigs are actually in one community. And for instance, any lorry that is going transporting pigs to Germany or Ukraine or Poland, when it comes back, they have made dedicated washing stations so that it will be completely disinfected before it's driven in again in the country. And that's actually not funded by the government, but funded by the pork producers. But the surveillance and documentation of freedom of disease is funded by that. But then we're using samples that have been sent in as part of the pork producers' own security. So we take a subsample of all those that are sent in for diagnostics or part of the slaughterer's own funding of SPF system that we then use to document freedom of disease. So it's a little bit of both, but it could not be done without an active government funding.
Very impressive for your presentation. I have two questions. One is because now you can show that in Denmark you have eradicated many diseases. So the first question is now in the farm in Denmark, what is the common vaccine program that used in the farm? And the second question is that also you need to know about the immunity status and also vaccination status to the farm. So what is the method or what is the tool that you use this in Danish farm?
Yes, if you hang around to the afternoon, actually the last talk about antimicrobial resistance and vaccination, I will give an example of a vaccination program and how to do that. So I think we can take that discussion if you have any questions after that. Because that's also a little bit practical of what we are doing with example of how to get the best vaccine response.
Thank you. My question is too soon. Sorry.
We have about 1 minute for one more question. Are there any? โอเคครับอันนี้ผมจะสรุปคำถามของคริสเตียนให้ฟังก่อนนะครับคริสเตียนถามว่าเจ้าการสกรีน CSFV ในเดนมาร์กเนี่ยที่แบบทำปีหนึ่งหลายหมื่นตัวเนี่ยใครเป็นคน funding ใครเป็นคน behind ใครเป็นผู้อยู่เบื้องหลังการสกรีนขนาดนี้ก็บอกว่าเป็น government แต่ก็มีหลายๆกรณีที่เป็นทั้ง pork producer ทั้ง government ร่วมมือกันในการสกรีนเหล่านี้เพื่อร่วมมือกันในการ eradicate โรคไปจากประเทศนะครับก็เป็น initiation ที่น่าประทับใจนะครับส่วนคำถามของพี่หมอเบสนะครับก็คือถามว่าอยากให้โชว์โปรแกรมวัคซีนในเดนมาร์กหน่อยนะครับดรยุงก็เซนก็เลยบอกว่าอดใจรอไว้เดี๋ยวนึงเซสชั่นสุดท้ายเนี่ยเราจะโชว์ให้ดูว่าโปรแกรมในเดนมาร์กเราใช้อะไรบ้างนะครับก็ขอให้อยู่ด้วยกันทั้งวันนะครับจะได้เห็นโปรแกรมของเดนมาร์กกันนะครับก็ I think it's just about time for this session. Thank you so much, Dr. Youngkersen.
สวัสดีค่ะ Good morning. Another 15 minutes and we are into to the afternoon already. So I'm not trying to keep you so long because I understand everyone is quite hungry right now. What I'm going to discuss today is that I'm going to discuss about the work that we've done here on vaccine research and development. And unfortunately, the model that I'm working with is not as good as the PCV2 vaccine. So we see a lot of problems during the research and developments on PRRS vaccine. And I'm trying to introduce you a little bit about our concept of vaccine, which may be a kind of unconventional thought of how we develop the vaccine, but I hope that would actually incorporate well with the mainstream vaccine development in the futures. And then with I have the opportunities to work on, analyzing the vaccine development within this Southeast Asian region. So what I'm going to do today is also trying to provide some basic information, how we're doing in terms of vaccine manufacturing in these regions, and hopefully in the future we can actually discuss more on how can we actually improve vaccine manufacturing capacities in Thailand, this is the developing countries, one of the developing countries that we are going to focus on. So yes, I'm going to discuss more on today. The outline would be PRRS vaccine development at our laboratories compared to others, and also to look at my view on vaccine research and developments and manufacturing within this region. Okay, everyone's probably know PRRS, porcine reproductive and respiratory syndrome virus, very diverse RNA virus. We have both type 1 EU strain and type 2 US, and recently it's been an outbreak of the highly pathogenic PRRS, which has been circulated in this region quite a lot. And most of the clinical signs would be related to the reproductive failures in the breeding herd, and also respiratory infections that leads to porcine respiratory disease complex, which is probably the big problem that we've seen or observed in the swine farm in this region. Now, let's look a little bit on, provide you some background information about immunology of PRSV, which I think is quite interesting. When you have the virus infection, usually viremia lasts approximately only 1 month. Then the virus go into the tissue and they stay there forever, probably most of the lifetime of the pigs. If you look at in terms of immunology, immunologists and thinking about this infection, we think that this is quite interesting because why didn't the virus was clear out of the infected pigs? And we don't know why. So people start to look at how the pigs response to the viral infections. And what they found is quite interesting. First is that usually when we get infected with something, we have the immune response, both innate and adaptive immunities response to those infections. And usually the innate immune response comes first, very early, during the first few days. You have inflammation, you have activities of phagocytic cells, and a lot of pro-inflammatory cytokines produced within the body of the host. But if you look at the PRRS infection, we'll see that most of the time we cannot really detect much of this innate immune response. Very poor pro-inflammatory cytokines productions. And when we look at the adaptive immunities, it's even more interesting. Yes, we do have some non-neutralizing antibody response that we use it for testing the virus exposure, the IDEX ELISA. That was detecting the non-neutralizing antibody, which has been produced during the first week, 2 weeks of the infection. However, this antibody is not protective. They're actually helping the virus to go through the target tissue better. So this is actually not good for the host. The neutralizing antibody, which is supposed to be the good one, right? Blocking the virus to get infect to the cells of the host, is actually come very, very late, and actually after the viremic stage has gone. So the virus has already completed its infection cycle, it's gone already. And then we just wake up and producing neutralizing antibody. And it's very low and peak around 2 months following infection, and usually the level of neutralizing titers always quite low. 1 to 8 is good enough for PRRS immunology, whereas you can get 1 to 3,000 for after classical swine fever infection. So it's very low for PRRS infection in terms of the antibody productions. Cytotoxic T cells and cells producing interferon gamma, which we consider that as one of the parameters or indicators of the cell-mediated immunities, are actually building up very late as well after the viremia. So very weak and very slow induction of adaptive immunities, and that's why usually when pigs get infected with PRRS, nothing can stop them because there's no effective immunities built on time at the time of the viremia. So that's the problem. Now, people start asking questions. This is not like when we get infected with some other viruses. It's really different. It's very slow, and it's quite unexpected to see this pattern or profile of active immunities mount following any type of virus infection. So it's very unique. We and other research groups also look at what's going on or the interaction between the host and the virus in terms of immunological response. And there are several reports from our group and also from other part of the world that actually suggest that the virus is actually very good in terms of they tend to induce a very strong immunosuppressive factor, difference of several kinds of them at the early stage of the infections. We report that the virus actually induced a very strong IL-10, which is a very potent immunosuppressive cytokines at the early stage. First 2 weeks of infection, we have a high level of IL-10 within the system of the infected pigs, and we also went on characterizing the protein that's responsible for IL-10 induction, which turned out to be the nucleocapsid protein of the virus. I'll come back and discuss about this N protein later in our work. Apart from there, we know that induction of T cells in the presence of high IL-10 in the microenvironment usually leads to induction of regulatory T cells. This is one of the suppressive cells that Bill has already mentioned in his lecture before the break. And yes, of course, we show here that PRRS without this IL-10 can actually effectively induce regulatory T cells in the infected pigs, and that lasts quite long, and that's probably involved in suppressing of the active immunities following PRRS infection. And of course, when you have PRRS, IL-10 kick on, and you also have some other immunosuppressive factors and mechanisms that's been identified by other research group. We recently confirmed that there's also another one, immunosuppressive cytokines induced at the first few days following infection, which is the IL-1RA. And that's another type of immunosuppressive cytokines that we've been working with recently. So we know that whenever PRRS get infected to the pigs, all the immune cells, they're going to sleep, because we start suppressing the system at the beginning of the infection cycle. So that's a problem. It's difficult for us to try to think about how can we fight with these enemies. For example, if you think that we maybe immunize the pigs with the vaccine, and then we have memory cells that we talked about early before the break, that if we can actually build a good memory cells, maybe when the pigs, the vaccinated pigs get infected, these memory cells would then actually become activated, expand, and then work, protect the pigs from infection. However, with these scenarios, even though you have a very strong memory cells mount from your vaccination, then you're going to face one problem, is that you have a very strong immunosuppressive condition at the early stage of infections, and therefore your memory cell will not be able to get activated properly, and therefore it's not going to work in this situation. So this is a very unique immunological setting that we think that by understanding more on immunology of this particular virus, it gives us more understanding why vaccine doesn't really work in this situation. And unfortunately, we know that for PRRS, we have both kill and modified live vaccine available in the market. And people may ask, what about the modified live vaccines? Are they giving us the same situation or same profile like what the local or field strain give us? The answer is yes, unfortunately. Those modified live vaccines still keep some of the characteristics on induction of this IL-10 and Treg. And therefore, you also have some kind of induction of immunosuppression following the modified live vaccine as well. And that's what you've seen in the field, where when you have modified live vaccine and you give modified live vaccine PRRS to the pigs, sometimes you see some immunocompromised stage and come followed by some secondary complication. And we've seen that in the field as well. So this is a very different scenarios. It doesn't look like PCV2, which seems to be working very well with just the Q vaccine. And we are actually working with the virus that's very clever. And this is just what we know. Maybe there are some more factors that we haven't been able to identify, that we suspect that there are more for the virus. We just recently show as well that of course, we show that PRRS infection induced Treg in the systemic, in the circulation. And people ask, well, are they in the lung? Are they working, or are they suppressing the response in the lung as well? So we just have one recent publication coming out and show that in PRRS infected pigs' lung and tracheobronchial lymph node, you see a lot more PRRS specific Treg in the lungs, and they are also producing a lot of IL-10 in the lungs. So that's also explained why you have active PRRS infection within the lung, and you have these scenarios of having a lot of Treg in the lung, and therefore you start having, seeing the clinical signs of secondary complication because of the immunocompromised condition within the lung. So these are our basic or current understanding on how PRRS affecting the pig's immune system and also affecting the local immune defense mechanism at the target organ. Now, how can we control them then? This is a very difficult question, right? And I think a lot of people want to know as well, how can we do that? The answer for me is that we still don't know, but we need to test. We need to think about what can we use to help controlling this type of immunological pictures occur following PRRS infections. Now, of course, there are so many vaccines and research on PRRS vaccine in the literatures. of course, modified live vaccines available in the market. Q, we have that. Subunit, DNA vaccine, and viral vectors. Those vaccine technology has been used or has been studied for making a better PRRS vaccine. So far, we still don't have any good or better candidate than the modified live virus. And of course, the modified live virus, they show to have a good effects or effectives for providing a good clinical protection, but they don't really give good complete protection like other vaccines. And of course, because PRRS are so varied in terms of their antigenicity, and therefore they protect only against the homologous strain and not the heterologous strain. So we still have problem with the modified live vaccine, that they are not really well protected across PRRS strains. And also, as I mentioned before, that these modified live virus also have some negative immunomodulatory effects following immunization. So you have to be careful, you have to be thinking thoroughly before implementing this modified live vaccine within your farms, whether the strains are matched, whether your pig's conditions are okay, or how can you prevent the loss from partial immunosuppression following MLV vaccination. So you need to think about that quite a lot when using the vaccine. So therefore, I think we need a better PRRS vaccine, but we don't know how. Because there are different pictures in terms of the host and virus, the virus and the immune system of the host interaction. Most of the literature would still suggest that yes, the best we can do now is still the modified live vaccine, but I think we need a better vaccine than what we have. Okay. Now let's think out of the box, and this is what I'm going to lead you to our vaccine, which I say that it's an unconventional thinking about how to do vaccination. Let's be kind of think about this. If these horses are the immune system, and this is the PRRS virus, we have the horse that trying to run to their would like to run forward. But then we have this virus pulling the horse with all kinds of immunomodulatory factors, right? So most of the time when people say, well, let's vaccinate, when you do vaccination, this means like you actually trying to whip the horse to run faster, right? Because you want more antibody, you want more cytotoxic T cells or memory helper T cells. You want to whip the horse to run faster. But you forget one thing is that the horse is actually tied with this immunomodulatory factor, with this string that actually they're tying off, and they cannot run as fast as we want, or in the good direction that we would like to, right? How can we make the horse go faster? If you look at this picture, how can you make the horse go faster? The first person said, well, you can shoot this guy, right? You can shoot this guy, they fall off, and then the horse can run, right? However, we have a lot of this guy, right, in the farms, right? So this guy is down, the other guy is going to come up, right? And they still pull our horse. The other way to do, easy way to do, like for this type of comparison, is that you can just actually cut this rope, right? So if you cut this string, there's no control between the horse and the bad guy. The horse run, right? So in the direction that we want. So that's the way that we think that it may be possible. Let's forget about protection, but let's prove one hypothesis. Can we actually get rid of that string and prove that the immune system is actually working better when we take off those strings, right? So that's my hypothesis. I'm not going to say that I'm going to produce a universal PRRS vaccine, but I'm going to test my hypothesis that if I take off those strings, can the immune system work and control PRRS as it's supposed to be? Now, apart from highly pathogenic PRRS, those conventional PRRS, those old style PRRS, they don't really kill the pig, right? They are just annoying, right? They're just there, and they just kind of mess up with the immune system. They induce more secondary complication, right? So we hope that, I mean, if the immune system is working well, they should be able to clear the virus by itself. Maybe we get a better clearance, we have less complication. That's my hope. Okay, let's test the hypothesis. Okay. This is just to explain to you, most of the people will do that, right? You have this infection with induction of a lot of immunosuppressive factors, and we have effector cells, the efficient immune response on the other hand of this balance. So most of the people would do like, okay, let's immunize, let's try to induce antibody, let's try to induce cytotoxic T cells. However, if once you get the memory cell prime, but then when you get infected with virus, these things still win over those vaccines. And I say, like, how if I get rid of this one, I hope that my immune system or the pig immune system will actually work much better, and then maybe they'll be able to control the virus. So let's prove that. How can we prove that? So that's one thing. I use Star Wars a lot because I went to give a talk in one talk, and they asked me, like, what is the theme of your talk? And I said, well, maybe The Force Awakens would be a theme of my talk. I really like Star Wars, so I think that's a good one. So that's why there's a lot of Star Wars things in this talk. So sorry about that for if you, it's maybe too complicated. So we mentioned before already that in our laboratories, we identify or characterize protein that is actually responsible for IL-10 production. Now, for immunologists, we don't really have technology to map the Treg epitope right now, but we hope in the future we'll be able to do that. But at least we know that this protein is really important for induction of IL-10. And we know that in immunology, IL-10 is actually important for induction of Treg. And we know that, and we proved that, published that already. This is very interesting antigen. The nucleocapsid proteins is encoded by ORF7. It's very highly conserved, good for us, because it doesn't matter EU or the US or the high pad or the low pad, they are all conserved for the nucleocapsid protein. They express at a very early phase of viral replication. It has nuclear localization factor, don't know why. The most abundant virions and the most immunogenic protein, however, it's not protective. Right? We know that it's very immunogenic. It's produced a lot at the early stage of infections and is actually responsible for IL-10. Now we can see that maybe we should try to reduce the activities of this nucleocapsid protein by induction of effective immune response against this particular protein. And here's just to show you that how efficient this protein can induce IL-10. We just take the gene of the virus and then transfect them into the PAM cells, and then measure the level of IL-10 production in the cells. So you put the genes, the nucleocapsid genes into the cells, and then you have a very strong IL-10 productions for both nucleocapsid protein from the US or the EU strain. So both strains, nucleocapsid proteins, induce very high level of IL-10. This red bar is actually the positive control. It's the cells being stimulated with LPS, which is the positive control. And if you see that the level of IL-10 is pretty much the same as our positive control, it's very strong IL-10 inducer. Now, lucky that we also searched some more literatures, and there's literatures suggest the structure of this nucleocapsid protein. And what they suggest is that the last 10 amino acids at the end of the protein, it's actually very critical for forming the conformational of the proteins, which means that if you remove this 10 amino acid at the end of the protein, then this protein will not form conformation as it's supposed to be. It becomes just a single it's a polypeptide chain. So it lost all the conformation, but it still maintains these polypeptide chain structures. And we test that, how if we remove these 10 amino acids, how the IL-10 induction would happen. And we see that we have significantly less IL-10 production in this linear form of the protein instead of the conformation. So this is another hint. We know that conformation is important. And also we know that if we have the protein, but we don't make them form as it's supposed to be, the ability to induce IL-10 is also reduced. Okay? So what should we do next? Of course, the strategies to develop the vaccine against this nucleocapsid protein. Now we know that, of course, this is the important one. It is responsible for IL-10, so we're trying to induce immunities against the immunomodulatory proteins. We are not talking about protection. We just try to reduce immunomodulations. And we know that in the farms, when you have serological surveillance on PRRS infection, most of the test kit, those IDEX ELISA tests, are actually testing against the antibody against nucleocapsid protein. So designing this vaccine, we will have to think about this take this into our consideration, that we do not want to induce immunities that will actually confuse the farmers, right? If we have the full protein, immunize them, then pigs have antibodies against N nucleocapsid protein. That's going to confuse the farmer whether this is from vaccination or from infection. So what we designed is that we took the plasmid DNA got from Lon Balbeek, my advisor from Saskatoon, and then we insert the gene encoding the linear nucleocapsid protein. So it's the nucleocapsid gene without that 10 amino acid. So our plasmid will actually encode truncate nucleocapsid protein in the polypeptide chain form. So it's not forming conformation. So theoretically, it should not induce any antibody detected by those IDEX test kit. So we are actually trying to induce a T cell response against the immunomodulatory protein of the virus. Okay. So we test the hypothesis. Plasmid DNA or DNA vaccine is very simple. Once you clone it, you purify, propagate them, purify them, and you can test it in the pigs. So what we test is that we use the PRRS seronegative pigs and immunize with the DNA vaccine twice, and then challenge them with the homologous strains, the same virus that we cloned the nucleocapsid gene from at day 56. And what we've seen starting early after challenge, we start seeing, first thing we want to test is that, of course, we don't want antibody response or priming of antibody response by our plasmid DNA. And we proved that this truncated gene did not prime for antibody productions. And good signs, we start seeing some good signs, is that this plasmid DNA is actually can help enhance the virus clearance. So with the immunization with truncated form of nucleocapsid gene, you start seeing a better viral clearance in these infected pigs. Now let's see at the immunological response. We see reduction of Treg, reduction of IL-10, which is what we expect. And we also see improvement of interferon gamma producing cells. Now, we don't know whether it's a CTL or not, but at least we use that as an indicator for cellular immunities in our system. So therefore, with this hypothesis, we're now testing a hypothesis and proving that. I mean, it will actually induce the immune response against the immunosuppressive protein or the nucleocapsid protein of PRSV. You can actually enhance or improve the immune response. The horse is run faster now. They are running into the direction that we want. And we later on proved in several clinical trial and field trial. From that earlier study, we then proved that using the DNA vaccine, we can actually modulate the immune response against PRSV and shorten the time of the viral clearance time. We know that our DNA vaccine can induce a very strong cellular immunities and reduce the immunosuppressive parameters. And in the past, we used like intradermal vaccination at the ear penis, which is very laborious. And I guess the 2% is also in this room. So later on, we changed. We collaborate with Muriel. This time, right now is BI right now, but Muriel has transdermal injector, which is the needleless injector. You can actually introduce the DNA vaccine into the skin transdermally of the pigs, and then it works the same. So later on, we test it with the transdermal vaccination. It is working as well. So we know that with this concept, we now being able to adjust the immune system and try to bring them back to work the way that we want. Okay, what's next? The horse is running now, but it's running at the, I don't know, any direction that they want. We cannot control the direction, right? Our novel DNA vaccine does not contain the protective epitope. It just reduced the immunosuppression, right? So how can we actually incorporate this into what we have now? And maybe we can actually have the vaccine that is help reducing immunosuppression and also have a good directions on mounting a proper antibody response and CTL against PRRS within the field. Even immunology, there's one of the terms that people usually say it's about prime boost strategy, right? If you maybe you've heard about that. Priming with one type of vaccine and then boosting with another type of vaccine. And hopefully you can actually have a good effects on induction of effective immunities against PRSV. Of course, we already have commercial modified live vaccine in the market. Unfortunately, this vaccine still also induce Treg and IL-10, although not as strong as the few strain. They also show some delay vaccine-induced immune response and some safety concern. So we're asking the next question, whether if we can use a prime boost strategy, first prime with the DNA and then boost with the modified live vaccines, whether we can improve the immune response against PRSV in the vaccinated pigs. So this is just a simple experiment. We do the prime boost using we prime the pigs with DNA vaccination 2 weeks earlier, and then followed by vaccination with modified live vaccine, and then we challenge them, I think. No, sorry, this one is not challenge. We just follow the serological response in these vaccinated pigs. And as you can see that with the prime boost strategy, you can actually have even higher SP ratio, higher level of antibody productions, and the consistency is much better. And by 2 weeks following modified live vaccination, you have 100% of the pig seroconverted. Where if you only have the modified live vaccine only, you have only maybe 60 to 70%. So we can actually improve the magnitude and also the consistency of the antibody response in these immunized pigs. If we look at the cellular responses, and this again using flow cytometry, we can actually follow the dynamics of cellular immune response within these vaccinated pigs. And the red colors is the modified live vaccinated pigs, and the blue color is the prime boost group. And if you can see that there's a switch of this, the response between the two groups, you have higher IL-10 and higher Treg in modified live only vaccinated group, where you have a very strong interferon gamma production in the prime boost. The prime boost control the IL-10 and still control the Treg induction quite well. So it's actually doing works like we expected. So I think it's at least in this experiment, we show that with this prime boost regime for vaccination, we can actually reduce the negative effects following we can reduce the negative effects and also enhance the cellular immunities following vaccination with the modified live vaccine. So this is good. I think in the farm, if we have just a regular PRS infection, use prime boost strategy, we should be able to nail it quite good, and we should be able to control the virus quite well. However, one day someone the company came and said, how if we test it with the HiPad virus, what do you think is going to happen? And this is actually based on Professor Rungroth projects that, I can't see how many, 5 minutes? Okay. that we're trying to test them with the highly pathogenic virus challenge and see what's happened. So we test that. As you probably already know before, aware before, that the highly pathogenic just recently emerged in these regions. It's on the lineage 8, and I think yesterday discussion was also, Dr. Rungrod also mentioned a bit about this highly pathogenic PRSV. So we test if this prime boost strategy will be able to control the high path virus. Okay. And the experimental design is quite simple. So prime with the DNA, boost with the modified live vaccine, and then challenge them on day 42 after modified live vaccination. If you look at the viral load, of course, following the modified live vaccine, you have some small peak of the viral in the serums. And then after the challenge, of course, the modified live vaccine, they don't do complete protections. So you still see, or you still be able to observe the increase in the viral titer, but actually they are reducing faster than the non-vaccinated group. Comparing head-to-head between the prime boost and the modified live only, not much difference. So bad signs for immunologists. Farmers not going to buy this vaccine, I think, because if you look at the clinical side, there's no difference. Yeah, if you look at the respiratory clinical score, not much difference between the modified live only or the prime boost. However, it's much better than the non-vaccinated one. These peaks are very well happy at 6 days following infection, which is very good, compared to the one that's the control group, which is still very sick after a week following infection with the high path viruses. And the lung scar lesion is pretty much correspond well to the clinical signs. So not much difference in terms of the clinical protections or virological protection. But let's look at the immunological part. If we look at the immunological part, just to show you here, the prime boost would be the green line, and this black line is the modified live vaccine only. As you can see that they are totally different in terms of the immunological features. We can still control the induction of Treg and IL-10 much better than the single immunization with modified live vaccines. And if we look at the interferon gamma production, we can still see that a much better enhancements of interferon gamma production in the prime boost vaccination. So in terms of immunological profile, we are very happy. But for clinical protection, it's not much different. Okay, so and look at the antibody response. And of course, the modified live is here, the prime boost is here. So we have actually better in terms of the antibody response following the challenge, and this is a neutralizing antibody titers, and we can show that there's a good difference in terms of the antibody anamnestic response following the challenge. So at a glance, I think in terms of immunology, the prime boost did it work, does its job quite well, and I think the way that we expected. However, if we look at the clinical signs and the viral load, it's very comparable between the two regimes. So for this, in this case, I think when you recommend that for the farmers, I don't think anyone will buy the DNA vaccine, right? Because it still gives pretty much comparable to the clinical protection. But if we look at the immunological change, I think with this prime boost, in the long run, we will have a better control of secondary complication and other infection in the later stage. However, because this trial, we were doing the challenge studies and we didn't keep the peaks very long, so we did not see those long-term effects. So it's just the short-term effects following the challenge. So the way forward, I think for PRRS, we need to think both. Like, how can we improve protective immunities, and how can we reduce the immunomodulatory effects of the virus? So I think PRRS is very unique, and we need to think about it in the different aspects as well. But also we show that the DNA vaccine is very powerful in terms of priming and also directing the response against the PRSV. So this is what we learned, and we hopefully that we will try to improve our vaccine, or at least trying to find some more or better improve how to improve our DNA vaccines or how to improve our PRRS vaccines in the futures. This vaccine has been now under the patenting process. We've been collaborated with Merville since 2009, so it's almost 10 years now. And so I'm sorry, I'm going to be a little bit late. Okay, and right now it's already got the patents in the US, and we are on the national phase in the EU and other countries. Now let's move a little bit to what we've seen for the vaccine development during the last few years. I have a chance to analyze some of the capacities of vaccine manufacturing in Southeast Asia in the last few years. And this is the project that we did with IDRCs that I'd like to share maybe a few of the information. Not a lot, but hopefully that will actually kick off some of the idea of we start thinking about vaccine manufacturing more in Thailand. As you can see that I think for the animal vaccine market is still a fast growing fields, right? You can see that with this in Asia and Africa, you can see that the combined annual growth rate is more than 10%. So it's still growing. It's very good, have a good potentials for development of this type of vaccine within the regions. If you look at the type of vaccine available in the markets, 40% would be either the live vaccines or the kill vaccines, and that's what we expected. We've seen that a lot in the market. And the rest is recombinant virus, around 10%, and toxoid, and then DNA vaccines start to come up as well. However, I like to point out that making vaccine is not like 2 months or 1 year project. It takes such a long time to produce or manufacture and get this vaccine into the market. And so you start from the discovery, preclinical trial, maybe clinical trial. This is what we're up to. We know that the vaccine is working now. But for the registration and the manufacturing, this is usually, it's a bottleneck here in this region. A lot of people think of a new vaccine, but it's never go into the market. And that's why, and one of the reasons is that it takes such a long time. It takes a lot of collaborations and a lot of information that need to be compiled together before the registration and also if you look at the manufacturing, it's even worse. I mean, if you have the vaccine manufacturer come in and present to you about how they test their vaccine, you will be amazed because it can take for one product maybe 6 to 36 months for producing the vaccine, and 70% of the time is related to quality control. So it's not that anyone who can grow the bacteria or virus can make the vaccine into the market, and that is totally wrong. I think you need a lot more expertise and a lot more information and a lot of quality control system to be implemented before you can actually license those vaccines and put them into the market. So manufacturing is one story. Put them into the market in another story. So there are a lot of steps before we can actually get the commercial vaccine out to the market. So the background is that around 2 years ago, I worked with the IDRC, the Livestock Vaccine Innovation Fund, which is funded by Bill and Melinda Gates Foundation and also the government of Canada. So we are trying to analyze the vaccine manufacturing situation in Southeast Asia and to identify and profile these manufacturers and look at some of their SWOT analysis to see some of the strength and weak point. So I'm not going to discuss or tell you all of the information that we've obtained from this project because some of them is confidential. But I'm going to try to compare some of the countries within this region in terms of their capability of manufacturing vaccine. And then we can think about what about Thailand? What are we going to do next in the future? So here are the pig production data in Southeast Asia from 2010 until 2016. And of course, Vietnam is probably the biggest pig producers, right? It's probably 50 million per year in 2016, like followed by the Philippines and Thailand, Indonesia, and Myanmar is pretty close, around 10 or 15 million pigs per year. So these are the size of the market within the countries. if you look at the numbers of manufacturer, and this is the vaccine manufacturer actually selling the vaccine in the market. Vietnam's got 5, Indonesia's 4, China has 8, Thailand has 2. One is, as we know, the Department of Livestock Development facilities. The other one is a private company, which is a very small, and they use it just for the pilot, not for selling. So that one is actually not counting. So we actually, for Thailand, I think we only have 1, right? So it's just public at DLD, which is quite old manufacturing plants already. If you look at the commercial market for this vaccine, where are they going? You can see that Vietnam, Indonesia, and China all have vaccine manufacturer with the licensed vaccine that actually export the vaccine to other countries. As I said before, that this is a big market, high potential, and these countries, our neighbor countries, are actually sending vaccine out and selling the vaccine to other countries already. Thailand, we hopefully someday. If you look at the available vaccine technology, of course, for the simple live Q, lyophilization, vaccine propagation, everyone seems to be able to do the same way. But if you look at the potential of the new high-tech vaccine in the future, China has all recombinant subunit at the reverse genetics technologies. Vietnam's already have 1 company that have reverse genetics technology, whereas Thailand and Indonesia, we still have nothing. So these are something to think about. Our neighbor is actually growing, and they are selling, making monies, and actually they are improving their product, the production through several technologies. About Thailand, what can we do? I think these are just my viewpoint. I'm not blaming anyone, this is just my feeling. So hopefully that this can actually create some discussion in the future that maybe we need to think about whether we can actually compete with those neighboring countries. For the setup, I think we are a tropical climate country, and we have all kinds of disease. We are very lucky, unlike Denmark, that they don't have any disease left in their country, right? But we are very lucky, we have everything we want, like virus, bacteria, parasite, protozoa, you have anything, you name it, okay? And we also have a very, we're not island, we have very dynamic movement of animals and disease within our regions. We have all kinds of newly emerged viruses coming in the last 5 years. You see a lot of new virus coming in this region. So the setup is that we are actually at the endemic zones. We are actually at the war zone of this fighting with those pathogens. If you look at the research, the vaccine R&D mostly focus or mostly done within the university, some university and some laboratory in the university. Of course, for conducting a field trial, we have advantage, we are cheaper. We have connections with farms, we have connections with, and we have several animal facilities that we can actually conduct the animal trial. So as you've seen from my example, that we will be able to do that, okay? However, we have very limited resource in fundamental immunology and vaccinology research. As we mentioned this morning, those tetramer things is not in the countries yet, but hopefully it's coming soon. We are discussing about how to collaborate and bring those technology into this region as well. If you look at the vaccine production, Thailand is really lag behind others, right? We only have 1 government-owned vaccine manufacturing plants. We have very limited and on the know-how in terms of the upscale vaccine production, and also the know-how in terms of the QA and QC process. We need more expertise on that, and I don't know. I mean, we need collaboration to get those expertise in. Policies, government, that's one thing that for medical field, they are more advanced. They have vaccine production plants for medical field with very high standards. So they are more advanced, but we can learn from them. I think this is one of the opportunities that we should think about that. However, we still have very limited and sometimes not very clear policies and directions on the vaccine research and development and the manufacturing in Thailand. Can we have our own private vaccine manufacturer in Thailand? I think with our knowledge we can do it, but with the regulatory framework, I'm not really sure. We need to discuss that with the authorities. And regulatory framework issue is very limited, so I think we need to do some studies or discuss more on that. So there's still opportunities. We are lagging far behind other neighbors' country, but I think there are opportunities on that. For the final thought, I think with this talk on the vaccine manufacturing aspect, I think despite the strength of the swine production industry, and we have very advanced veterinary vaccine research and practice, I think Thailand has limited resources in supporting regulatory framework for vaccine development and manufacturing. So we still lag far behind other neighbor countries. How can we solve this problem? And if we want to have a manufacturer in Thailand, how can we do that? I think, of course, there's an urgent need for capacity building. We still lack of a lot of expertise that we need for vaccine manufacturing. Public-private partnership and collaboration with the existing vaccine network could accelerate the speeding up the process of the vaccine R&D and manufacturing in the countries. And maybe in the future, spin-off companies from the research labs under the regulatory framework and good manufacturing standards should be explored. I think this is the way to go in the near future. But right now, we hope that I hope that this presentation would actually start to give you some information to think about that we actually have a good potentials on vaccine manufacturing, but we're still far behind, and there are several steps that we need to work on, and hopefully that's going to happen sometime, maybe in my lifetime, I think. So thank you very much for your attention. I'd like to acknowledge the university, granting agencies, also some other private company who's actually been helping us on the PRRS vaccine research, and also IDRCs on the way that to give me a chance to look at those vaccine manufacturing situation in Thailand. And thank you very much for your attention.
Thank you so much, Dr. Suratat.
อ๋อสรุปใช่ ลืมบอกเขาไปว่าจะสรุปเองค่ะก็ไม่มีอะไรนะคะวันนี้ก็คุยให้ฟังคร่าวๆในเรื่องของการพัฒนาวัคซีนในอีกมุมมองหนึ่งนะคะที่คิดว่าอาจจะเป็นมุมมองที่ไม่ได้เหมือนกับการพัฒนาวัคซีนในรูปแบบทั่วๆไปนะคะก็ให้เห็นว่าจริงๆแล้วเราต้องการความรู้พื้นฐานทางด้านภูมิคุ้มกันพอสมควรในการที่ทำให้เราเข้าใจเรื่องของ interaction ของเชื้อโรคกับระบบภูมิคุ้มกันที่จะทำให้เราสามารถพัฒนาวัคซีนที่มีประสิทธิภาพในอนาคตนะคะอันนี้ก็เป็นตัวอย่างที่อาจจะยังไม่ถึงขั้นที่ว่าเราได้วัคซีนสำเร็จรูปที่ใช้ได้เลยแต่ว่าอย่างน้อยมันเป็นข้อมูลที่เราจะใช้ในการประกอบกับการพัฒนาวัคซีนในอนาคตนะคะส่วนเรื่องพาร์ทที่2ที่เกี่ยวข้องกับการวิเคราะห์กำลังความสามารถของประเทศในเรื่องของความสามารถในการวิจัยและพัฒนาและการผลิตวัคซีนเพื่อใช้ไม่ว่าจะเป็นในประเทศเองกับหรือว่าส่งออกนะคะอันนี้ก็เป็นข้อมูลพื้นฐานที่นำมาสรุปให้ฟังคร่าวๆว่าในประเทศเพื่อนบ้านเป็นยังไงบ้างแล้วก็ในประเทศไทยเป็นยังไงบ้างนะคะแล้วก็เสนอทางออกไปบ้างแต่ว่าอย่างน้อยอันนี้ก็จะต้องใช้ความร่วมแรงร่วมใจจากหลายๆฝ่ายรวมทั้งจะต้องมีการเสริมสร้างบุคลากรที่มีความรู้ความชำนาญในด้านที่เกี่ยวข้องกับการพัฒนาวัคซีนการผลิตและการควบคุมคุณภาพอย่างเหมาะสมด้วยถึงจะสามารถพัฒนางานทางด้านนี้ต่อไปอย่างมีประสิทธิภาพแล้วก็ก่อให้เกิดประโยชน์กับประเทศประมาณนี้ค่ะขอบคุณค่ะ
And welcome back to the afternoon session of the first CUVC Swine Vaccinology Symposium, in which Dr. Goldie will continue his talk about the analysis of porcine immune responses to access vaccine performances. Please welcome Dr. Goldie.
Okay, so we're going to talk now about applying some of the things that we were talking about this morning to analyze vaccine performance in developing new vaccines. So again, my focus at my years at USDA Plum Island Animal Disease Center was foot and mouth disease virus and developing new vaccines against FMD. And just for anyone that wasn't here this morning, I'll quickly go through some of the characteristics. Foot and mouth is a very highly acute and contagious disease of cloven-hooved livestock. It's occasionally lethal, and actually strains that have caused outbreaks in recent years are unusually lethal for both swine and cattle. It induces a complex immune response. There's a lot of significant innate responses to the infection. And again, one of the challenges with foot and mouth disease vaccines is there's 7 major serotypes, many serological subtypes. So the antibody response is not protective against much other than the strain that you've been infected with or vaccinated against. So when you're dealing with outbreaks in areas and you're going to try and control disease with vaccines, you have to match your vaccine strain to the strain that's causing disease in the outbreak very carefully. These are positive strand RNA genomes, and they mutate really rapidly. And the structural proteins are the key to the icosahedral capsid of the virus, and that's what the antibody response is against. So again, the killed virus vaccine was developed in the 1950s off of the Salk vaccine for polio. These both FMD and polio are picornaviruses, so the same technology worked in both cases. There's 2 adjuvants used commercially today, either double oil emulsion or aluminum hydroxide and saponin. This is for the IM vaccine. This is for the subcutaneous vaccination. And it's a classical needle delivery. I've already said that. So it's chemically killed virus and adjuvant, and there's very little cross protection. And protection lasts about 6 months. So there's been a lot of attempts over many decades to make a better vaccine than the killed virus vaccine, and all of these have been put into clinical trials and failed. Peptide vaccines, DNA vaccines, replicons, subunit vaccines, which are bacterial expressed proteins, plant expressed proteins, many different platforms that various groups have tried, and none of them have reached commercialization. And again, going over a little bit of what I talked about this morning, at Plum Island Animal Disease Center, my colleague Marvin Grubman developed a new recombinant vaccine that's based on the vaccine vector, which is a replication defective human adenovirus, serotype 5, that is used to deliver the structural proteins of FMD and the 3C protease that processes those proteins to allow the capsid to form. And so basically you end up with empty capsids, and it's a DIVA vaccine because the diagnostic is against a non-structural protein, 3A and 3B. So the vaccine does not contain those proteins, so we have a diagnostic that can differentiate infected from vaccinated animals. Onset of protection is 7 days. It's actually a little shorter than that. You can get partial protection as quickly as 4 days, but the protection seldom lasts as long as 6 months. So the classical killed virus vaccine that's been around for 70 years and this new adeno vaccine that we developed at Plum Island essentially have the same performance characteristics. The big advantage being you don't have to grow billions of live virus to make your vaccine like you do with the killed virus. And we have this DIVA capacity in the recombinant vaccine. So again, reviewing a little bit from this morning, you essentially are making the same thing in both cases. So you grow up live virulent virus and then you chemically inactivate the genome, you end up with an empty capsid, or you inoculate the adenovirus vector that has the polyprotein for the structural proteins and the 3C protease that processes them into the different components of the capsid, and you essentially end up with the same thing, an empty capsid. And that makes for a very efficient B cell response, but has all of the problems I spoke about this morning. Basically, you're not inducing any immunological memory. You're just inducing a very short-lived B cell response. And so your protection is based on the half-life of antibody in the serum. So this is a cartoon of what happens. The naive B cells are stimulated by the empty capsid. The B cells expand, but there's not enough antigen to keep driving B cell differentiation to memory. So there's just a short-lived expansion, differentiation to plasma cells, and production of the antibody against the virus. And again, we've developed a system to analyze the B cell responses by putting a tag on the virus and using that as a probe to look at B cells. And when we look at the presence of antibody secreting B cells in animals either infected or vaccinated, this is the infected data I showed this morning, but the vaccinated, we get the same thing. So we also use the needle-free system that Sani talked about in her talk to do intradermal. We get a little bit better response using the intradermal needle-free than we do with the intramuscular needle inoculation. The early response IgM rises very quickly, peaks at day 4, and is essentially gone by day 14. The longer lasting IgG1 response, this is in cattle, the dominant antibody isotype is IgG1. A little bit later response, but just as in the infection, all of the cells making antibody have left the circulation by 21 to 28 days post-vaccination. The more minor subtypes of antibody, IgA and IgG2, have a much lower response. In fact, we didn't detect any in the needle inoculated animals, but in the needle-free inoculated animals, we got a much lower response, and again, it essentially had disappeared by day 21. So in these animals vaccinated with the adeno system, we got the same data with the killed virus. Basically, after 28 days, all you have is antibody circulating in the serum. There are no B cells producing antibody left in the animal after day 28. My colleague, John Artz, who is the pathologist at Plum Island, was doing a series of experiments where he sacrificed the cattle and did an extensive analysis of pathology and viral persistence and such in those animals. So we were able to also look at lymph nodes, spleens, and bone marrow, and we got the exact same data as we did with the peripheral blood. The presence of antibody-secreting cells went on till about 21 to 28 days, and after that we could not detect any of these antibody-secreting cells, even in the lymphoid tissues. So there was no source of antibody, and thus you're living on the circulating antibody as your protective effector, and that wanes between 4 and 6 months after vaccination. So you have to boost every 6 months to keep your animals protected against infection, which is problematic for the farmers. So the plasma cells disappear, the memory cells never generate, and you're living on that antibody. So again, going back to the complexity of an immune response, basically you're getting a very short-lived CD4 helper cell response that's only pushing this short-term B cell expansion and plasma cell development. Because the CD4 Tregs probably kick in when the antigen is no longer driving things, you don't get that generation of memory B cells. You don't get generation of CD4 memory. You don't get generation of CD8s at all, even early development of CD8s. So we're living on this short-lived antibody response as the way this vaccine works, which is not satisfactory at this point, again, because you have to vaccinated every 6 months. So we took the approach of redesigning the vaccine to induce a T-cell response. And the reason we did this is because some years ago, after a lot of very hard work, Jared Patch in my lab was able to show that when you infect pigs with foot and mouth disease, you get a very nice cytotoxic T lymphocyte response at a point where I would absolutely predict you'd see it, which is 3 weeks after infection. And these cells very efficiently and specifically kill virus-infected cells. So if you test these cells against cells infected with vesicular stomatitis virus, they don't show killing. So it's virus-specific, and it's classic MHC restricted. So if you use the wrong MHC, so you mismatch your animals in the SLA type, so you have a different SLA in the target cell, they also don't kill. So these are classical cytotoxic T killer cells. So clearly the infection's inducing CTLs. So it would seem if you want to control this infection, you want to at least do the same thing and induce the cytotoxic T lymphocyte response with your vaccination strategy. So we're going to focus now on this part of the immune response, which is driving that CD8 response. And again, the surface, the antigen-specific receptor on B cells is antibody. The antigen-specific receptor on T cells is this more complicated T cell receptor that sees peptides derived from your pathogen, in this case our virus, and those peptides are bound to an MHC molecule and presented to that T cell receptor to activate the T cell in an antigen-specific manner. So Jared and I decided that one approach to this would be to screw up the processing of the polyprotein that encodes the capsid proteins and not allow the capsid to form, and simply by mutating or eliminating the 3C protease. And as I showed this morning, we clearly interfere with the processing of the polyprotein, the P1 precursor of the virus capsid proteins. Here's the virus processing into the components. Here's the Ad5 vaccines also processing into proteins. But by eliminating that 3C protease, we only have the precursor protein. And we predicted or hypothesized that having that polyprotein trapped in the cell would lead to those proteins being degraded into peptides and loaded into MHC molecules, and thereby stimulating a nice class 1 T cell response. And the whole idea here is that we would create complementary vaccines that we could use together. The B cell vaccine originally developed, and now this modified version of the B cell vaccine, which is stimulating a T cell response. So in fact, that's what happened. This is vaccinating animals, prime in the blue, boost in the red, with just the T cell vaccine, and with just the B cell vaccine. And what you can see is you get a very nice CTL response in the T cell vaccine. You don't see anything in the B cell vaccine until you do the boost vaccination. And it's not as good as the T cell vaccine. But again, in this idea of being complementary, because you want to optimize all aspects of the adaptive immune response. When you use the two together, you get a much better response in the priming and a noticeably better response when you do the boost. So this was our system from this point in, and what we wanted to do was analyze the specificity of the T cells that we were inducing with this new T cell vaccine. Using the tetramers I talked about this morning in the technology talk, so that we could take a situation like this where you have no idea what any of these cells in the peripheral blood of the animal are doing, even when you take them out and analyze them in secondary in vitro. But now using these tetramers, we can essentially target the cells that are specific for the virus and even target differential function within those cells that are specific for the virus. And again, this is how that technology works, that the peptide from the virus is bound by the MHC molecule in this binding groove. Our colleagues at University of Copenhagen have developed an algorithm for predicting what those peptides are. When you get your predicted peptides, you run actual biochemical analysis to show which of these peptides that the computer told you would bind are actually bound and become T-cell targets, and that allows you to develop the MHC tetramer technology to look at those T-cells. So in the original experiments, because the computer algorithm didn't have any porcine data in it, this was the first porcine data introduced into the computer algorithm. It originally predicted 10 peptides, of which 5 actually bound and which were tested in the first experiment. Each time we did an experiment, we were able to essentially educate the computer algorithm so it became better at the prediction. And as a result, the neural network learned from the new data and actually predicted more peptides, which we tested, and ended up in the last experiments, we had 16 different tetramers that we tested in on all of these pigs to look at fine specificity of the T-cell response. The other advance that we developed that allowed us to do the critical experiments I'm going to tell you about now was the graduate student that did this, Lasse Pedersen, came up with an excellent way to measure the negative staining controls. So if you don't use a tetramer at all, you get a lot of background staining. And if you use a tetramer with various other specificities, peptides that have a T-cell stimulatory capacity, we were getting a lot of background staining. So what Lasse decided to do was create this null tetramer. And basically what the null tetramer did for us was give us a peptide. What Lasse did was design a peptide that would bind the MHC molecule as well as a normal antigenic peptide. So for class 1, the binding positions in the amino acid peptide are usually position number 2 and position number 9. The swine class 1 looks exactly like the human class 1 in that aspect. And what Lasse did was he made a synthetic peptide that had the binding motifs at position 2 and position 9, but the rest of the peptide was all alanines and glycines. And as a result, those amino acids don't have any side chains. And as far as the T-cell receptor was concerned, it looked like a piece of ice. It was just flat and nothing to bind to. So we called that the null tetramer, and that was critical in getting data that we could actually analyze from these experiments, because what you can see here is if you put in no tetramer, you get very, very low binding. That's for a lot of reasons. One of the main ones being there's no fluorophore in there at all. There's no staining signal in there whatsoever. So if you use that as your negative control, everything's positive. Every tetramer you look at is positive. But if you use these null tetramers, which are in red, which are these peptides that have nothing for the T-cell receptor to see, but they bind to the MHC molecule so you can make your tetramer, what you see is now you have a very different result. You've eliminated all of these tetramers because their binding is no better statistically than the null tetramer, and you have these standout tetramers that are real binding. So now you're identifying your antigen specificity. And this is important because since T cells only see stretches of 8 or 9 amino acids, two of the residues of those peptides are binding the MHC molecule. The other residues are available for the T cell receptor to see. What the potential is, if you look at it mathematically, is you can have common peptide epitopes across many strains of FMD. So unlike the antibody situation where you have to match your strain perfectly to protect against being infected with that strain of the virus, here in the T cell situation, what you're going to find is you'll be able to identify a lot of cross-protective epitopes that are in a lot of different strains of the virus. So that has been the goal in FMD vaccine development for decades, and this is the first hint that we might be able to get there. So I'm going to show you the ultimate experiment because it had the most tetramers analyzed, but then I'll go back and show you all three experiments and how they have the same trend. So when we primed animals with the vaccine, and again we're using the T cell and B cell vaccines in combination here, we had one animal that only had a single specificity in its T cell response, second animal with that same specificity, and then we had two animals that had dual specificity for two different tetramers, so two different T cell epitopes were induced by that vaccination. When we boosted the first time, very interestingly, now all four animals have this dual specificity. So the process of boosting has brought these two animals that had only the single specificity up to the point of the other two, which has the dual specificity. When we boosted a second time, we actually got tremendous expansion in T cell epitopes that the T cells we were able to detect from these animals, with the exception of one animal. One animal stayed at that two different specificities, but the other three animals now had reactivity across four different tetramers. So you're seeing with each prime and boost an expansion of the T cell specificity. And this again is exactly what you want in a virus situation like FMD where you have such varied serotypes. You want to expand your coverage of the different serotypes. So this is the summary of all three experiments. In the first experiment, we only had a single specificity in all the animals after priming. And then in this experiment, actually, we didn't boost, we challenged with live virus. And we expanded that specificity in all animals in the cohort from a single specificity to a dual specificity. In the second experiment, interestingly, even though these are genetically identical pigs, we had a different specificity following the priming vaccination. And then again, the expansion upon boost, and if you waited long enough, you could see even another clone of T cells emerging with yet another specificity. So we had specificity for three different T cell epitopes in those animals. And then in this last experiment that I just showed you, we had a limited specificity after priming. That specificity expanded after challenge and expanded even further after the boost. So what you're seeing, again, is what is actually one of the holy grails for FMD vaccines, which is expansion of the specificity of the immune response so that you can cover more and more strains of the virus with this vaccine. So this is critical. This is the first time I think it's ever been shown, certainly in swine. And it matches some of the data coming out of human now. So if you look at, there's a lot of analysis of people that have been vaccinated with the influenza vaccine year in and year out and showing these similar things, that if you can refocus the response to your vaccine to the T cell compartment, you can show expansion of specificity versus seeing the opposite in the B cell compartment where your specificity narrows with each boost. So this is critical for vaccine performance. This is exactly what you want out of a prime boost regimen, and it shows improved vaccine performance by adding this T cell targeting portion of the vaccine. So that's our only example that we've been able to do so far. So what we're doing now is we're taking this approach with bovine herpes virus, obviously in cattle, bovine respiratory syncytial virus, and then this is exactly what Sonny Surdat and I want to do with the PRRS virus, is start redesigning these vaccines to target the T cell response and get broad specificity and coverage of the divergent strains of the virus. So the people who did all of this work, Lasse Pedersen is the graduate student who did all the tetramer work. Jared Patch is the postdoc that did develop the CTL assay and was able to carry that further. I didn't show you the details, but we started with a classic assay for cytotoxic T cell killing, which is measuring cell death in the target cells. And what Jared did was develop that technology I talked about this morning, where you can now do that whole assay by flow cytometry. And we applied it to African swine fever, and it works there as well. So it's a technology I think we're going to be able to take pretty far. Mary is the technician in the laboratory who developed the ELISpot assay, and using the tag virus as a probe in the ELISpot. So we can use it both in ELISpot and in the flow assays for B cells. And this is a group of crazy scientists that were involved in a grant from the National Science Foundation that was all about MHC and tetramers and T cell specificities in East Coast fever and foot and mouth disease. The critical person being Soren Buss, whose lab developed the MHC tetramer technology for human that we have now applied to cattle and swine, and Morten Nielsen, who is the bioinformaticist that developed all the algorithms that allow us to do that analysis. And with that, I'll take any questions. Thank you very much.
Thank you so much, Dr. Cody. I'm going to give a summary in Thai for 5 minutes, then we carry on to a Q&A session. Alright นะครับก็ดร. Cody นะครับพูดจริงๆแล้วงานดร. Cody เนี่ยเป็นฝันที่เป็นจริงของสวายอิมมูโนโลจิสทุกคนเลยนะครับคือที่ผ่านมาเนี่ยงานศึกษาภูมิคุ้มกันในสุกรเนี่ยในส่วนของ T cell นะครับเป็นสิ่งที่เป็นไปไม่ได้เลยเพราะว่าเราไม่มี tool ต่างๆเหล่านี้เลยแต่ทุกวันเนี่ยเห็นได้ชัดเลยว่าเราตามโลกของ T cell immunology ในคนทันแล้วเรามี tool มากพอที่จะศึกษาในระดับโคลนได้แล้วอย่างที่ดร. Cody พูดได้ถึงนะครับก็คือเนี่ยเป็นการวัด CTL response ในระดับ single clone ของ T cell นะครับที่เห็นว่าเป็น SLA 1134พวกเนี้ยมันคือการวัดด้วยเตตาเมอร์เนี่ยเป็นการดูที่ระดับโคลนเลยเพราะเตตาเมอร์จำเพาะต่อ T cell receptor ของโคลนนั้นเท่านั้นแล้วก็เห็นว่าเมื่อกระตุ้นด้วยวัคซีน FMD construct ที่เป็น human adenovirus type adenovirus 5เนี่ยเราเห็น breadth ที่กว้างขึ้นด้วยคือจากเดิมที่แค่พรามเนี่ยเราเจอแค่1โคลนใช่ไหมครับแต่พอครั้งที่2ที่เราบูสต์ปุ๊บเราเจอว่าโคลนมัน expand ขึ้นครั้งที่3โคลน expand ขึ้นอีกอันนี้คือ classic T cell response เลยกระตุ้นครั้งแรกเราไม่ค่อยเห็นหรอกครับเพราะว่า naive T cell เป็น T cell มีแค่ไม่กี่จำนวนใช่ไหมฮะแต่พอครั้งที่2ครั้งที่3 export บ่อยๆเนี่ย breadth มันกว้างขึ้นซึ่ง breadth relate breadth ความกว้างความหลากหลายของ T cell response ต่อไวรัสนึงเนี่ยลิงก์โดยตรงกับ protection เลยเห็นได้ใน HIV เช่นกันนะครับซึ่งอันเนี้ยจะทำให้เรา break barrier ของ FMD vaccine ได้อย่างที่ผ่านมาในวัคซีนไวรัส RNA ส่วนใหญ่ที่มี genetic variation กว้างเนี่ยปัญหาคือเราไม่สามารถกระตุ้นวัคซีนชนิดเดียวและครอบคลุมทุกsubtype หรือ serotype ได้ใช่ไหมครับแต่ด้วย T cell ที่เอมจำเพาะต่อเหมือน Achilles heel อะครับเพราะว่าไวรัสทุกตัวมันมี epitope มีโปรตีนบางโปรตีนที่ต้อง conserve เอาไว้เพื่อรักษาบางหน้าที่เอาไว้ถ้าเราสามารถกระตุ้น T cell response ต่อส่วนที่เป็นจุดอ่อนของไวรัสได้และ conserve อยู่ข้ามทุกๆ serotype ได้เราจะได้ T cell vaccine ที่เป็น universal T cell vaccine จริงๆซึ่งอันเนี้ยก็มีการผลิตวัคซีนชนิดนี้ในอินฟลูเอนซ่าแล้วโดยที่เขาเอมไปที่ transmembrane ไปในส่วนที่เป็นสต็อกของ HA ไม่ใช่ส่วนที่เป็น antigenic variation สูงๆอะไรเงี้ยนะครับก็ถ้าถามผมนะผมเป็น T cell immunology ผมตื่นเต้นมากเลยกับเทคโนโลยีเหล่านี้เพราะว่ามันทำให้เราลงไปดูในจุดเล็กๆของแต่ละเชื้อได้แล้วครับเราโฟกัสจุดอ่อนของไวรัสได้ในอนาคตนะครับ ก็ต้องขอบคุณดรโกดีมากๆเลย Thank you so much I'm quite exciting about all this fun stuff that we can do together now all right we have one question from the floor so why FMDV serotype O have a very poor antibody induction capacity with trivalent vaccine.
The trivalent vaccine.
Yeah, it's okay.
That I think that changes depending on what your combination is. So what happens is because there's more than one strain circulating, when you're trying to control FMD, you make a multivalent vaccine. And in Kenya, for instance, it's a quadrivalent vaccine. In a lot of places, it's a trivalent vaccine. I don't know anywhere that's not at least a divalent vaccine. So there are combinations of strains that seem to cause problems with the other strain in the multivalent vaccine. So there's been times in Kenya where the tetravalent vaccine is OA, SAT1, and SAT2, SAT standing for South African Territory. There's times when it works just fine. You get beautiful antibody responses to all 4 strains of virus. And there's certain combinations, they have to adjust the strains that are in the vaccine based on what's circulating. They come up with situations where there's combinations that there's interference. And it's not just O. Sometimes the A response is lost. Sometimes the SAT1 response is lost. It just depends on the specific strains that are involved and the relative relation. Well, the relationship, I guess, is what I'm trying to say, of the antigenicity of the different capsids from the different strains, which is another reason taking the T-cell approach is a better way to go, because those are going to be epitopes that are common across strains. So you don't have to worry about this interference that you get when you're dealing with the 3-dimensional structure of the capsid that the antibody is responding to.
ก็คำถามนะครับก็คือทำไม trivalent vaccine เนี่ยถึงกระตุ้นได้ไตเตอร์ต่อ O ที่ต่ำหนึ่งเลยคือเวลาเราเป็น trivalent vaccine เนี่ยคือเราต้องการจะเพิ่มความกว้างของการตอบสนองโดยการใส่เข้าไปหลายๆสเตรนถูกไหมฮะก็แปลว่าแอนติเจนเหล่านี้มันจะไปแข่งขันกันในการกระตุ้นภูมิคุ้มกันในตัวสุกรหรือในตัวโคอย่างเงี้ยครับซึ่งอันนี้ก็เป็นคลาสสิกเหมือนกันก็คือเหมือนอย่างเห็นในเดงกีวัคซีนก็เหมือนกันครับ type 1 2 3 4เนี่ยเจอ1 2เนี่ยดีตลอดกาลเลย3 4กระตุ้นไงก็ไม่ขึ้นเพราะว่าแต่ละเชื้อมันมี immunogenicity ที่ไม่เท่ากันนั้นเราเอาตัวที่มันเป็น immunogenicity แรงๆไปอยู่กับตัวที่ไม่ค่อยแรงเนี่ยตัวแรงก็กลบทั้งหมดเลยนะครับซึ่งดรโกดี้ก็เลยบอกว่าอันนี้เป็นส่วนที่ T-cell vaccine จะมาแก้ไขได้เพราะเราโฟกัสจุดที่เหมือนกันเราไม่ได้เพิ่มความกว้างด้วยการใส่หลายๆสเตรนแต่เราหาสิ่งที่หลายสเตรนมีเหมือนกันและโฟกัสอิมมูนไปที่ตรงนั้นนะครับมันก็จะช่วยตอบโจทย์ตรงนี้ได้นะครับ
Yeah, Bill, I'm just curious about the Ad5 vector, that it gives such a poor T-cell response. I mean, I thought that making replication deficient but still live virus vectors would have all the TLR8 activation. You would have, I mean, because that was supposed to be the advantage compared just to give the recombinant proteins. And yet you see a short-lived antibody and no T-cell response. Is that common for that vector? Or?
Yeah, absolutely. Well, because the antigen product that you're gonna stimulate anything with just the B-cell vaccine is the empty capsid. It's just like the killed virus. But in our case, we're not using adjuvant. So all of the pig work that Marvin and I did, whether Marvin looking at the B-cell responses or me looking at the T-cell responses, we never adjuvanted. So the difference was when we went to licensure in the cattle, the company that we partnered with wanted their vaccine in it, wanted their adjuvant in the vaccine formulation. It really didn't change the length of antibody response, at least coverage-wise, from the experiments we had done with the killed virus. You still had decent immunity at 6 months and completely lost at 9 months, and those are the only data points we have. complete protection, 21, 28 days, all the standard places that you might do your test challenges. But in these experiments in the swine, I never put adjuvant in anything. So the adjuvanting capacity of the adenovirus itself is minimal. Even though we're giving ridiculously large doses, the doses are 5 times 10 to the 9th adenoparticles for these vaccinations.
May I ask some question too? So do you see the difference in expression level between FMDB and FMDVT construct?
No, I showed you those gels. Those are
Same.
so the, you know, if you extrapolate the intensity of the P1 band and the intensity of the process bands, they're in the same,
Okay.
you know, within reasonable
So
level of each other.
โอเคคำถามของดรยังเกอร์เซนนะครับ I'm quite confused with two language I'm so sorry is that คือว่าทำไมadenovirus vector เนี่ยซึ่งจริงๆแล้วการทำวัคซีนด้วย adenovirus vector นะครับเราหวังจะให้มันเหมือนของจริงโดยที่ไม่ต้องใช้ไวรัสจริงๆใช่ไหมครับเพื่อไปตอบจุดอ่อนของ CUVC หรือ subunit vaccine ต่างๆแต่ทำไมก็ยังคงได้ T cell response ที่ค่อนข้างต่ำอยู่ดีหนึ่งคือทั้งดรโกดี้ก็บอกว่าหนึ่งคือเราไม่ได้ใช้ adjuvant ในกระท้องในสุกรก็เป็นไปได้ว่ามันไม่ได้ prime T cell response ได้แบบที่เราต้องการได้นะครับส่วนคำถามที่ผมถามก็คือว่าเป็นไปได้ไหมว่าพอเราเอา protease enzyme ใน construct ออกไปเนี่ยซึ่งเขาจะหวัง express เป็นโปรตีน P1 ยาวๆเนี่ยครับเพื่อกระตุ้น T cell response เนี่ยมันทำให้ expression level ดีขึ้นหรือเปล่าพอ antigen เยอะขึ้นก็เลย T cell response แรงขึ้นอะไรอย่างเงี้ยนะครับดรโกดี้ก็ตอบว่าไม่ต่างกันจะ B หรือ T construct ปริมาณโปรตีนที่ express เหมือนกันนะครับแสดงว่าการเอา protease enzyme ออกไปเนี่ยใน construct เนี่ยอาจจะช่วยให้เมื่อเป็นโปรตีนสายยาวอะทำให้มันโฟกัสไปที่ T cell response ได้มากกว่าครับโอเค
Just having kind of hypothetical questions. You say that because the B cell response is not sustained due to the lack of the efficient induction of the memory T cells, and we would probably understand that it's probably the helper T cells that is not really sustained well enough to be able to kick off the B cells memory. But then you move towards development of the cytotoxic T cells, and it seems like every time that the animal is exposed to the virus or the antigen, then you have a broader breadth of the CTL productions or CTL clones. Now, just come back to the antibody questions. That would it also happen the same way with the helper T cells? Would you see a more cross-reactive T helper cell response following these numbers of exposure?
I would predict that, but we have 0 data for class 2 T cell specificities in swine. So hypothetically, absolutely, but we got to do the experiment to.
Because
know.
I'm greedy, because I think that I like to have both antibody and CTL, and that's why I keep coming back to that T cell and B cell response.
Well, I think one of the things we can conclude from our T cell experiments is that the expansion of the T cell clones that have specificity A versus B versus C versus D there doesn't seem to be a loss of those expanded T cells. Now I haven't done the experiments like we did with the B cells of looking in the lymphoid tissue to see if they're hanging out. But my prediction is they are, because we keep drawing out with each boost more. So you take a very rare precursor, one that you don't see, there's too few to detect after you do the prime, but you have expanded it. It's just that you haven't expanded it enough that your technology can detect it. So when you do the boost, you now are starting from a higher point and expanding it further, and now you can detect it. So that says to me that those cells are not lost, but they're different cells. So T cells don't differentiate to something like a plasma cell that has a short half-life. So I think what happens in the B cell situation is most of those expanded B cell clones turn into plasma cells. Very few of them go back to quiescence and sit there in an expanded capacity the way the T cells do. What happens with the helper cells? I have no clue, but I'm guessing it's probably similar to the CTLs, that they go back to quiescence and they're hanging around waiting for the next exposure.
อันนี้จะเป็นคำถามที่ค่อนข้างซับซ้อนนิดหนึ่งนะครับก็คือผลเนี่ยโชว์แล้วว่า CTL to expand ได้ใช่ไหมครับแต่ว่าคำถามของอาจารย์สันติภาคือว่าแล้ว B cell memory ที่ไม่มีล่ะซึ่งการจะได้ B cell memory เนี่ยจำเป็นต้องมี CD4 T helper cell มาช่วยใช่ไหมครับก็เลยถามว่าแล้วเราเห็น CD4 T helper cell เนี่ย expansion เหมือนที่เห็นใน CTL ไหมคำตอบก็คือเรายังไม่มี SLA class 2 tetramer ที่จะไป detect CD4 T helper cell ได้เราก็เลยไม่รู้ว่าจริงๆแล้ว CD4 เนี่ย expand ด้วยไหมเหมือนที่เห็นใน CTL แต่ถ้าตอบแบบให้ hypothetically ครับเหมือนกับสันนิษฐานดูก็น่าจะ expand เหมือนกันใช่ไหมครับก็เลยคิดว่าแบบถ้าได้ CD4 T helper มันเนี่ยมันก็คงไปซ่อนอยู่ในลิมโนดรอที่จะ exposure ครั้งต่อไปแล้วก็ expand ออกมาในลักษณะเดียวกันกับที่เห็นใน CTL ได้เช่นกันครับ Any other questions Okay is that all Thank you so much Dr. Godi Impressive work
Thank you. Nice of you to staying around. I think Ik is doing an amazing job translating all this into Thai. I'm very impressed both with the questions and the talks. So this is going to be the last talk about a lot of T-cell work, and some of these things will be repeated that Bill has said, because I've been trying then to see what can we move on. After Lasse, our joint graduate student, moved on to a private company, we tried to see, can we actually make a tailored vaccine? Can we use this information we now have on T-cell immunology and make a vaccine that fits exactly the genetic makeup of that pig as a proof of principle to how that works? So the talk has a little bit again of repetition of what's happening with T-cells and viruses and MHC presentation, but my experience from talking about this to veterinarians in Denmark is that it's really nice to get it 3 times because it's so different from what you're working with today on everyday basis. So I will keep it anyway. So this is just very repetition about what happens with the infection. This is the level of a microorganism, and this is development of immunological memory. And something in here is reducing this microorganism and taking it out again. And we know that is the adaptive immune response. That's the one that protects us eventually. So when we want to make a vaccine, we're trying to mimic this so that we can induce an artificial activation of this immune response to get it down. And this is the standard thing that once you do it with response to one antigen, you have a response, and then when you give it again, you have a secondary response that's better and bigger. And that's, of course, what we're doing when we're giving the first vaccination and then boosting and trying to activate another thing. You cannot train an immune cell to react to any antigen. You can only expand whatever they're reacting to in the first place. And that's important when you are thinking about how this works. The problem is when we are working with immune responses, we have different types of infections we have to deal with, and the immune response needs to respond to that challenge. So, for instance, if you have the extracellular bacteria, Clostridia, Actinobacillus pleuropneumonia, you can have antibodies because they can neutralize the toxins, or they can attack the surface antigens. So an antibody response is sufficient. You don't need any particular T-cell response to deal with those infections. But for intracellular bacteria and viruses, you need the T-cell activation as well. And that is so difficult to obtain with the vaccines when you only have this simple antigen activation from the standard adjuvants like alum and also even with adenovirus. So the problem is, how can we get this combination of antibodies and T-cells into our vaccine development? And all this has to be mediated somehow into the immune system. So the normal way it does, this is a dendritic cell, an immune cell that is now in contact with whatever is attacking it. This looks like bacteria. So then they are taking it up, and depending on the profile of the TLR activation, the PAMPs that are associated with this infection, the immune response goes one way or the other. So this is a very, actually very simplified look of it, but you don't need to think about, just need to know that there are these dendritic cells. These are the orchestrators of the immune system, and depending on their activation, they can drive a CTL response towards viruses, a Th1 type against intracellular bacteria, or a Th2 and B cell response against the extracellular bacteria. So we need to sort of find out how this activation goes on in order to make vaccines that have the most efficacy with the least number of side effects. And that's what we've been trying to do. So going into the virus, this is what happens when you and I become infected with, for instance, in an influenza. The virus comes in, and the virus does not carry any reproductive system itself. It carries the genes, but it needs to use us, the pigs, the cattle, whatever, as the factory for reproduction of new viruses. So they simply unload their genetic material, and it moves into the nucleus, and then they start transcribing it just like it was any of our own proteins. And because viruses are so fantastically developed, that these proteins, once they have the appropriate proteinases, they self-assemble into the virus particles and then leave again, taking a full advantage of this whole production system. We have to produce all the proteins we're doing all the time, but now they're taking over and as a hostile enemy and producing these new virus particles. So that's really nice. The problem is, of course, the antibodies have nothing to attack. Our antibodies are circulating in our bloodstream, in our extracellular fluids, but they do not go into the cells. So when a virus is inside the cell, there's nothing to attack it. So antibodies can attack them outside. They can prevent them from going in. They may prevent them from going out, like for instance with the H and A and N antibodies in influenza. But they can't stop the replication, and that's where we need our T cell help. So this is again another simplification of what Bill has showed several times. This is then the virus proteins. It's being proteasome processed, and they're put inside the endoplasmatic reticulum into this MHC molecule. Actually, this happens all the time with all your proteins. So all our proteins, some of it is being sent out as a protein that is being used, but a little bit of it is being processed so that every cell in our body is constantly displaying small fragments, small peptides of what is actually going on inside that cell at the moment. So then our T cells can circulate and see, is there something we react to? And all the T cells that react to our endogenous peptides, the peptides we have in our own repertoire, they have already been eliminated in the thymus when we were infants or fosters. So therefore, we only have T cells that recognize foreign. So whenever a foreign protein is being processed, the peptide is different, and that's a basis for an attack for an immune system. So that's what we're trying to see. Can we then find out what are these peptides? Can we use those peptides in a vaccine? And then we may be able to drive exactly the type of immune response we want to peptides reflecting that particular infection. And this is then what happens. This is an antigen-presenting cell that is driving this CD8 T cell response that is a co-receptor for this antigen recognition. So these cytotoxic T cells that have then been expanded, they're then just circulating all the time and seeing, is this a friend or a foe? Is this something we have to react to, or should we just leave on? And whenever they find something that is foreign, then they start giving away their cytokines that will induce then a killing of this cell. Again, another example here, an infected cell. These are the virus, but now it is displaying small particles of the virus on the surface, and therefore the cytotoxic T cell can say, hey, we need to kill this, and they assemble around it and induce apoptosis of these cells. And how do we measure this? Bill just showed you nicely how we can actually do this with recombinant tetramers, because we need to find that same interaction between the T cell and the peptide in an assay outside of the body, and that's really difficult, but now something that is actually possible. So this is what's happening again, the same slide from before, and as Bill showed, this is the peptide sitting on the infected cell, and then the T cell receptor recognizes those parts of it that are sticking out and activates this cell to induce the killing. And if we believe this is true, we should be able to actually use this in our vaccine design. The problem is, of course, that we are all different, and so are pigs. So this is one pig, and this pig has an MHC class 1 molecule that recognizes only these peptides. So a very small fraction of the universe of peptides is recognized by this pig's MHC molecule, and also a very small fragment of the peptides in that particular virus. And another pig will have another MHC molecule that may recognize some of the same, but most likely will recognize others. And a third pig, of course, will then have a third MHC molecule, so that each pig will identify different parts of the MHC molecule, which makes us diverse and wonderfully resistant to variation in the virus, because we don't have the same attack points as many of the B-cell antibodies that are shared between all of us. So that is, of course, really nice, but also a big, big challenge, because you need to then tailor the vaccine to that individual genetic makeup that we're having. And we have, together with Søren Buus, now made recombinant MHC class 1 molecules for different production pigs. So these are the most common alleles we have in Denmark. Each cell, if it's homozygous, that means it's got the same MHC molecules from mother and father, will have both an SLA1, SLA2, and SLA3, but most of them will be heterozygous, so they will have 2 SLA1 molecules, 2 SLA2, and 2 SLA3 molecules that can potentially each identify a set of peptides. So we have, for production pigs, we are now also moving into more and more into using pigs as large animal models for human diseases, so both in different metabolic diseases and also a genetic cancer model developed with Larry Shook in Illinois. But nevertheless, this is then being used, and again, same picture as Bill used, because it's such a nice one, and none of us made it originally. These are the tetrameres. You can put those tetrameres into an into a streptavidin molecule, so that you have this assembly of a recombinant expression of peptides, and that will then emulate the interaction between the T cell, because this binding is actually relatively weak, and by corporate binding, having several of them in the same molecule, you can then actually simulate this interaction and get a signal when you're putting on a fluorochrome, and that gives you that signal. So antibody responses are strong and bind nicely. T-cell responses are much, much weaker, and we need to have that corporate binding in order to be able to use that with any reproduction in the laboratory. Okay, so following Lasse, we had another PhD student where we then tried to say, can we take advantage of this? Can we do this? And he made some fantastic animations in PowerPoint that is now, I hope, really instructive to understanding how he did this from the very early onset until the final identification of what happens. So we wanted to make a vaccine against PRRS, and Sani already told you all about PRRS, and you know it already, so I'm not going to say anything about PRRS. You know it's really important. That's enough. And then we can then immunize by specific peptides that match whatever makeup our pigs have. So the first thing that Simon did was that he looked at whatever is available. I think that is no, that's right. So the first one is to find what are the peptides available. So he looked at the whole PRRS genome, and he also surveyed the whole database for what's happening in terms of what are sequenced, and translated all those into the different open reading frames, and some of them are overlapping. So these are the different proteins that are available in the PRRS genome. Then he translated those into amino acids and found out what are the different possible strains then that we have available, and are they all representative of actual proteins? Are they starting with a start codon and stopping with a stop codon? And based on that, he found that there were 104 full genome sequences available from all the PRRS peptides, PRRS strains around. So there's about 200 extra, but they're all laboratory mutations that doesn't really fit. So the problem is, of course, if you want to use 400 full genome sequences, there are really a lot of peptides. So next thing is to do, what can we sort of find out what's happening? So what are the possible post-translational cleavage sites, and what would that be in terms of possible peptides? Well, that gives you 91,000 peptides. So if you want to synthesize 91,000 peptides and put it into a vector or anything, that's obviously ridiculous. So that's where the genetic makeup comes into play. So we used the most common SLA alleles in Denmark and said, okay, how do they bind peptides? And put this into a machinery that sort of say, okay, what if we match those with the different peptides? How are those binding scores actually happening? And we actually did is we used a combination of two. The first one is this positional scanning combinatorial peptide library that Bill also mentioned before, but I'll just walk you through how it actually works. So Søren Buus has already, when he made these different MHC class 1 molecules, he tested them with what's called a 9x random peptide library. That means for every of those 9 amino acids, he has a mixture of peptides that are random except for one amino acid. So this one is amino acid A in position 1, in position 2, position 3, and 4, and then test the binding for all these against our MHC molecules. And this is then interpreted. So for instance, you have a peptide sequence like this, LAQ. That means that L has a binding strength of 0.6, A has a binding strength of 0.8, Q has a binding strength of 0.2, and so on, and that gives you then a combined binding strength of how much would these peptides be expected to bind based on this random 9x peptide. And then you can see how will that fit into the the combination of all peptides, and in this case it's pretty bad. It's only in the top 32%, so that's not really good. But it's still, it's not very good binder. Then we've got the neural networks that Morten Nielsen developed, which is taking into account the amino acids surrounding the peptide binding groove of the MHC molecule. There are 28 of them, and emulating their peptide binding to the different peptides, he can then predict what would be the expected binding strength of this peptide into this peptide binding groove. And in this case, he ranks it as 0.4, so that's actually really good. So obviously, the two things don't always match up. And we then used a combination of those and said, okay, what is the harmonic mean? It's 0.8. Let's say this is a good. So when you have a computer, you can really do a lot. So we did this for the 91,000 peptides against each of the 5 different alleles and find out, okay, how are they going to do? There has to be in our top 2%, and that could be in our candidate. Now, we only have 6,000 peptides left. Of course, it's still quite a lot, but this is at least something to work with. Then Simon used this pop cover algorithm to say that we want to have peptides that are preserved and bind and has to bind to as many MHC molecules as possible. So this ranked all the 6,000 peptides, and then the number 1 rank binds to 4 different alleles, and it's present in 98% of the 104 strains. And then the next one, this algorithm says, okay, now we need to find another one that covers the one we lost, but is still as conserved. And then we got a next one, and based on that, it just ranks all 60,000. And then we can take the top ones and say that will cover whatever we have. We have a lot of conservation, which means that the peptides will work against all the different PERS strains. And we are covering the different SLA molecules, which means we have a chance to activate as many T cells as possible. And then we just took the top 53 peptides and say, okay, let's have something to do with that. So now we are working into an area that can be used, but it's still too many. And as Bill also said, and as you could see, the predictions don't always match up. So next thing is we test all of these 53 peptides against our recombinant MHC molecules. And you could do that by a simple measure, because if the peptide is bound to the MHC class 1 molecule, the beta-2 microglobulin is also bound. But as soon as the peptide falls out, the beta-2 microglobulin is also lost, and we have a secondary antibody recognizing the beta-2 microglobulin and not the peptide. So when the peptide falls off, the signal is lost, and then you can actually make a nice little titration and find out what is the actual binding strength of this one. We have another assay, both of those have been developed by Søren Buus and actually started out for humans, is measuring how long is this interaction taking place. That means what is the stability of our peptide to MHC molecule. Because as you can see, this is a stable peptide, this is a slow peptide, but they're actually having the exact same affinity to the one. So we need to have those that are binding for longer time, because if it drops off after 20 minutes, there's not much time to actually induce an immune response. And that means that we have here affinity versus stability, and we want to have those candidates that have both. So that is then now reducing our 53 peptides into the actual binders that stick for a little bit longer. So now we have 33 binding peptides. And now actually moving from 91,000 to 33 with just a little bit of laboratory analysis and of course a lot of computer work, but that's actually something that's doable and that we can work with. So now we have some candidates to a vaccine. And then we thought, what about if we put them together? Say if this one is matched to a MHC makeup, if we take just the peptides that the T cell should react to, put them with a little spacer in between, we would have a vaccine that only produces T cell responses that are maximized to attack the PRRS virus. The concept is nice. Let's see if it works. So to do that, we work together with IVI in Ghent. So they have developed what's called a replicon. So this is a classical swine fever-like virus. It looks like classical swine fever. It infects like classical swine fever, but they've taken away the structural protein, the eRNS structural protein, and therefore it cannot replicate. So it simply misses some of that it should respond to. And we have then, instead of that, put in our string of PRRS peptides. So this is how it looks. This is putting in here. This is the normal classical swine fever. You take away the eRNS, and then you put in a little bit of extra work into it so that you have this recombinant live virus that does not replicate. And this should then work in a way so that when you have this being translated, you have an N-Pro, then we put in ubiquitin as an extra spacer as well, because ubiquitin will then, when the N-Pro is off, will then lead the rest of the protein into the proteasome, and then you would have the peptides put into, with high efficacy, into the MHC class 1 molecule and put out onto the surface. So designed again to maximize the T cell expression and again instruct into a cytotoxic T cell response. And you can grow them in SK6 eRNS cells, so you have cells that are transfected with eRNS, so they are producing actually the missing part of the protein. So in that cell line, you can make new viruses, you can grow it up, but when they come into another cell line, it won't replicate. So that's really nice. And then you can infect that, and we look at those by flow cytometry to see how are the cells actually functioning. So this is a treatment control to see if it works. If you have no virus-like particle in here, that's of course shouldn't do anything. Then we've got the virus-like particle and put in a flag protein where you have an antibody to, and then you have the virus-like particle without this flag protein. And this is just to see how it works with a normal control, and then with a proteasome inhibitor, epixomoxin, which means that now the ubiquitin transmission won't work, to see if there's a difference. And then looking at that in flow cytometry, if nothing, you don't put anything in, nothing happens. If you don't have flag, you don't have an anti-flag response. If you inhibit the proteasome, then you have the flag response because now the protein is being produced. But if you are not using this inhibitor, the ubiquitin drives also the flag into the proteasome, and everything is being translated into peptides and put on display on the surface. So really nice, again, validation that this should work, so we get everything loaded onto MHC class 1 for a peptide for an activation. So of course the question is, does this work? Because it sounds so terrific. Well, let's see, we did an animal experiment, and of course, since we designed it for specific MHC molecules, we needed to test the pigs as well. So first we went into the herd, identified sows which had possible MHC combination. Once they'd given birth, we tested their litters, and then out of these 4 litters, we then selected a number of pigs that were expressing at least one, and most often 2 or even 3 of these. Actually, there were none with 3, at least 1 or 2 of these SLA molecules. And the SLA1041 is expressed in all of them. That's the same one that we used also in Bill's studies that you saw before. So we had, of those, 11 animals that received the vaccine, and then 7 animals that received a similar vaccine, but now just with a standard mouse, well-known T-cell epitope, the synfecal epitope that has nothing to do with PRRS. So we vaccinated pigs. We had them coming in as young piglets, vaccinated them actually 3 times. Then we did a challenge, and then culled them after 1, 2, 3, almost 4 weeks to see if there was any differences in the PRRS infectivity. They were given intradermal injections, 100 microliters of the virus-like particle behind the right ear. We didn't have any Marielle collaboration to help us with any guns. So we then challenged them with low pathogenic PRRS virus at 2 × 10⁶ of the tissue culture infectious dose 50. And what happens? Well, all pigs responded to the virus-like particle with an antibody response to classical swine fever. So the virus-like particle was active. This has nothing to do with what we put in there, so whether they had our peptides or the synfecal, they all responded to the E protein of the classical swine fever, because that's, that was part of that. So we know that worked. We had seroconversion to PRRS. There was nothing before we challenged, but there was PRRS replication in afterwards. So we didn't expect any seroconversion based on our peptides, but it would probably have been nice to see that there was maybe a lower PRRS seroconversion in those animals that had the vaccine, because in that case it could be an indicator of a lower replication. But we can't say that happened. That doesn't necessarily prove anything in terms of protection. Then we looked at interferon gamma ELISpot, so trying to use and only stimulating with the small peptides, so that should favor a cytotoxic interferon gamma activation. And basically we couldn't see anything. So the interferon gamma ELISpot in these peptides was really, really low and not much of an indicator. That's actually the reason why we gave it a 3rd challenge, because we had hoped to have this indicator of an actual immune response. But we can't really conclude it. Then post-challenge, what happens after the response? Sani showed that there is an interferon gamma response coming a little later, and we killed them already after 28 days, so maybe it's a little late for it, but still a little early killing. But we wanted to see if there was a boosting response of anything that we had vaccinated to. And what happens is that there is a little bit. So this is the difference between control animals and test animals. So control animals with the synfecal. So you can see that there is a higher number of peptide responses in the blood and compared to the non-vaccinated ones, but it's still relatively small. So a slight increase in interferon gamma cell-mediated immune responses to the peptide. And looking at lymph node cells, we also saw some indication, but again, the numbers were really, really small. But we did see something, in that case, only in our vaccinated pigs. And what about virus? Well, it's not as good as you would hope for sure. This is the virus load in serum. There's absolutely no difference either 5 or 13 days post-challenge. Looking into the lungs when we killed them, the cranial and the caudal lobes, there was not much of a difference, slightly lower in the vaccinated animals, but statistically 0.03, so borderline statistic. And also the same thing in the middle lobe, a little bit of a response, but absolutely nothing up here in the cranial lobe, and that is, you can't rely on having protection only part of the lungs. So we can say that there's, and when we then look at those animals that actually responded to the peptide, it gets a little better, because then you can see that some of those that had a nice peptide response were actually also those that had the lowest number of virus replications inside the lung. But it's still nothing compared to what we had hoped for. So conclusions is that, yeah, you can do something, but I think it's only a relatively few numbers of the pigs that actually worked well enough. And most of them is that we did not see the cell-mediated immune induction we had hoped for, and we did not see the level of protection that we had hoped for. So why? Yeah, we had, of course, only 33 peptides in the vaccine, so of course we had taken out 91,000 other candidates. So of course the selection of peptides could be wrong, but I don't know how to do it else otherwise. We know that the replicons worked. We know that the assays matched with what we wanted. So even though that vaccination worked in 4 of the test pigs, it's nothing more than the CTLA epitope has some potential. But I think that making 9 myosynthetic peptides into a completely artificial DNA setting with random spaces in between is too far away from what we have evolved to recognize. I think that we need to go back a little bit and say, okay, we can find stretches of maybe 30 or 40 peptide amino acids that include both T cell and Th1 and MHC class 1 and MHC class 2 epitopes, and then take those natural stretches and put into a vaccine. It would be much better than going all the way in like we did here. So certainly optimization is required. But the perspectives are really good. I mean, the concept that we can actually do it. I didn't show any tetramer stainings because the responses were too low. So there's no reason to do that. But so right now it's not nearly good enough, but it's small steps, it's baby steps moving forward into vaccine design, tailoring the vaccine, and knowing what can actually work, and see how you would be able to sort of generate a library of different peptides where you mix a vaccine that actually fits your population. And I've got 5 minutes, but that's not a problem. I'm almost done. So yeah, so I think the possibilities and the perspectives are really fantastic, but we certainly need a lot more in the execution. So with that, Simon Wellner, he did all the work, so I was just a supervisor together with Lars Larsen. And then, of course, we had wonderful collaboration again with Søren Buus, who delivered our peptides and did those developed the assays to measure both affinity and stability. Arthur Sommerfield and his lab in Institute of Virology in Switzerland made the replicons. Simon was staying there for some months. And then Morten, who is always coming up in these talks, he's now actually spending most of his time in Buenos Aires in Argentina, but he is maintaining all these neural network peptide predictors. So, with that, I'll take any questions.
Thank you so much. Very impressive work. Yeah, like a dream come true for all T-cell immunologists.
It's like a dream come true, but then you wake up and it's a nightmare. Yeah.
Yeah. โอเคครับผมสรุปให้ฟังสั้นๆนะครับก็คือเรื่องทั้งธีมของวันนี้จะเป็นแบบสไตล์เดียวกันหมดเลยคือเราต้องการจะหาวัคซีนที่กระตุ้น T cell response ให้ได้นะครับซึ่ง barrier หลักของ T cell response ก็คือ MHC restriction หรือในกรณีของสุกรคือ SLA นะครับซึ่งอันนี้ก็จะโฟกัสที่เฉพาะ CTL ก็เลยดูแค่ SLA class 1 นะครับก็อย่างที่เห็นว่าคือทีมของ professor Jungersen เนี่ยพยายามจะหาซีรีส์ของเพปไทด์ที่สามารถบายได้โดย SLA หลายๆโมเลกุลที่เป็น dominant SLA ใน population ของเขานะครับซึ่งเขาก็ใช้อัลกอริทึม predict มากมายปุ๊บได้เป็นซีรีส์ของ9 amino acid เพปไทด์ออกมาเขาก็เลยเอาเนี่ยโคลนเข้าไปใน classical swine fever ให้เป็นเวกเตอร์นำเข้าไป express โปรตีนนี้ในตัวสุกรนะครับแล้วก็เทสปริมาณการตอบสนองของ T cell response ต่อซีรีส์ของเพปไทด์นั้นด้วยการดู interferon gamma ELISpot ก็อย่างที่เห็นคือ response ไม่ค่อยสวยนะครับ protection ก็ไม่ค่อยดีอันนี้ professor Jungersen ก็เลยพูดให้ฟังว่าเป็นไปได้ว่าการ prediction อาจจะผิดหรือเราอาจจะเลือกเพปไทด์จาก 90,000 กว่าเส้นที่ predict มาได้เนี่ยอาจจะไม่ครบ ก็ได้หรืออีกอย่างหนึ่งก็คือเอพิโทปที่เราเลือกใช้เนี่ยความยาวแค่9อะมิโนแอซิดซึ่งในความเป็นจริงอ่ะมันอาจจะไม่ได้เป็นอย่างนั้นก็ได้เลนส์มันอยู่ที่ประมาณ7 ถึง 11 9อาจจะเล็กไปก็ได้อะไรเงี้ยนะครับนั้นก็เห็นได้ชัดว่าอย่างน้อยอันเนี้ย CSFV VPR เนี่ย work ได้ในการเป็น delivery system แต่จะเอาอะไร deliver เข้าไปเพื่อให้ได้ T cell response เนี่ยเป็นอีกโจทย์หนึ่งอาจจะต้องถอยกลับมา19แล้วดูว่าการตอบสนองผ่าน T cell ต่อ Pulse จริงๆแล้วเนี่ย target ที่สำคัญควรจะเป็นตรงไหนมากกว่าแล้วใช้เพปไทด์อย่างเดียวเนี่ยจะ work ไหมหรืออะไรเงี้ยนะครับโอเค question
Thank you very much. It's very interesting, and I can see how difficult it is to screen from 90,000 peptides to what you have left in the replicon. Now I have two questions. Is it where are those peptides on the protein? Is it any particular protein, a PRS virus, that those peptides are actually on that particular proteins, or it's just scattered, non-specifically?
Actually, I think that they were taken anywhere because he did the full translation of them all. We considered whether we should go into GP5 or M protein or anything, but I'll have to check up. Yeah. But he did the full translation of the whole virus genome, so I think that they could be anywhere.
Okay.
And I'm sure he did analysis of those eventually 33 peptides that we took out. Maybe that could be another target to go into that area. Yeah.
The other question is that, do you think it is possible that those replicon particle, which also been shown to be able to induce CTL in some situation, do you think that there's a competition between the induction of cytotoxic T cells against the replicons versus the Pers peptide?
It's possible because as you could see, they were all seroconverting to the E2 protein, so all the other proteins are also being put in. But our peptides were set in right after the ubiquitin, so they were supposed to be driven into that area. It is possible that could be improved as well. It's Nicholas and Arthur that developed the system, and I think that they just published other stuff where I think it worked better, but that wasn't the peptide design that we used.
ครับสำหรับคำถามแรกนะครับคำถามคือเอพิโทปเหล่านี้ที่เอามาดีไซน์เป็นโพลีเอพิโทปโปรตีนเนี่ยมาจากโปรตีนอะไรของเพิร์สนะครับเพราะว่าอย่างที่เราทราบกันคือแต่ละโปรตีนก็มีความเป็นอิมมูโนเจนิซิตี้ไม่เท่ากันก็คือดร. ยูงเกอร์เซนบอกว่ามันมาจากโฮลจีโนมดังนั้นก็ตอนนี้เขายังนึกไม่ออกว่ามาจากโปรตีนอะไรบ้างนะครับแต่ก็คือมาจากโปรตีนของเพิร์สนั่นแหละสำหรับคำถามที่2นะครับคือผมลืมครับแป๊บนึงครับเป็นไปได้ไหมว่าที่ T cell response ต่อ Pers peptide แล้วเนี่ยมันต่ำเพราะว่าเราใส่ replicon ของ CSFV เข้าไปทั้งตัวมันก็ compete กันใช่ไหมครับเพราะว่า resource ของ T cell ที่มีในร่างกายเนี่ยตอบสนองแค่ระดับหนึ่งถ้าเกิดเราใส่ replicon เข้าไปก็ตอบสนองต่อ replicon แทนเป็น bias ไปหา replicon ไม่ใช่ต่อ Pers ของเราก็เป็นไปได้เป็นอีกหนึ่งในประเด็นที่ต้อง explore ดูนะครับ Welcome back to the last session of this vaccinology symposium, in which Professor Jungersen will tell us about how to use vaccination to fight against antimicrobial resistance. Quite interesting topic to sum up the whole day of symposium. Please welcome Dr. Jungersen.
Thank you for hanging around. So, vaccination to fight antimicrobial resistance. Sani said that would be a hot topic, but we're fighting antimicrobial resistance in the next room as well. So, and it's been a long day, so it's quite clear that people are getting tired. I will see if I can spend all 45 minutes. We'll have to see. So, first of all, antibiotic resistance is ancient. Long before we had antibiotics, the genes that are actually mediating antibiotic resistance were there. So these are studies from an isolated, I think 4 million years isolated cave in New Mexico. They looked at the bacteria at the bottom of the cave, and some of them had antimicrobial resistance genes present. Also in permafrost in Siberia, there are some of these antibiotic resistance genes. And this paper in Nature, I think, also said from Yukon, Canada, that antibiotic resistance is ancient. This is from before the mammoth. So obviously, it's not humans that invented antimicrobial resistance. That's something that bacteria apparently have, for one reason or other, something that they can actually deal with. But that's also maybe related to many of the antibiotics we have are actually something that we have learned from nature. Of course, penicillin, the obvious example, it is from fungus, and therefore bacteria and fungi have seen each other long before we started using them as antibiotics. But also, antibiotic resistance correlates with use. These are human data, and the number or percentage of penicillin-resistant Streptococci pneumoniae, and the total antibiotic use in daily doses per 1,000 population. And it's quite clear that countries like Spain, France, also Taiwan, apparently, where Portugal, where they really like to go to the pharmacy and get medications, the antibiotic resistance is really high, while other countries in Denmark, we're very restricted in, and the doctors. Okay. This one's better, yeah. Other countries, apparently, the penicillin resistance is really, really low, and Danish doctors have always been very restricted. If they think it's a virus infection, you won't get antibiotics. They'll say, go home and go to bed and come back in a week. And fortunately, that is really nice in terms of not inducing too much antibiotic resistance. But this is human data. What about the animal world? Well, 70% of all the antibiotics are being used in farm production in the animals. So obviously, that is really a lot. And it also changes quite a lot by country. So Norway and Iceland are up here in the top. It's from this review antimicrobial resistance that you can find. But that's also because Norway have one of the greatest success stories in reducing antibiotic in their aquaculture. So they are having a lot of their agricultural production in the water, in their salmon farming. And they used to spend a lot of antibiotics, but they were able actually with vaccines to reduce that tremendously. So now they are producing, I think, something like 40,000 tons of fish a year, and they are spending 400 kilos of antibiotics, which is absolutely amazing in terms of numbers. But we can't do that the way we are farming animals on land. So I don't know where you fit in here, but it's quite obvious that some of these countries have a possibility to reduce this significantly because, again, Denmark is my country, but also England and Australia, they have a nice weather, but the Netherlands are here. I mean, there's a long way to come down for some of those that are simply just overusing, in my opinion. So where do we use it? I was happy to hear yesterday that Thailand in 2011 also banned the use of antimicrobial growth promoters, because even though when I was in vet school and we heard about antibiotics, the professor in pharmacy said there's absolutely no chance of developing antibiotic resistance with using growth promoters because the concentration is so low. And he was obviously wrong, and we are all wrong once in a while, and particularly when you're an immunologist, you're wrong really a lot. But still, I think that is now proven over and over again that we don't want that. So where you use the least antibiotics is when you're only treating the absolutely sick animals. And probably where you get the most value for money is actually if you use the metaphylaxis approach, where you are saying that now we have an outbreak, we have animals that are at severe risk of disease, we don't wait until they get sick. We now treat the farm at this particular incidence. So I don't like it when you're treating all your herd when they become 10 weeks of age, because now you know that Lawsonia is probably going to come within the next 4 weeks. But I want you to keep an eye on the animals and make sure that you're ready to treat them at 9 weeks or 11 weeks, whenever it fits the most appropriately, because otherwise you need to treat them too long anyway. So that's how we do it today, and we cannot live without antibiotics in modern world, and it's going to be a huge problem for animal welfare, and also for, of course, the human health, if antimicrobial resistance continues to grow. So this is well recognized. OIE has a strategy on antimicrobial resistance involving awareness and understanding, strengthening the knowledge through surveillance and research, encourage implementation of international standards, and of course also support good governance and capacity building, and that's exactly where we come in as veterinarians, that we have the possibility to improve animal health and welfare, but also putting some regulations, some restrictions. It's not too bad if if you're told to stop and think and actually use less antimicrobials if possible. Oh. So what about vaccines? Is that the way to do it? And that's of course the topic here. Can we use vaccines to prevent antimicrobial resistance? So can they counterindict? This is a sketch of the molecular mechanisms of antimicrobial resistance. So the different genes that are, it's always encoded in the genes of course, but the different mechanisms that are there. For instance, you have some genes that are breaking down the antibiotics, so proteases or other stuff. You may have something that can alter the enzyme here so the antibiotic doesn't function anymore, doesn't necessarily break it down, or you may have increased transport out of the cell again, either so it doesn't come in or simply taking it out. These are most likely mechanisms of antibiotic resistance. So can you vaccinate against that? In my opinion, no. We can't relate any of these mechanisms to the pathogenicity of the bacterial. There's not. If we put in a vaccine that drives antibodies against an internal enzyme in the bacterium, it doesn't really matter. If we have an antibiotic against, again, another thing that changes inside the microbial, this antibiotic won't work, and it won't help in terms of protection against disease. So it's much better to focus on actually making a prevention of the infection than thinking you can change the biology of the bacterium by giving a vaccine that will make it more susceptible to antibiotics. I don't think that's reasonable. I don't think that should be used. But I think vaccine has a great role because you can prevent the infection, and by then you can reduce the use of antimicrobial resistance. And we think that is probably also the way then to reduce antimicrobial resistance. Can they? And there are actually some conflicting evidence that I would like to put in there as well. So I talked about PCV2 earlier today, and that's pretty well documented that introduction of the PCV2 vaccine both reduced the use of antibiotics, but it also changed particularly use of antimicrobials. So stopping the growth promoters has really reduced the genes or the frequency of antimicrobial resistance bacteria, at least in Europe, where it's been studied a lot. And we can certainly also decrease the use of bacterium or antimicrobials by PCV2 vaccination. But this is a couple of recent papers coming out of Denmark, where they compared antimicrobial consumption in weaning pigs and Danish sow herds with different vaccine purchases during 2013. And what they found was that the sow herds that applied, in this case mycobacteria and PCV2 vaccines, had more antibiotics than those that did not vaccinate against mycobacteria and PCV2. And this one was a more detailed. It was over an 8-year study period where they did more into details and looked into it. And their conclusion was that the thing that really mattered was the baseline average daily use of antibiotics. So the higher the baseline use of antibiotic when the study started, the greater risk of having a reduction in antimicrobials by the end of the study compared to anything. So does that mean that vaccines do not prevent infection? Well, let's look a little bit more on the data. So this is antimicrobials with gastrointestinal indication and looking at the herds that were vaccinated against different things. So the non-vaccinating herds, and then the different herds. And they say, well, there's no effect. You cannot reduce the use of antimicrobials because you're giving Lawsonia. Well, you don't have the higher now, so that's what it looks like. But there are actually some here where they have PCV2 and Lawsonia, which are still high. And if you combine all vaccines relative to no vaccines, there's hardly any change. But I think you have to ask yourself, why are they not vaccinating? So I would say that these herds are not vaccinated because they don't need to. And if they don't need to, then of course they don't need to use a lot of antimicrobials. And the level of use here is, for many countries, extremely low. So therefore you need to be careful how to interpret studies like this. Is the same study, but now looking at antimicrobials with respiratory indication. And again, the use of those antibiotics compared to other ones. I mean, those that vaccinate with PCV2 and mycobacteria, yeah, they still have a higher production, a higher use of antimicrobials. So is that because vaccines don't work? I don't think so, but maybe there's a little bit here. But what they saw was that if you have a high use of antimicrobials, you'll probably go down. So it's all regression towards the mean. If you're having a low use of antimicrobials, you're probably going to go up within the next 5 years because disease changes. And sometimes you're in a good period and sometimes you're in a bad period. Maybe it all comes down to feed or something else like that. So you can't say that vaccines don't work, but you can say that when you vaccinate, it's because you need it. So if you're taking that approach, then you may want to look at, okay, when is it that our vaccines work the best? And most of the vaccines are those where you have a single agent infections. These are diseases where there's one infection and it causes one particular disease. And they most often work quite nicely. The vaccines against Lawsonia, Actinobacillus, and PRRS are probably not perfect, but there's still an effect for sure. And they can many times certainly be worthwhile using them. When you're vaccinated against multifactorial diseases, it's much more difficult. And that goes the same with us. We are vaccinating humans against influenza because we know influenza works, but we cannot vaccinate against common cold because there's so many things that give us common cold. And we would like to be vaccinated against diarrhea or other stuff, but there's too many things that are playing into hand. So the vaccines are working the best when they are single, and maybe, like the PCV2 example, you actually can get really good value also from vaccinating in multifactorial diseases. And certainly the PRDC combination with Mycoplasma and AP and so on, if you're having all together, you have a chance of making something good, but you need to do it right. So one of the reasons why I think that the Danish antimicrobial use is so low and why this effect was difficult to study in Denmark is probably because we have this SPF system. So I talked before about how we are keeping many infections out, but also at the herd level, actually the level of hygiene is really high, and there's a lot of effort being done. So this was developed all the way back in 1969 with cesareans, so starting out with notobiotic pigs and putting into new farms. And nowadays we have 70% of all Danish sows and 98% of all the breeding sows are in this specific pathogen-free system where they are controlling for Mycoplasma, Actinobacillus, atrophic rhinitis, dysentery, mange, lice, and now also PRRS, where we are actually giving the different herd status, whether they're infected with the type 1 or the type 2. So all this is monitored. If you have suddenly an outbreak of Mycoplasma, then you're changing status, so you're still SPF, but then you're SPF MIC. So that means that you will be able to sell to others that have the same disease status. So everything is really monitored, and it's the responsibility of the farmer to send in samples every year to document that they are still free of all the serotypes of AP, that they're free of Mycoplasma and so on, in order to be able to trade with others at the same level of health control. So that's of course really nice to do that. Supplementary, they also have to indicate the Salmonella status, so we have almost eradicated Salmonella in chickens and it's still present in pigs, but at least we are keeping it under control and slaughtering pigs with a high salmonella status. At the end of the day, at the slaughterhouses, everything to try to keep it out of the food chain as much as possible. So all this is very, very well organized, and there's the SPF Denmark system is working it. So this is based on, of course, protection against infection, so trying to keep things out, so they are having a strict biosecurity, but also knowing what you're buying. If you're buying from somebody that tells you his status is good, it's not as good as buying from somebody that can show you the papers that is actually tested free of these things. So also health inspection, so that everything works, and then this declaration of health status that can be used. And this is implemented everywhere into trade, so you can go back and find who was buying where and when, and everything is documented and reported into central place. So that, I think, has been reducing use of antimicrobials in Denmark much more than you can do with any specific vaccine intervention or other things, but generally having a high level of hygiene. You cannot go into a Danish pig herd without taking a shower and changing clothes many places. It's not because they're afraid of you, it's because they're afraid of the pigs, that you will bring something into them. And that's, of course, something they work with. But does that mean that we should discourage vaccines? Absolutely not. Vaccines are terrific and one of the greatest ways of reducing infections in the world, both for humans, but certainly also for pigs. But if you want to reduce the use of antimicrobials by vaccines, they are not perfect. As I said this morning, no vaccines are perfect. They're always imperfect, and if you challenge them too much, you're just shooting yourself in the foot. If you want to be a veterinarian that is improving the health of your customers, you need to help them make the vaccines work. So if the hygiene is poor, the farmer will complain that they don't work, or maybe he's not being instructed enough to do it appropriately. So actually the rest of the talk, I'll give you a few examples of how this can be done, what the problems can be, and just simply as a reminder that there's a little bit of technicalities in doing it right. It's not just taking the vaccine out of your pocket and then this is solve everything. You just need to do the whole procedure the way it should be done in order to make it work. So these slides were produced together with a friend of mine called Preben Mortensen. He's now working in SIVA, but has been very active in pig consultancy as a veterinarian, and also worked with Merial before. So he has outlined some of those things in these slides that I've also then amended somewhat. So vaccines, of course, they are simulating the host's own immune system. We all know that. We're trying to do that as much as we can. But that means that we are, as Bill also mentioned, there's a time limit to it. A great vaccine, you need to give it once, and then it's for the rest of the life. But that's so rare, and it probably also, then you need to have an infection that is only dangerous at a certain point of life. So if you want to use a vaccine, make sure that you actually follow the recommendations, use the correct dose. In Denmark, lots of farmers think that they can save money by using only a half of the dose or a quarter of the dose. But if an imperfect vaccine is not good, it may work in some periods, but the second the hygiene status is lost, then you may risk losing everything, and that's really not good. So use the appropriate dose, and also do it right. And then you have to think about the quality of the immune system. If you want to boost the immune system, treat it with respect, right? I mean, at the time of vaccination, but also at the time of infection, what is actually going on? How does that fit in? And then, of course, the infectious pressure. I talked about herd health and herd immunity, and that's you will protect even the vaccinated pigs by vaccinating appropriately, because immunity can be broken in many cases. So first of all, handling the vaccines. You just need to turn it around and make sure. So you don't want to shake it too much, because you get foam and then you'll get air, and then suddenly you can't see what you're vaccinating. I guess you're also using multi-dose bottles in here. So you want all the vaccinated pigs to get the right amount. Use, of course, a clean needle and the syringe. when you have to mix several ones, do it right. If you want to mix different vaccines, they have to be made to be mixed. I mean, that's right, you can mix some of them, but that means that they are in the same formulation, they're using the same adjuvant, and they've been tested to do that. You can't just mix from two different companies just because it's easier. So stuff like that, and of course then keep it right with refrigeration. But you don't want to have it refrigerated cold when you give it in. You actually want to have it. That's not a problem here, I guess. In Thailand, they'll pretty quickly get it into body temperature. Then when you're vaccinated, this is a schedule of the skin. So you have the epidermis, then you have the cutis, the subcutis, and then down into the muscular. Particularly in pigs, the fat layer is so thick that if you vaccinate in the fat, you will just not get the appropriate cell recruitment that is needed. So almost all of them are intended for intramuscular use. That means you need to get intramuscularly. So that means that you need to pick the needles that fit the appropriate size of animal. So if it's a breeding animal, you need to have a syringe that's close to 4 centimeters long, because there's no reason not to do it. And suckling piglets, of course, the smaller ones. So make use of what is actually being done appropriately. And then, of course, you should, if you have a 4 centimeter needle, you need to put it in before you vaccinate. This is the thickness of fat here, so you need to get pretty far in until you get there. Otherwise, you simply won't get the effect of the vaccine. And then, of course, the injection technique. Yeah, use clean and sharp needles. When you're in vet school, you probably start using ethanol, and whenever you're doing anything with a needle in the skin, and the medical doctors do that. We would never do that in a Danish farm. I don't think you would either. And a farmer vaccinating thousands of pigs, probably not either. But I don't think that's necessary, as long as you certainly keep it clean and dry, and change it whenever you have a suspicion that something went wrong. And then put it appropriately. So if you're vaccinating several vaccines at a time, you can do that. I don't think that's a problem. But you shouldn't mix them in the bottle, and you shouldn't mix them in the musculature. So that means you need to put them separately, so that you will stimulate different cells with the different adjuvants and the different types. That shouldn't be too difficult. So what is this? This is in terms of intramuscular injection technique, so a multi-dose injection. This is not exactly perpendicular to it. It's so what can you do? Well, look at the farm here. It's a little bit of a problem when you are suddenly having where you can't get access into the pig. So make sure that this is the best you can do, but it would be much better if you actually had the room to operate, so you can do it appropriately. So if possible, do it perpendicular as here, and then behind the ear, where there's the least level of fat. So in this case, I mean, that's not really good, is it? The way that you have to go in and lean in, and actually this now becomes an angle of maybe 30 degrees or something, and that is bound to be deposited into the fat. So therefore you won't get an effect of the vaccine, and therefore this is a sow, so it won't transmit the antibodies to the offspring, and it's just a waste. Do it right. And the same thing here. Actually, here he could do it, but he's still not having the appropriate angle, and there's too much of a risk that he's not reaching into the muscular. So when you're using the vaccines, of course, you need to have an idea how to do it. So the approaches in modern pig production is that you can produce against reproductive losses, you can protect against damage to the gilt sour boar, you can use it to induce a colostral protection to the young ones, or you can use an active protection of the young ones. So the differences are obvious, and you need to, of course, to use your veterinary education to do it the right way. So if you want to have protection of your offspring, that means you need to have antibodies in colostrum. And if you want to have antibodies in colostrum, the colostrum is being formed at least the last 4 days before delivery. and it takes 1 to 2 weeks to get the level of antibodies you want into the pig. So that means if you vaccinate closer than 2 weeks before farrowing, actually the antibodies won't reach the piglets and it's too late, right? And to you, 2 weeks is probably 2 weeks. That's me for a scientist, but for a farmer it's something like maybe 8 or 20 days, right? I mean, 2 weeks it's something like that. So to be sure, probably it's a good idea to say 3 weeks before farrowing. The antibodies won't disappear, and you actually have a chance that the farmer will do it right, even though he has a different perception of time than you have. So that's just a matter of communication and making sure that he understands what's happening, but maybe he doesn't need to understand if you just give a little bit of a buffer in your recommendations. So the problem with vaccinating 2 weeks is also that the sow starts downregulating. There's a cortisol induction with close to farrowing. There's a downregulation of MHC. There's a different things happening at the prior to farrowing that's actually a little bit negative to the immune response. So make sure that you do it at the right time to get the maximum effect of your vaccine. So this is an example of a swine breeding stock circle. So the first year, the young ones, non-mated gilts, are coming in here at 6 months. Then they are being mated around 7.5 to 8 months. Then they have their 160 days pregnancy. They're moved into the farrowing unit, and then they are lactating. And you have the sows after this lactation, they then move back in for weaning, 5 days for mating, and then the pregnancy and the lactation. So how would you fit a vaccination program into this? And that was actually back to your question from before. What would you vaccinate against in Denmark? So enterotoxigenic E. coli, you want that to work for the piglets, so therefore that is a prime and boost of the gilts, so that you give the last one 3 weeks before expected farrowing. And you need to have a 3 to 5 week period in between. So that is where you do it. And you would then give a booster for the sows 3 weeks before the second lactation. And then that's what you need to do. For PPV and erysipelotrix, you actually want to have the non-mated gilts up here because this protection of these animals are not necessarily something you need to have for those ones. So you need to have your prime and your boost before mating so that they are robust when they come into that situation. And probably, I don't know if you are, you're giving a booster also in this place, I imagine, but I'm not sure. But you would, give boosters just before farrowing, so you have that transmission of passive immunity to the piglets as well. So you have both the protection of the gilts and the piglets. PCV2, exact same situation. You want to protect the non-mated gilts so they have a good pregnancy, but you also want to protect the post-weaners to give passive immunity so they will survive. And in Denmark, we also vaccinate against influenza, if you want to. As you could see, many farms are not vaccinating at all against these things, but quite commonly they are. And they would also then give these three vaccinations in the development and then a booster prior to lactation. So that would be a typical vaccination scheme for in Denmark in a pig herd. And then you treat the young boars as the non-mated gilts, so they would get this vaccination, and then revaccinate them every 6 months against PPV, PCV2, and influenza. So if you want to protect against embryonic losses or reproductive losses, so the stillborn mummified embryonic death and infertility, the SMEDI, caused by parvovirus or circovirus, or high fever and later abortion or infertility with influenza or erysipelotrix. There's also vaccination against Glazer's disease, but it was all outlined how you would put it in there in the previous vaccination schedule. So what you do is, of course, give your first vaccination 2 vaccinations 2 to 5 weeks apart. You don't want it to wait too long because you want to have that immunoinduction. And you have to remember what's happening. As I said, you cannot train an immune cell to recognize anything you vaccinate, but you can boost those that actually recognize it. So all our immune system is already educated so that you have about 10 to the 8th different T cell specificities circulating, I think. No, that's 10 to the B cell specificities, and it's higher for the T cells, but because others have been deleted. But that is 100 million different possible antibodies that your immune system can produce, or even more. So, and what you do when you vaccinate is you find those few ones that recognize it and make them divide so they become a larger portion of your immune system. And once they started expanding, you want to hit them again with a second vaccine so that they become an even larger portion and they drive in towards memory. And if you wait too long, they will collapse again and you will not get the effect of that booster. So it's this understanding of the basic immunology has an importance in terms of both the timing, and then once you've done it, it will disappear, but you can give a booster again within 1, 2, or probably 3 years and still get the booster effect, because now you have expanded that population originally. I believe there's no problem giving several vaccines at the same day, but if you want to give several vaccines, you shouldn't do it over several days, because then you might challenge your immune system. The breadth of the specificity is not a problem, but you may have some immunosuppression, particularly with live vaccines, following your vaccination. So then you need to wait until it's fully capacity again. But you can certainly vaccinate several things at the same time. So joint diseases, erysipelotrix or Glaser, you may have some general immune competence in PCV2 and influenza. Again, same thing as before. Give the primary vaccinations, and then typically they're moving from one herd to another, and if you want to control it, boost them when they're coming in again, so you have your protection running in your herd. And then usually give a booster every 6 months. These are inactivated vaccines, so don't expect the memory to be too long. It's typically much, much better with a live vaccine than what it is with a killed vaccine. If you have a new vaccine you haven't worked with before, the first thing you look at is what it's against. The second thing you look at is it live or killed, because the dynamics of live and killed vaccines are so different, and also the expectancy of what you can get is so different with live and killed vaccines. So the piglets we are vaccinating against E. coli, erysipelotrix, also influenza and PMVD, PCV2. And again, this is then typically done through the colostrum, and then you need to vaccinate before farrowing, as I said, 3 weeks before, to get it in there. Then, when you want to vaccinate the piglets, what about maternal immunity? Because, as you know, the piglets, they need to have the colostrum to get their antibody responses because of the multi-layered placenta that pigs have, just like cows, which is so different from us humans, where you don't need to have the colostrum to get an antibody response. So you should, to get that colostrum supply, you need to have, of course, the sows to be in good condition, because otherwise they won't produce the antibodies. And then they should stick around with their mother. In Denmark, they're doing a lot of moving piglets from one sow to another to even out. I don't know how many, but if you're using Danish breeding stock, you probably also get sometimes 18 piglets from a sow, which is of course too many that she can handle to give them appropriate antibodies. There's some talk about lymphocytes also crossing the young intestine, and I still don't know what to make of it. There are some pretty convincing papers, but whenever we tried, we couldn't make it work. But if there's something to it, and as I said, immunologists are often wrong, then for sure you need to think about this, that it has to match the mother. So therefore, if those lymphocytes have to traffic and help instructing a cell-mediated immune response, then you need to have them living with the mother for at least the first 24 hours. So then you can start evening out But I don't know. It's you might also have a look at which animals have already had a good feed of colostrum and then take them to another sow. It's not really scientific proven, but it may be a good idea to just be on the safe side. Then there's a possibility of having such a, if you're having a new outbreak of something, you want to have flushing of the system. So now you're suddenly giving a mass vaccination. So that's where you're suddenly into damage control. You have an outbreak of something new. You want to do something. You want to give, irrespective of whatever program you had, you just want to give a basic vaccination to everybody. And that can certainly be a possibility once in a while. But again, of course, be aware for the females less than 14 days before farrowing, because not only will it not help the colostrum effect, but you may induce abortions in some cases if you are unlucky, and you really don't want that. So then give first all breeding stock, and then with a 3 times interval, and then again give a booster, and then start using this normal procedure that you have established, that the way you want to do it. You can also have a single mass vaccination. That's for the piglets. So that means that all the young stock will be vaccinated very early, and then introduce that from the new one. So that would begin. You don't want to risk vaccinating sick animals if possible. So be aware that you vaccinate them early, and then not those that are actually sick. For once, you might severe their chances of survival, but the vaccine won't work either if they're sick already. Just like you wouldn't take your child to the doctor if it's sick and has fever, you'd better come back 2 weeks later and then get the vaccine. So there you need to use your veterinary education and give the appropriate advice in this emergency situation, and find out what seems to work the best. It might have different things in Thailand with the weather and what you can do and what you can't do. But you will for sure make some notes and find out what would be the way that you would supervise in the future. But this would be a way to do it in Denmark. So that was actually what I wanted to say. Vaccines against antimicrobial resistance, yes, because you can reduce the use of antimicrobials if you do it right. But if you don't do it right, vaccines won't work either, whatever you're doing. And no vaccines are perfect. You need to have something some assistance in order to get what you want. So, how much can you use vaccines for reducing antimicrobial use? I was actually part of this committee for EMA and EFSA to find out what are the possible measures that you can do. And there are a number of things that you can do to use antimicrobials. It's a 250-page report, but it's accessible on the internet, and you can use it if you want to. But there are many things that are important. First of all, you need to have a national strategy, and that was something that was also told yesterday. I don't think that you can put it up to the individual veterinarians to reduce antimicrobial resistance. You need to have a strategy, and you need to have some kind of monitoring system to see what's going on. Otherwise, it's very, very difficult to keep track of what you're doing. So that has to be centralized. Somebody has to make some decisions and put some money into it, and then say, okay, what are we going to do, and where are we aiming? So Thailand now has some reductions. They wanted to reduce by 30%, I think. So I think they are doing the right things. But unless you're monitoring, if you don't know where you're standing, it's really difficult to find out where you're going. So that's, of course, the first part. Then what we have done in Denmark is that we've made this is called a yellow card, similar in football. So you get a warning if you're a veterinarian that is prescribing more antibiotics to your farmers than other veterinarians. So that means eventually you might lose your authorization if you are a veterinarian that is using too much antibiotics. So that puts it into the mind of every veterinarian that this is actually a common task. We need to do something about it, and we need to use antibiotics prudently, think about it, and don't just distribute it whenever you want to. So that's also a part of the national thing to put that into place. You can't expect that from the veterinarian. The veterinarians will do what the farmers tell them to do if he can't argue against it. And if the farmer says, I want antibiotics now, you need to say, well, I don't think it's necessary, and I don't want to do it because I might lose my job if I'm giving you antibiotics without any proper prescription. So training, education, also of the veterinarians, is really important so that you have that level of confidence to say that this is not what you want to do now. You need to improve your hygiene. We need to go into the herd level and actually work something on that. So then you need to find out, can I then test what is happening? So can I rely on some diagnostics to tell me what is actually going to do? And again, that's not your possibility, your distribution. There must be some national laboratories where you can send in the samples and find out what is actually going on here. How can I treat that the best? What is the proper antimicrobial use in this case? And then use those diagnostics to make your decision and make your handle into that herd. Then, once you're on the herd, then you can do some on-health plans. What is actually the status? There was a Japanese yesterday where he had made some biosecurity risk assessment. I think that sounds like a perfect idea. Do something so that you can find out what is the weakest spot, and don't just do what you're already good at. Improve where you're really not doing a good job. And then is vaccination. So improve your husbandry, your management procedures for disease prevention and control, including vaccination. This is where vaccination comes into part in terms of fighting antimicrobial resistance. There's so many other things that need to be in place, and then we can use the vaccine to really improve what's happening. But vaccines won't work if there's not a health plan into that herd. It may work against erysipelotrix, but many of the other things, it probably just won't do it. And then, of course, that's happening at least in Denmark. Now they're trying to make antibiotic-free swine production, trying to sell that at a higher price. So trying to do everything they can as a farmer to grow pigs without antibiotics. Whenever an animal has to be treated only individually, it's taken out of production and put into a different place so that they can sell these feeds to the public at a higher price. Of course, we're always looking envious to Italy and Spain that are selling ham at $1,000 a kilo sometimes, just because they cure it a little bit. So that's obviously, the farmers, the consumers will pay if they get a product that they like. And can you use alternatives to antimicrobials? We have done a lot, not me, but others have done a lot in terms of looking into Chinese herbal medicines. Of course, nowadays probiotics and changing the feed to promote different microbiome constituents. It could be a really nice way to favor, for instance, anti-Lawsonia microbiome. But it's still very difficult to use that in any national strategy. And if you look at this, most of these are national, right? So there's some on-farm things where you can go in there, but unless you have the support from the national authorities, somebody has put in a policy to actually do what you want to do, it's not going to work. You need to have that country working together, and then actually I think it's really feasible, and we can worldwide reduce antimicrobials a lot. Of course, it's bad for the sponsors that are selling antimicrobials, but those sponsors that are into vaccines, the vaccines will only work better if you have a higher vaccine health level. So conclusion, vaccines work great if you know what to vaccinate against. So make sure you know actually what's happening. You need to know how to vaccinate. You need to instruct your farmers how to vaccinate if they're doing it, and I guess they are. They need to handle the vaccines appropriately, and you need to tell them how to do it. What is the schedule? When are we going to do it? How does that actually fit the best way? Then you need to have a good general hygiene, and then the veterinarian must be an educated, active consultant, right? You can't just do it once and then expect that they will go. You need to follow up. You need to, are you actually doing what we agreed upon? How can we do together? Can we maybe make some education sessions for all the farmers in the herd? Stuff like that to get you involved. And I think that's really important to make a good antimicrobial action plan. So yeah, reduction of antibiotic use is a national task, but eventually you're the one that's going to do it. So I want to thank Lars Erik Larsen, against a virologist and vaccination guy at my institute, for some inspiring talks about this, and then for some of the slides on the actual handling of the vaccines in the herds from Preben Mortensen. So, ready for the IC translation?
Yes, I am. Yeah. Yeah. All right. Thank you so much, Dr. Jankersen. Yeah. โอเคก็สรุปสั้นๆนะครับอย่างแรกเลยคือว่ายีนดื้อยาเนี่ยมีอยู่ในแบคทีเรียตั้งแต่สมัยดึกดำบรรพ์แล้วนะครับแต่เพียงแต่ว่าการใช้ยาอย่างหนักหน่วงที่เราทำกันที่เราทราบกันว่ามีการใช้กันจะเป็นตัวช่วย select ให้ยีนดื้อยามันคงอยู่ในแบคทีเรียไปเรื่อยๆอะไรอย่างเงี้ยนะครับเราเลยต้องช่วยกันลดการใช้ยานะครับและแน่นอนวัคซีนไม่มีผลโดยตรงต่อการควบคุมยีนดื้อยาเหล่านั้นนะครับวัคซีนเพียงแต่ช่วยป้องกันโรคที่จะนำไปสู่การป่วยและนำไปสู่การใช้ยาอีกทีหนึ่งแต่ว่าวัคซีนจะส่งประสิทธิภาพสูงสุดได้เนี่ยเราต้องใช้ถูกต้องใช้ถูกโรคใช้ถูกโปรแกรมในฐานะสัตวแพทย์ทุกคนเนี่ยก็ต้องโปรโมทต้อง educate ฟาร์ม educate ทุกท่านให้ใช้อย่างถูกต้องนะครับซึ่งมีดีเทลจุกจิกเล็กน้อยมากมายแต่ผมเชื่อว่าสัตวแพทย์ทุกคนเนี่ยมีความเข้าใจกันอยู่แล้วแต่ก็ต้องช่วยส่งเสริมการใช้งานอย่างนั้นให้มันดีขึ้นนะครับแล้วก็การควบคุมการใช้แอนติไมโครเบียลเนี่ยมันเป็นทั้งระดับชาติและระดับฟาร์มในฐานะสัตวแพทย์ควบคุมฟาร์มเนี่ยเราทำระดับฟาร์มได้ระดับชาติก็ต้องช่วยกันส่งเสริมให้มีการควบคุมเป็นเชิงนโยบายต่อไปนะครับโอเค ready for question and answer any question from the floor okay
Your answer for my question in the morning session is very wide range, that it make us to make decision to do that for ourselves. So I have one question. Is the situation that I always found when I travel along Asia? Is the vaccination in young piglet less than 3 weeks of age? There are many veterinarian or even professor in university advise the farmer to do vaccination first week, second week, or even somebody said that before 7 days old. What do you think about this?
I haven't done any research in this at all myself, so I will have to talk about my general immunology knowledge, and actually Bill and Sani could also chip in if they want to. The piglet is fully immunocompetent when it's about 8 weeks old, so we know that it does take some time before it can respond fully. And particularly in my experience, we started this with Lawsonia, that the cell-mediated immune response is not very good in the beginning. So they can develop antibodies. I believe they can develop antibodies even as a foster inside the mother, but typically they won't do it because they are not challenged. So the immune system is not challenged to do that. What happens when they are born? They can respond to a vaccine. They will, I think, respond better with making antibodies. And the immune system, I think the evolution of the immune system is so that, of course, you can for specific infections, you can get tired down, you get energy. But most often, the immune system can cope with really, really a lot, because it's so important for our survival. So if you think of a newborn baby or a newborn piglet, they will immediately meet things in their environment. And of course they are relying on antibodies from their mother against known pathogens, but it's not like the immune system won't function until they're later on. It may challenge them a little bit, and the PCV2 example is of course a really good example of suddenly something that doesn't really play together. And maybe early vaccination was part of the PCV2 PMWS outbreak. But now that that's under control, and if they get antibodies against PCV2 from the mother, then that won't be a disturbing factor. And I can't say that vaccines won't work. I can only say that you'll probably get a better effect if you wait, but if it's too late, then you better get started early. And the immune system can cope with it, but it will cope more efficiently with it when it's older. That would be my answer. And the two experts are nodding a little bit.
โอเคครับก็คือพี่หมอเบสถามว่าบางที่นี่มีการทำวัคซีนตั้งแต่ลูกสุกรอายุต่ำกว่า3วีคบางที่ต่ำกว่า1วีคอีกก็เลยถามดรจุงกุนซันว่าคิดว่าวัคซีนจะเวิร์กไหมใช่ไหมครับจุงกุนซันบอกว่าคือลูกหมูเนี่ยระบบภูมิคุ้มกันจะ develop เต็มที่เมื่ออายุ8สัปดาห์ขึ้นไปแล้วก็นั้นเราเลยหวังว่าคือถ้าเราทำวัคซีนเร็วเกินไปก็อาจจะเวิร์กหรืออาจจะไม่เวิร์กแต่คือเขาตอบไม่ได้เพราะว่ามันต้อง evidence base ใช่ไหมครับยังไม่ได้ดูศึกษาตรงนี้โดยตรงแต่ว่ามันคงดีกว่าถ้าเรารอให้ภูมิคุ้มกันมัน develop เต็มที่แล้วทำเพื่อได้ประสิทธิภาพสูงสุดและระหว่างนั้นเราคือเรา rely on ภูมิคุ้มกันที่ได้จากแม่ผ่านทางนมน้ำเหลืองแทนแต่ก็มีบางโรคเช่นกันเช่นใน PCV2 เนี่ยทำเร็วก็เห็นประสิทธิภาพเร็วแต่ถ้ามันคุมได้โดยการทำวัคซีนในแม่แล้วก็ไม่มีความจำเป็นต้องทำเร็วต่อไปมันเป็น case by case ด้วยอย่างเงี้ยครับแต่ตอบไม่ได้โดยตรงว่าจะ work หรือไม่ work อะไรเงี้ยนะครับมีคำถามอีกไหมครับ
Well, this is not my questions, but I've heard about this question so many times, so maybe I ask you for your opinions. A lot of people in these regions are talking about using the autogenous vaccine for controlling bacterial infections in the farms. What is your comments on that, or do you have any experience in Denmark? If they've been using like that, something like that?
Yeah, we actually used to make autogenous vaccines at our institute, but then the regulations became so they wanted to have it GMP manufactured, which means you can only make one vaccine at a time in every unit, and that means that you can only make one autogenous vaccine at any given time point, and that takes a couple of weeks, so that was just completely unfeasible. So we had to stop, and now we're sending samples to Germany to a company where they are doing it, and they have less strict regulations. They're using it particularly in chickens, also a little bit in fish, and also some pig production. I know that's been used a lot in many of the Eastern European countries, and actually also in the Netherlands a lot more than what we are doing here. I like it a lot better than the backfeeding, but that's because there's some kind of control in it. I'm really not too much in favor of this cannibalism thing that you have to do with PED. So I really hope that they will get a vaccine working at some point. And autogenous vaccines, I mean, if you haven't got anything else, it's for sure better than nothing.
But if you already have the vaccine, some other vaccine in the market, do you think that's necessary to go for the autogenous vaccine?
If the vaccine doesn't work, if you have vaccines like in Japan when they said that the vaccine was developed 20 years ago against a strain that's now completely shifted because they had a reintroduction from the States, obviously you need to do something else. So that's up to the companies to make sure that they also follow the diagnostics. I mean, nowadays you can sequence anything very quickly. So I think it's really important that the diagnostic laboratories, they monitor what is the development. And there was a recent development also in some new, was that a new strain, I think, of either PCV or PERS, where there was some mutations, and that was actually sitting right in the diagnostic primers that we used in our lab. So very quickly they had to adapt that. So you're monitoring everything. You need to be on your toes, but you also need to find out, are the vaccines actually working against those that we have at the moment? And if not, you need to go into dialogue with the companies. That would be nice then to have your own production facility so that you could relatively quickly do it. But it's not easy for the companies either to change a vaccine, because it is one project. You can't just change the adjuvant because it's considered one project. You need to go through the whole registration every time. And that is not so easy, but if you're losing the market because it doesn't work anymore, you need to do something.
ก็สำหรับคำถามที่เป็นคำถามยอดฮิตนะครับก็คือว่าเราคิดยังไงกับการใช้ autogenous vaccine นะครับก็คือคำตอบง่ายๆก็คือเมื่อก่อนProfessor Jungersen ก็ใช้แต่ว่าหลังๆมาใน regulation ของเดนมาร์กคือต้องเป็น GMP นะครับก็คือผลิตได้แค่หนึ่งวัคซีนต่อ1ช่วงเวลาก็เลยไม่ practical ที่เดนมาร์กต่อไปก็เลยมีการส่งวัคซีนเนี่ยไปทำที่เยอรมนีแต่ว่าหลักคิดง่ายๆก็คือว่า ถ้าวัคซีนในท้องตลาดมีแล้วมันมีผลการทดลองที่ดีกว่ามันก็ดูน่าจะดีกว่าออโตเจนัสวัคซีนถูกไหมครับแต่ถ้าไม่มีอะไรเลยก็ใช้ตามสถานการณ์นะครับแต่ว่ามันก็เป็นหน้าที่ของ diagnostic lab ด้วยเช่นกันที่จะคอยอัปเดตข้อมูลต่างๆทุกวันนี้เทคโนโลยีในการซีเควนต์มันเร็วมากแล้วเราสามารถอัปเดตข้อมูลได้ตลอดเวลาอะไรเงี้ยแล้วก็ไม่ใช่เรื่องง่ายที่บริษัทวัคซีนจะเปลี่ยนวัคซีนภายในเวลาแบบปี2 ปีอะไรเงี้ยมี regulation อีกมากมายนะครับโอเคครับมีคำถามอะไรอีกไหมครับ any other questions okay
You highlighted that antimicrobial reduction is a national task. Of course, you will have different players within that area. You would have the pharmaceutical companies providing the antibiotics. You have the veterinarian or the practitioner prescribing. You also have the farm endorsing the use and purchasing. And then within these 3 players, you have a referee, which is the regulatory authority, I would say, enforcing the rule. Could you share with us more insight in terms of how it works, this system of yellow card? What are the accountabilities of the farmer, of the vet, of the regulatory authority? In practice, how it operates?
I'm not an expert in that at all, but the way I understand it, it is strictly a veterinarian. They were trying to sort of get rid of what we call the highway veterinarians, those that are being able to inspect 10 large pig herds a day over a stretch of 400 kilometers, and that is just not. And they were prescribing antibiotic everywhere. You need to go into the herd, so they wanted to not make it the farmer's decision whether they should have antibiotics and make it more the responsibility of the veterinarian. And some veterinarians could make a lot of money because they were actually selling the drugs as well, so they get a little bit of a percentage. So they took that away. So veterinarians are not pharmacists anymore in Denmark. You need to give them a prescription and then they buy it somewhere else, because a lot of veterinarians actually were making money on how much antibiotics they were prescribing. That's of course the one problem. But nowadays they are also actually facing restrictions if the farms that they are supervising are using more antibiotics than they should, or than the national average. And I'm not too sure how it's implemented. I actually think it's described a little bit in that European report, the RONAFA report in there. Otherwise, I can find something I can send you. But it is very much putting a responsibility on the veterinarian to be what I would say a good veterinarian, somebody that's actually using his education and not just his right to prescribe antibiotics to treat infections.
ก็สำหรับคำถามของคริสเตียนนะครับก็คือแน่นอนการลดการใช้แอนติไบโอติกเนี่ยมันเป็นเรื่องระดับชาตินะครับซึ่งก็ต้องมีผู้เล่นมากมายมีทั้งสัตวแพทย์มีทั้งเจ้าของฟาร์มมีทั้งบริษัทยามีทั้งผู้เล่นอีกมากมายรวมทั้งมีเจ้าหน้าที่รัฐที่ต้องคอยกำกับดูแลการควบคุมยาให้ถูกต้องด้วยและทีนี้คือโปรเฟสเซอร์ยังกระเส้นได้พูดได้ว่าในเดนมาร์กเนี่ยมีกฎเหมือนกฎให้ใบเหลืองใบแดงเหมือนในฟุตบอลนะครับว่าถ้าใช้ยามากไปต้องโดนใบเหลืองพักงานอะไรอย่างนี้หรือเปล่าก็เลยถามว่าดีเทลของเรื่องนี้เป็นยังไงบ้างซึ่งโปรเฟสเซอร์ยังกระเส้นไม่ใช่คนทำงานเรื่องนี้โดยตรงถ้าอยากรู้รายละเอียดมากกว่านี้ให้ไปอ่านในรายงานฉบับนั้นที่โชว์ให้ดูในสไลด์นะครับแต่หลักการง่ายๆคือเราต้องตัดตอนเหมือนกับว่าคนมีอำนาจจ่ายยาอาจจะไม่ใช่คนที่ต้องขายยาโดยตรงเพื่อให้เขาไม่สามารถแบบมือนึงล้วงกระเป๋ามือนึงจ่ายใบสั่งยาอะไรอย่างเงี้ยครับนะครับก็คือสั่งยาฟาร์มไปซื้อที่อื่นแล้วก็จะมีการควบคุมว่ามีโควต้าว่าฟาร์มหนึ่งใช้ยาได้เท่านี้ใช้เกินกว่านี้มีกฎลงโทษมีข้อบังคับอะไรอย่างนี้แล้วก็หลักการอีกอย่างนึงคือมันจะมีเหมือนกับภาษาคือ highway veterinarian นะครับเหมือนกับสัตวแพทย์มือปืนที่แบบวันหนึ่งวิ่ง400กิโลเข้าฟาร์ม10ฟาร์มอะไรอย่างเงี้ยซึ่งที่เดนมาร์กเนี่ยมันจะลิงก์กับการที่เขาเป็นคนขายยาได้โดยตรงอะไรเงี้ยก็เลยพยายามกำจัดเหล่านี้ออกไปเพื่อลดการใช้ยาอย่างไม่ถูกต้องด้วยคือทำไงก็ได้ให้การตัดสินใจที่ใช้ยาเนี่ยอยู่ที่หมอไม่ได้อยู่ที่ฟาร์มเมอร์ไม่ได้อยู่ที่คนขายยาอะไรเงี้ยนะครับหลักการในภาพกว้างนะครับจะดีเทลเดี๋ยวไปดูใน regulation อีกทีหนึ่งนะครับมีคำถามอะไรไหมครับ Yeah I think that's us Yeah Thank you so much Dr. Jungersen Very impressive talk. So So that's us. That's the end of this symposium. Thank you so much for bearing with us. It's quite intense in some topic and quite general in some topic, but I hope like all together you get the big picture of how we should go forward for swine vaccine development. Thank you so much for your attention. Thank you so much.